Good morning, everybody. You might be surprised I'm not Erin. Welcome, and I hope you enjoyed last night's celebrations. I was impressed with some of the historic field gear, but some other people look like they doing field work in the city. You know. Welcome to this discussion this morning. I thought that I'll just give a quick rundown on why we called this a breathing landscape, and that we're going to have one or two little discussion groups. We were really interested in, given where sand parks are shifting away from working much more beyond the park boundaries, that it may be useful to have a bunch of discussions and then utilize that to, as a group, have about 5 minute or 10 minute discussions. Firstly, when the herbivore people finish, we're going to have Kensani Nkuna, who will do a little bit of a facilitated discussion and asking, well, how do herbivores contribute? Um, then we will have the carnivore guys, and then Dan will help us to do the same. And before tea, we'll have, I think, four other talks on elephants, and Rob Hildemont will help us too. And then after tea, there's a couple of more presentations, and then we'll have a slightly longer discussion, and we're going to ask this question: what should Kruger look like, or should we actually ask another kind of question? And Lisa LaRue will facilitate that for us. So just keep those things in the back of your mind when you're listening to what everybody is saying now. Think a little bit about that. So thank you very much, and I'm going to hand over to Erin to take over and run the show. Thanks.
Thanks, Sam. Welcome, everyone. So the first presenter today is going to be Manintle. She will be presenting on the breadth of the savanna soil. Take it up.
Good morning, everyone. I'm so glad to see such a great turnout after last night's gala dinner. Um, so, do I just click next? Oh, there we go. So, when we talk about ecosystem breathing, what we're actually talking about is soil respiration. Oh, sorry. When we talk about ecosystem breathing, what we're really talking about here is soil respiration, daily respiration, seasonally. And what we've been hearing a lot about this week is how savannas are heterogeneous or patchy. And this heterogeneity is important, even whether it's random or clustered or organized, because it's key to understanding how carbon moves through the system. And carbon flux is structured by vegetation, soil moisture, as well as biogenic structures, the things that organisms build within the environment. And among these biogenic structures are termite mounds. So I just wanted you all to have an idea of what I mean when I speak about termites and termite mounds. I took this video in the field, and hopefully you guys can see the termites, Macrotermes natalensis, in the mound over there. So ecosystem engineers, that's what termites are considered as. They're not just inhabiting the soil, they're actively modifying the soil texture, porosity, hydrology, as well as the nutrient distribution through the construction of their mounds, as you can see in another video that I got from the field. So what emerges from this is a landscape that has distinct patches, nutrient-rich soil surrounded by not-so-nutrient surroundings. So termite research is almost as old as Kruger is. It's about 90 years old, and it can be separated into three distinct phases, starting with the foundation phase, where studies such as those conducted by Alan Jones and Dangerfield et al. that looked at carbon cycling, soil fertility, as well as just ecosystem engineers as termites in fungus farming termites. Then in phase two, we move on to a mechanistic understanding of termites in terms of what they do within their mounds. So the study by Jamali et al. Looked at the diurnal or the daily flux of methane from night and day, as well as seasonal flux. And this study by Noya in 2018 is actually quite interesting, where they started looking at the microbial communities within the termite mounds and found that at around about 50% of the gases that termite mounds produce in terms of methane is actually mitigated by these methanotrophic bacteria before they're emitted out into the surroundings. The final phase, which is where we currently are with termite research, is in just understanding how termites select for, if they do, the surrounding microbial communities and how they create distinct microbial patches within their mounds, which is what I'm interested in. So a lot of these studies have looked at either environmental factors, microbial factors, or the structure of the mound itself in isolation. And so we don't have studies in sub-Saharan Africa that actually combine all three for a specific species, which is what I was interested in. So I went and I investigated how Macrotermes natalensis, a fungus farming termite, regulate carbon flux across the wet and the dry season, as well as the soil chemistry associated with it and the bacterial community organization. So I did this by going out into the field and looking at gas flux using a Lycor 8100 system that measures gas flux on the mound and surrounding mounds seasonally. I also conducted soil chemistry, looking at basically the normal suite that you would look at of physical chemical properties, your soil organic matter, soil moisture, pH, electroconductivity, and a handful of macro and micronutrients on the soil compared to the surrounding soil. I then also extracted DNA for 16S sequencing that we conducted at a neighboring university. The study was conducted at Wits Rural, which is a semi-arid savanna. We've been hearing a lot about those. The annual rainfall here is about 650 millimeters, and it is abundant with Macrotermes natalensis. And I want to be transparent from the start. So this study is a focused data set. I have 4 termite mounds, so I'm not making continental claims. I'm using this as a clear window into the mound scale processes that matter for how we measure and interpret savanna soils. So this slide might be overwhelming in the beginning, and this is our results for carbon flux, but bear with me here. We have the on-mound fluxes on the right and the off-mound fluxes on the left. And what you'll notice here is that in winter, instead of the flux on the mound going down, it actually increased. And our surrounding soils, we see what we would expect to see, and that there's a decline in soil respiration or carbon flux in winter as opposed to summer. And also in the summer season, we notice that there's no significant difference between the wet and the dry, so the flux in the environment was the same. And then on to the environmental drivers. So these have been identified in literature as well as in my study, that soil moisture and soil temperature positively contribute to carbon flux on termite mounds. What we'll notice is that in the summertime as well as the wintertime, the termite mounds were able to decouple their environment and maintain soil moisture throughout the season. In terms of temperature, there wasn't much of a difference on versus off the mound in terms of the summer as well as the winter, although there was obviously the normal shifts in temperature that we would expect between the two seasons. And so in the winter, what I did to understand the spatial dynamics of flux, I added a third measurement on the mound, or a second measurement on the mound for a total of 3. So I added a measurement on the chimney, which is the opening of the termite mound. So I obtained these results. So the blue and the green you're already familiar with as on versus off the mound, but the purple measurement that you're seeing there is the gas flux that's from the chimney. So an interesting image that starts to emerge is that termite gas emissions or carbon dioxide emissions are not just passively being emitted through the mound, but are being directed through a singular channel, that is the chimney. Onto the mound enrichment, what we found is that on these mounds there was an enrichment of copper, potassium nitrate, soil moisture, as well as zinc and electroconductivity. Something worth mentioning, even though it wasn't significantly different, was that the pH on these mounds tended to be more neutral than the surrounding soils. So just a summary of what we found for the physicochemical properties, specifically these elements, as well as elevated moisture, EC, and a pH shift towards neutral. Onto the microbial communities, what we found was that there was not much of there wasn't a significant difference between the diversity of the mounds versus the surrounding soils, together with the richness, which isn't uncommon. It's difficult to develop a clear picture of the microbial communities at this point in time because a lot of the research that's coming out now is new, but there are some studies that have been done on fungus feeding termites that have found this exact result. But what I was more interested in, which I'll get to a bit later, was the actual communities themselves, because the richness will speak to the number, but we're also able to understand what types of communities exist on the mound, even if what we're seeing there is stability. So after modeling and putting in a PCOA, what we find is that some of the physicochemical properties or the nutrients in the soil are drivers of this diversity. Onto the dominant lineages, what I was speaking about 2 slides ago, about how the communities might look like they're the same, but we have different taxa that are enriched on the mounds. Specifically, for the sake of time, I only have 2 here for you today, so I'm going to try and pronounce these names properly. I've been practicing. So the Sphingomynadelase of the phylum Pseudomonata is specialized in the degradation of complex recalcitrant matter, which we found to be enriched on the mound. So it has the potential to modulate trace gases that are associated with termite activity. So we found this on the mound, but not significant. It was significantly different on the mound versus off the mound in terms of taxa. Then we also have Calotenuales, which is of the phylum Chloroflexa, which is a key player in carbon cycling within the enriched mound environments, and it's also associated with carbon dioxide and methane oxidation pathways in the soil, which I thought was quite interesting in that even though the community might look stable, but what we have is somewhat of a change in the taxa specifically, which would be interesting to look at if this was put together with your community level physiological profiling through ecoplates to actually look at the metabolic activity of these communities seasonally, which unfortunately I wasn't able to do, but hopefully we'll be able to do soon. And so if we're putting this all together, what we find here is that there is an integrated picture. Termite mounds modify the soil structure and concentrate nutrients, moisture, and pH. They also filter microbial communities and influence both carbon production as well as gas transport pathways. And in this way, the mound regulates both biogeochemistry and the physics of gas exchange. So what does this mean for ecological implications? The Natalensis mounds function as persistent biogeochemical hotspots, which is what I found in my study, and it thus makes it very important that when we're looking at carbon budget studies, that termite mounds need to be factored in more. I think Kate mentioned that earlier this week in her talk. And we also finally need to consider the spatial resolution and understand that in termite mounds, we're not just dealing with passive emitters, because a lot of the research in the early 1970s to late 1990s was more focused on trying to understand how much gas flux is emitted from these mounds, and it made for a very negative, I guess, perception of termites in an anthropogenic scene where we have climate change. And so understanding that that termites are not passive emitters, but they're modulating gases within their mounds as well as structuring gas exchange. Thank you.
Thank you. We don't have any time for questions, but we will have those discussion sessions. So if you do have any questions and you also know now what she looks like, please catch her afterwards. The next presentation will be by Joris. He will be talking about living grasslands. Thank you.
Thank you. Good morning. I'm not going to talk about animals. I'm actually going to talk about people and animals and plants. So I'm not sure if Louise is still here, but if there's still prizes, then I'm here as well. Let me start with saying that I'm talking on behalf of quite a big team. So this is the team here. So it's actually a combination of natural and social scientists, and it's also, you will see several non-academic people in there. So it's really a transdisciplinary team of people, and we are running this program together. And the context of this program is this concept of nature-based solutions, which is about harnessing and restoring ecosystems to address environmental and societal challenges. We actually prefer to use nature-based opportunity or nature-based approach to emphasize that there are actually very few solutions for many of our problems, because many solutions come with trade-offs. So there's solutions, but that solution will come with a trade-off in relation to some other parameter. So then we became interested in this concept of wilding or rewilding, which is increasingly highlighted as a nature-based solution. And rewilding is a form of ecological restoration that emphasizes autonomy, so autonomous ecological processes, and restoring autonomous ecological processes. We also prefer to use the term wilding instead of rewilding because we want to emphasize like a forward-looking opportunity and also not necessarily the recreation of something of the past. So rewilding in the European context is basically this, so restoration in Europe, especially grassland restoration, it's about lawnmowers, interestingly. It used to be a lot about lawnmowers. This is slowly changing, it was quite quickly changing, fortunately. And these lawnmowers are now being replaced by grazers. So this is wilding or rewilding in European context. This also emphasizes that wilding is about steps. It's not necessarily about creating wild nature out there. It's about small steps to restore autonomy of nature, so autonomous processes. So if you think about the local context, in the South African context, you can, for example, think about restoring fire regimes or bringing fire back into the landscape as one autonomous process that's heavily under influence of human control, of course. Another one, if you think about small steps, could be restoring wilder grazing dynamics. So this idea, for example, that Herding for Health is very active in this field, which is about moving from continuous grazing to a more rotational type of grazing approach. This is, of course, already very old. It has been ongoing for a long time. So important then if you think about wilding also, is that it's not just, or it's actually mostly not about protected areas. Wilding is very much about restoring autonomous processes in the landscapes outside protected areas. So this is an example of in this case, I think this was a Dutch landscape, and you can see it's really about restoring tiny processes sometimes. So basically leaving a small piece of grass aside, so that plants can actually come back and come. In this case, forbs can recolonize. So it's really about finding opportunities in that whole landscape to build autonomy of nature again. So this strongly responds to the protected area paradox, which is about the fact that we see ongoing biodiversity declines despite increasing conservation investments. And this is about the challenge of restoring and conserving biodiversity in the larger landscape, so outside protected areas, while also actually acknowledging the enormous challenges in these landscapes, such as human livelihood challenges. So this then again links very closely to Sandpark's new vision, 2040 vision, and I think this concept of mega living landscapes was already mentioned earlier in the week, which is about reimagining how people can conserve biodiversity on larger scales and in a more inclusive manner. But how do you do this? So that's still a big question, of course. Many people actually have already talked about this, especially during the people and animal section. And this also connects to ongoing and increasing critique on wilding, not just wilding, but increasing critique on nature-based solutions, in fact, as an approach. And one of the first critique points is a lack of empirical evidence for benefits beyond biodiversity restoration. For example, benefits for livelihood impact, but there's a whole series of potential benefits that these solutions promise. The second critique point is this suggestion that dominant rewilding narratives exclude humans and that they promote a separation between humans and nature. And this then links to the third critique point, which is about this imbalanced power relations and decision-making, leading to doubts whether rewilding or NBS in general can actually offer inclusive opportunities for rural development. And if you then scale that up, you can also link this to biopolitics and this increasing concern of rewilding being perceived as a form of neo-colonialism. Well, if you think about that human-nature relation, if you look at, this is just some images of the future of Europe, rewilding in Europe, and you do see that many of these images you have to look very, very hard to see a human in these images. So it's basically wild landscape with lots of animals, nice rugged landscapes, big rivers, but very, very few humans. I think there is actually a village there, so there's some humans in that picture. So our main research questions for living grasslands are basically how do you actually develop wilding in an inclusive way? So how do you develop wilding scenarios in grassy ecosystems in a way that's inclusive of the people living in that landscape, building on their needs, visions, and knowledge while respecting their plural values of biodiversity. And then also the second important question is about these trade-offs. So acknowledging that there's no simple solutions, but you actually always have to analyze the trade-offs and synergies between different sustainability challenges. So we are working on this. We are only starting this program, so this is really an introduction of our program and also a call for collaboration. So we're really keen to collaborate with anyone. So this project is based in the Umzimvubu catchment. It's an interesting catchment. It's the third biggest catchment of South Africa. It's also an undammed catchment. It's in the northwestern part of the Eastern Cape. We focus mostly on the upper catchments, so along the Lesotho border. And it's interesting as well because it's a mix of private land and especially community-owned grazing lands. But there's also some provincially protected areas. For example, Ongeluksnek is in the area. And then you have actually new landscapes initiative that makes it also very interesting. One of them is the new Grasslands National Park that's being proclaimed. And then this is one of the suggested mega living landscapes areas as well. And on top of that, there is also a building protected environment in that landscape. So you have all these conservation initiatives mixed within these different land uses. So we thought this is a great place for a project like this. And especially also because we work closely with these two NGOs. These NGOs are part of our team, Conservation South Africa and ERS. And they've been working in this landscape for many decades. So we have 3 core objectives. The first objective is to assess the local knowledge, so what biodiversity means in the eyes of the people, and also what different values they associate with various components of biodiversity. So then we will start monitoring biodiversity that the people have indicated, have highlighted, through scientific methods, and we'll be setting up participatory monitoring hubs across this catchment, where community members and scientists jointly monitor biodiversity and associated livelihood values. And then we will combine these two types of knowledges to integrate and try and integrate them to then together actually co-create scenarios for biodiversity restoration. So that's in very brief the 3 objectives of our program. I realize it's ambitious. We start with 3 years, but we hope to be in that landscape for much longer. So our plan is to build on that. And this is just like sort of a conceptual vision. So we basically have these 3 central work packages, like an indigenous knowledge work package, a scientific knowledge work package, that both feed into this co-creation of scenarios. But the co-creation of scenarios then also feeds into these 2 work packages. So they continuously shape each other, so to say. That's the vision for the project. So by doing this, we hope to sort of add to the ongoing work in terms of how do you actually do conservation in a more inclusive way. And as Mariska already mentioned, we are looking hard for still 3 postdocs. So we still have 3 postdoctoral opportunities in this program, 2 social scientist opportunities, and also 1 ecological scientist. So please come and find me if you're interested. And that's 16 seconds. Yeah.
Thank you so much for such a great presentation. Our next speaker is not going to be Rachel, it's going to be Adrian. He will be talking about familiarity, maternal tolerance, and aggression shape long-term affiliations in black rhinos. Thank you.
Okay, fantastic. Thank you. Yeah, so sorry, Rachel was supposed to be here and give this talk, but she's stuck in the States, and so I'm going to have to do a little bit of talking about her postdoctoral work that she did with me. Okay, so let's talk about basically solitary species. So solitary species, one thing that we know about them is that they mainly, if we had to define them, they mainly forage and sleep alone. However, They do have social relationships, but these tend to be very rare and simple, things like courtship and breeding. But some recent studies that have been working on some of these have found that actually some of these species can have really complex social behaviors, but those social behaviors are shared with individuals that they know quite well. Right? So let's talk about what one reviewer called the poster child of solitary species in Africa. And I know leopard people will probably deny this, but black rhinos. Now, one thing, if we see black rhinos, we predominantly see them on their own, but we do see them in groups of 2 to 3. In fact, in one case, if you look in literature, there's a group of 13 that's reported. How stable this group was, I don't know, and I doubt it's very long. But okay, so there is some degree of social. But the other thing we know about black rhinos is they're aggressive. So much so that death from fighting can be relatively common in some areas. So here you have 2 opposing type of situations. One, individuals want to be together, and the other, they want to kill each other. So how do we, this raises a couple of really cool questions. What is the role that familiarity plays in the social associations of black rhinos? And secondly, do these associations ultimately influence their survival? Does this affect their possibility of mortality? All right, so how did we explore this? Well, we worked with WWF South Africa, and we used their Black Rhino Range Expansion project. We worked in 14 reserves in 4 provinces, you can see there, and then 2 countries, 1 province or 1 reserve in Malawi. This gave us, well, for each of these parks, they had black rhino for somewhere between 3 and 20 years, and we had 69,920 observations of individuals, and 499 known individuals, right? So this is a massive sample size. We took these 499, we could divide them into age and sex classes, calf, which is less than 2.5, subadult from about 2.5 to 7, adults about 7. Okay. So, how do we look at these associations? Well, we used a pretty standard metric. This is the half weight index. The nice thing about the half weight index is that it deals with imperfection in the sense of you don't have the same number of observations of every individual, and also it deals with all the ones that you have in your data, the single observation of lone individuals. Right? We then took a look, because of the fact that all these different sizes had different population sizes, all these different sites, we then used a normalized degree where we took the number of associates that an individual had and divided it by the population of that site. Right? And this allowed us to compare across sites. We looked at age difference in these associations. We also looked at the sex ratio. I'll show you that we did this in relation to proportion of males. And we explored all this using linear mixed effects models. What about mortality? Well, mortality, we looked at natural causes, disease, accidents, old age, predation. We didn't include poaching, we didn't include removals for translocations, and we wanted to see what the actual natural aspects were. We looked at association rate and how that influenced mortality, the number of associates that an individual actually had over this time period, the sex of that individual, and then we explored these using Cox proportional hazard models. So what do we find? Well, first thing that jumps out, you took a look on the y-axis, is the proportion of sightings. This is group size on the x. And what you can see is that, well, just under 70% of all the observations are alone individuals, thus kind of giving this idea that these are solitary species. However, over 30 were found in groups from 2 all the way to 7, and these groups of 2 were not cow-calf pairs, right? So these are different individuals, not related in that regard. If we explore the association rates, and this again, this is the half weight index here, you've got calf, subadult, adult, male in blue, female in red. What you see is that calves and subadults associate a lot, all right? And basically with the adults, well, look at that. The females associate more than males. Okay, so this will come, you'll see this pattern coming up a lot. Okay, so that's association rate. What about the number of associations? Well, what you see is that calf, subadults, and adult males are all pretty much the same. All right, they associate with the same number of individuals, but look, what do you get again? Adult females. Now, adult females, the reason we're seeing this is that they're associating with subadults, they're associating with other females and exposing their calves to other calves. All right, so we're seeing these type of interactions there. What about the sex ratio? Well, the sex ratio, like I said, we took the number of males divided by the total number of individuals. So as you go along the y-axis, the closer you get to 5, it's more male bias. But what you can see basically is with calves and subadults, males associate with males predominantly, females associate with females. Right? But then you get to adults and they start to associate more. This is courtship, this is breeding. So we're seeing more of adults starting to interact. What about age difference? Well, surprise, surprise, calves tend to associate more with individuals that are different age than they are, but subadults tend to hang out with themselves, and adults generally tend to hang out with themselves, but once again, Females, because of their associations with subadults and females with other calves, we're seeing that they actually associate a bit more in different ages than adult males. So understanding all these individuals and understanding their associations, what does this actually mean? Well, if we take a look at subadults that are associating, if you see two subadults that are associating, the reality is they probably knew each other as calves. More often than not, they did. But what about adults? Well, we see a similar type of pattern where adults that are moving together predominantly knew each other as subadults. Right? This is probably a way of reducing aggression if you know the individual. But what we don't find is this. We don't see a situation where if you knew each other as a calf, you're going to know each other as an adult. Why? Well, what happens at the subadult phase? This is the dispersal phase. So even though you grow up in an area and you interact with other calves that are there, you get to subadults, you start to hang out, well, then one individual disperses that way and the other individual disperses that way. So the probability of being in the same area with adults that you knew as calves is pretty low. Okay? But what we're seeing is familiarity plays an incredibly key role in these associations, probably again to reduce that aggression. What about survival? Well, what we see is that if calves, the more individuals that they associate with, and this is again determined by their mother, the higher the risk they have of having an aggressive interaction with an adult male or a subadult. They're susceptible, right? What about subadults? Well, we expected, just like with white rhinos, that they would associate with each other, you act as social shielding, they'd have less aggression, no impact whatsoever. Okay? But what about adults? Well, look at this. Surprise, surprise, the more you associate with males, the greater the chance of mortality. Males are aggressive. These are the individuals you don't want to be around. Different ages, the more that you associate with different individuals and different ages, the lower your risk. And this is, again, reducing the probability of you interacting with an aggressive male, right? Males, however, have lower risk of mortality than females. Why? Well, take a look. Remember, females have a lot of associations. They associate with a lot of individuals. They interact more with males, and so the probability of interacting with an aggressive individual is greater because of that. Okay, so what does this all mean? What are the take-home messages of this? Well, the key thing that we're seeing with black rhinos is that associations are driven by familiarity. Again, probably to reduce that aggression. If you know that individual and you know they're not aggressive, you're more likely to hang out with them. Okay? When we take a look at with calves, association can influence the survival of calves and adults. Calves, the more that mothers expose their youngsters to a broad range of individuals, and the more that adults interact with each other, the higher the probability of interacting with an aggressive individual. So why is it important to understand the behavior of black rhinos and these associations? Well, one of the things that we're trying to do, of course, is expand black rhino ranges back into their historical areas. And we do this through translocations and introductions. So what's the take-home message? If you're going to move a bunch of subadults, make sure that they knew each other as calves, because then you have a lower chance of there being aggression in the new area. The same thing with adults. If you're going to capture and move adults, move those individuals that knew each other as subadults. That too reduces the chance. And with that, I just want to thank everyone that provided data on it, and I'm happy. I've got one minute. There you go. I can take a couple questions if needed. Thanks.
Thank you, everyone. Thank you, Adrian. We do have a minute if you want to ask any questions.
Thank you. That was a really interesting talk. So you talked about the fact that if calves are exposed to more individuals, there's a higher risk of mortality. Is there any positive thing for that? Like, would there be a reason, ecologically speaking, for a female to expose their calf to more risk?
Is it on? Okay, great. Okay, cool. So probably the reason for that, and that's a great question, is the fact that if you understand and you know individuals as calves, then you move as subadults, right? And so you do have less of an interaction. If you know those individuals, there's less probability of aggression at that. So this is the thing where we think mothers are kind of, it's like play dates, right? You interact them with the nice kids, and then as they go on, they have that, but it doesn't lead to greater survival as subadults because they're dispersing. So yeah.
Okay, thank you. That's all the time we have for questions. If you do have any questions, please find him at T. Otherwise, it may come up in the discussion sessions. Our next presenter will be Richard, talking about spatial ecology to guide South African southern giraffe conservation. Thank you.
Oh, cool.
Morning, everyone. Thank you for having me. So, my name is Rick Hoffman. I oversee the Southern African program for GCF, also finishing my PhD with University of Mpumalanga. And I'll be talking about linking national abundance patterns, spatial ecology, and genetics. I squeezed genetics in there. It wasn't in the original. Apologies for that. But it's something that's very interesting that we need to talk about. So just to give you an indication of the species that I'm working on. So as of last year, the IUCN have recognized 4 different giraffe species, which we're very excited about. It took 20 years of data collection from GCF and a whole bunch of different partners to get to that point. And so I'll be working on the southern giraffe. And you can see the map here, the distribution that is circled is the southern giraffe distribution, which includes South Africa and most of the SADC countries. And you can see by the numbers on the left-hand side that giraffe are doing really well. So we estimated around 30,000 giraffe in 1990, and so that has increased by quite a bit to 2025. We've estimated just under 50,000 giraffe, and we believe this to be quite a conservative estimate in any case. So we know that giraffe are doing well. Other 3 species are also doing well, either stable or increasing. And southern giraffe are doing really well as well. So how do we know this for South Africa? So this is the question that we tried to answer. So to do this, we looked at 3 different things. The South African population estimate, where are giraffe, how many are there? Southern giraffe movement, so we looked at some GPS data for giraffe and some of the private properties in the country, and southern giraffe genetics, so subspecies composition between the South African giraffe and the Angolan giraffe, which are subspecies of the southern giraffe. So we collected data from just under 1,000 properties across the country, and we estimated at around 29,500 southern giraffe throughout South Africa. What we found very interesting was that nearly 50% of these animals were on private property, and that giraffe were distributed across the whole of the country, including some what was previously deemed as extralimitral provinces, including the Western Cape, Northern Cape, and the Eastern Cape. Some other cool takeaways from that paper was that Limpopo dominated, so this was to be expected. Limpopo is a stronghold for southern giraffe, and mostly bolstered by the Kruger National Park population. And we found that Eastern Cape led in private conservation, so we found, I think, 95% of the giraffe in the Eastern Cape were on private properties, estimated to 4,500 southern giraffe. And you wouldn't expect populations to be as strong on the western side of the country, but we found around 804 southern giraffe in the Western Cape, distributed across 90 different properties and provided to us by Cape Nature. So now that we know more or less where giraffe are, how many there are, we had to look at how giraffe are moving around on these properties. So we tagged around 40 giraffe on 6 different properties, and we looked at how they were moving on these properties, habitat, and also distance from fence, looking at the edge effects as well. So the properties were 2 in the Lowveld, 2 in Mozambique, 1 in Eswatini, and then 1 in northern KZN. So we are aware that the properties in Mozambique are not in South Africa, but similar vegetation and rainfall, we're able to compare these. These properties also ranged in size quite a bit, from 21 kilometers squared all the way up to 1,500 kilometers squared. So it gave us a nice range in comparing these properties. So very basically, we saw that the smaller reserves had a strong edge avoidance, strong habitat selectivity, so the animals were more clustered, using a central section of the reserve a lot more than the outer sections, and they used open areas a lot more than the thick areas. So I think 4 of the 6 properties that we tagged on had line presence, so this could also be an indication of that. The compressed home ranges, so which makes sense, smaller properties, they seem to have smaller home ranges compared to the larger reserves, anything over 1,000 kilometers squared. We saw a weaker edge effect, so animals were actually using the fence lines a lot more, expanded ranges, so bigger home ranges, which makes sense, more land to use, so they utilize this land, and then more natural behavior. It was sort of like an ecological transition zone that we saw, happening over 1,000 kilometers squared. The animals seem to move more naturally, more dispersed, when we compared to other giraffe data that we had. So now that we know more or less how many animals we have in the country, where they are, sort of how they're moving around on these private properties. We moved to the genetics. So I collected around 315 samples across the country that we sent to our partners in Germany to do whole genomic sequencing. And what we found was quite interesting. So you can see the light blue color over there, and then the purple color. So the light blue is the Angolan giraffe subspecies, and then the purple is the South African giraffe subspecies. So these were control properties. This is where you would expect to have Angolan giraffe. These were situated in Namibia and Botswana, and then these were the provinces, as you know, of South Africa: Western Cape, Eastern Cape, KwaZulu-Natal, Northern Cape, Northwest, and Limpopo. And what's interesting is that we see quite a bit of admixture between the subspecies in the Western Cape, Eastern Cape, and these provinces here. We also see that it's quite pure in KZN when you're talking about the South African giraffe, and also, which makes sense because most of those giraffe would have come down from Limpopo and Mpumalanga area. And so we think these results, this is just a result from historical translocations throughout the years. Speaking to property managers, ecologists, wardens, during my research, not many people know where their giraffe came from. So they don't know where they came from, when they arrived, and how many arrived. So we expect a lot of these animals, at least in the Western Cape, in Eastern Cape, might have come from Botswana and Namibia in the past, and no one actually recorded this. There have been also results or genetic testing done in the Free State by Van Niekerk in 2019. They also found Angolan giraffe genetics, animals brought in from Namibia way back in the day. So what does this mean? Some strategic priorities for South Africa, in my opinion, would be to have some sort of centralized database between the provincial authorities. Now, there was a presentation yesterday on the hippos, also wanting some sort of centralized database. So I think this would be really valuable for giraffe as well, and a bunch of different species. I know there's some species where you're obligated to hand data to some sort of provincial authority or some body, I think for elephants and lion, etc. So having that for giraffe would be great for keeping track of the conservation status of animals in the country. Conducting a national audit for genetics, which we're currently doing, but just to continue doing that. And having a shift in translocation policy for if you are moving a giraffe, is there some way that we can try and implement policy where if you're taking a giraffe from somewhere, think a little bit about where you're taking giraffe from and where you're sending giraffe to. And then also obviously expanding space from small fence reserves to having something bigger, dropping fences, wildlife corridors, etc. So in conclusion, securing the future for southern giraffe, I think moving away from just expanding the numbers. Southern giraffe are doing really well, they're not in trouble. So expanding the numbers, thinking more about the integrity of the populations in terms of the genetics and the movement. Prioritizing large connected systems for giraffe. Preventing hybridization before it defines the population. How can we do that? Should we do it? Does it make a difference for giraffe conservation? And also strengthening the private and public stewardship. I know it's very strong at the moment and across a whole bunch of different species, but continuing to do that. Thank you.
Thank you very much. We do have a minute for questions, if there are any. Any questions?
Thank you so much, Richard. That was great. I'm just curious, is the Angolan subpopulation of southern giraffe also mixing with, so like the results that you've shown in South Africa with the mixing, is that something similar in the Angolan subpopulation?
So the question is South African and Angolan giraffe, are they mixing? We expect it to be so. So there is Angolan giraffe, so in the Western Cape, Eastern Cape, that we've seen with these results. These are preliminary results, so we're still getting the full data set back. And there are South African giraffe there. So it makes sense that they would be mixing. The effects of that, we don't know. Hybridization in the wild with giraffe is not well studied. So whether there is an effect of hybridization between species and subspecies, I'm not too sure. But I think they're definitely mixing in the wild at the moment, yeah.
Okay, Thank you so much.
Cool.
Our next speaker, oh yes, sorry, round of applause. Our next speaker is going to be Liana, and she is going to be giving a voice to the giraffes.
Great, more on giraffes. Terrific. Well, good morning, everybody. My name is Liana Zanet, and on behalf of our fabulous collaborator, I will be telling you about some of the experiments we've been doing here in Kruger, which are giving voice to the giraffe's pervasive fear of the human superpredator. Now, worldwide analyses show that humans kill medium, large, and very large mammalian and avian prey. If it's large enough to fill a dinner plate, it is fair game. And we do so at much higher rates than non-human predators do, suggesting that fear of the human superpredator may actually exceed fear of all other predators given our greater lethality. So the question then is, how scary are we humans? Well, to find out, we've been conducting a series of comparable experiments across the globe, all using our automated behavioral response systems. We designed these in our lab, and they consist of a motion trigger camera trap connected to a custom-built speaker that then allows us to test reactions of prey to 10-second playbacks of humans speaking calmly in locally used languages versus large carnivore vocalizations versus non-predator controls. Now here in Kruger National Park, our manipulations have allowed us to establish that greater fear of humans than lions is overwhelmingly pervasive throughout the savanna mammal community. Fully 95% of species in the community either ran more from humans and lions, or they abandoned water holes faster to humans than they did to lions. Now, the same was true of giraffe, who also ran more from humans.
My dad was a teacher, so I grew up in a home that the only thing I knew was sport. I played tennis, I played cricket, played rugby, and I loved my sports, and especially cricket and rugby.
Now, most other species did that. The one exception in the entire community was, of course, you probably guessed it, elephants, who did the exact opposite. Elephants actually ran more in response to hearing lions than they did to hearing humans. But what we discovered is that elephants were not running away from our speakers playing lions. They were actually running toward it. And they vocalize. These bunch up, and then they approach the lion/speaker. This one smashes the speaker/lion. And it vocalizes. Now, importantly, we rarely saw anything remotely like this in response to humans, indicating that lions understand that they can defend themselves against elephants understand that they can defend themselves against lions, but not humans. So we were wondering then, could we have something parallel going on with giraffes? And we asked, are giraffes more aggressive to lions than they are to humans, just as elephants are? I mean, giraffes are mega herbivores, and they certainly can defend themselves against lions. Now, we didn't see any videos of giraffes actually attacking speakers, but like elephants, giraffes certainly did vocalize. But wait a minute, because that is simply impossible, right? Because we all know that giraffes don't vocalize. Now, it turns out that this conclusion is entirely based on observational studies. For example, our dear friend Adrian Schrader, who just spoke, is one of the very few who have actually gone out there and reported on field observations of giraffes vocalizing. And their observation was that they never heard them make any vocalizations during their study, right? Fair enough. But when you experimentally test them, and you do this with predators, giraffes certainly do vocalize. In this video, a lion playback sounds, the giraffe walks away, turns, stops, and then it compresses its torso and makes a huff vocalization. That's one huff. 2. That's 3. Here, a lion playback sounds. The giraffe this time runs away, but it stops, turns, and compresses its torso. That was 1. And that's 2. Indeed, giraffes vocalize a lot in fully 114 of 502 videos in 21% of cases. And critically, giraffes vocalize a lot, especially when they have something to say to their predators. Giraffes rarely vocalize to our control playbacks, but they did vocalize a third of the time more to predator playbacks, indicating that giraffes are not just sort of randomly blithering, right, saying nothing, telling stupid jokes, that they are in fact talking to their predator. And indeed, we get the same results here even when we consider only videos that have a single lone giraffe. Because in those cases, there is nobody else out there for the giraffe to be talking to, except for the predator that it's just heard. So coming back to our question then, are giraffes more aggressive to lions than they are to humans, just as elephants are? Well, with respect to vocalizing, giraffes certainly do react more to lions. Giraffes vocalize significantly more to both lions and humans than our control, and importantly, they vocalize significantly more to lions than they did to humans, much like elephants do. Now, because giraffes run much more from humans than they do to lions, is this why they vocalize less? I mean, they ran, right? So maybe they're just not there in order to vocalize. Well, that's not the entirety of the story, because when we consider just videos where the giraffe ran, stopped, and then vocalized, like the one I showed you, they still vocalize significantly more to lions, indicating that giraffes know better to run from and not talk back to the human super predator, but they can certainly talk back to this guy. Thank you.
Thank you, Liana. We do have 2 minutes for questions, if anybody has. Okay. Oh, Tasha?
I have a question around the definition of vocalizing. And I guess it's debatable. Because would you say a puff adder vocalizes? A snake? Would you say a snake vocalizes? Or a crocodile? No, right?
Yeah, they make noise. Yeah, they vocalize.
Okay.
I mean, there's been
s-
stations of giraffes making noises, right? So that's been noted in the literature. They sometimes grunt, they huff, they snort, they do this or that, but it's thought to be rare, and and making noise is not communicating. Right? So nothing has any sort of context in terms of what they're doing or what they're responding to, or if they're responding, or if they're just blithering randomly, right? Like just making noise. So this is showing that they're not only vocalizing, but the vocalization has information in it, right? That they're communicating with their predator.
Okay. Okay. And then a second question. Those lion sounds, they sounded briefly when I heard it, either like they were mating or like they're around a kill. Don't you think that when the animal hears that and it runs away, it realizes that? Because usually when lions are hunting, they're not making a noise. So do you think the animal then realizes that it's not at risk because those lions sound like they're mating or that they're busy around a kill? Hence the animal then stops after initially running away.
No, I think that, I don't think it matters what they're doing. I think it matters that the animal hears that a deadly predator is in the area, and if they want to survive, they better leave. It doesn't matter, like when I'm out there, doing the ABR stuff, and I hear a lion, if it's a snarl or growl like you heard here, or a roar or whatever, I hear a lion, I'm not going to go up and pet it, right? I'm going to run from it. And that's what these animals are doing too. They're eavesdropping on the information that their predators, even if it's accidentally, that they're giving in the environment. Like maybe they don't want their prey to hear them, but they're going to hear them, right, if they're talking.
Thank you, Liana. Okay, so we are now at our first discussion session. So I am going to hand over to Kansani, who will then be talking about how are herbivores, big and small, contributing.
All right, thank you. We have a few minutes, 10 minutes to do the discussion. Sorry about that. Thank you to all of our speakers. So as Erin has already said, let's just get some few thoughts from the room on how herbivores, big and small, contribute to this breathing, mega-living landscape. Any thoughts, or one of the speakers can start us off? Or any questions for the speakers? Okay, we have one there, Mbali.
Thanks for the opportunity. I'll just kick off by being maybe a little bit provocative, and I'll riff off the really nice presentation that Eurus started off with. And I would suggest, from my perspective, working in North America and in Europe particularly, that the state of global biodiversity is in such bad shape that we have to do some fairly radical things with regard to understanding how humans fit in with biodiversity. And so when we're thinking about how do herbivores contribute to a landscape, for example, I would urge managers and scientists to think from a systems perspective and start understanding how the ecosystem represented by that landscape actually works, to understand not so much the patterns that used to occur there at some pristine stage, but to understand the processes and the interactions. And then think very hard about how one would go about re-establishing the functioning of that system. And what that requires is thinking in trait space, thinking about what is it that places individual species of, in this case, say herbivore, into functional groups, and what are the functions performed by those functional groups, and what are the functions that are needed to establish a self-organizing social ecological system. And then management can proceed by focusing on those functional groups in a way that is actually agnostic to taxonomy. Initially, we shouldn't be too hung up about being purist, about what species should or shouldn't be anywhere. But we should think about what functions are needed in order to enable that landscape to provide the ecosystem services that the society that depend upon that landscape need. And then once management starts thinking about, okay, these are the functional groups we need, and these are the biomass densities within those functional groups, then the next thing is to start thinking about what species could we then use to populate those functional groups that would be appropriate under the climate
climatic envelope that we expect to occur at some stage in the future. And so essentially what I'm recommending is to think in a systems perspective, to move away from the traditional species focus, think social ecological systems, and think self-organization.
Great, thank you. That's a great thought. Is there another hand? Sam? Can you please switch it on? All right, thanks, Sam. Is there any other hand? And maybe we can think from a management perspective with the functions that you were talking about. How do we then how should we manage herbivores, considering that they are now moving within fenced and fragmented landscapes? So those functions, how do we make sure that they're still there beyond the parks and the boundaries of the park or of the parks? We still have a few minutes. And if you have also a burning questions for any of our speakers, you can take the chance now. We have 2 questions at the back.
I think all I wanted to add is that in Joris's talk, where he had these pictures of the rewilding landscapes, right, with very few people within it, maybe the one picture in the back. But the other thing that was very clearly missing from that landscape were people's animals. And I think that that is something that we are not thinking about strongly enough, about how to integrate people's animals and wild animals. And I don't really have any suggestion of ways forward, but that is definitely something that I think in living, breathing landscapes, with the incorporation of people and the support of stakeholders, that is something that actively needs to be designed, I think.
Thank you. Yes. Go ahead.
Yeah. Good morning. I just have 2 issues to raise. The first one, let's forget about these ideal ecological systems, pristine systems, because it's gone forever. That's the first thing. The second thing that we need to know is the humans that are living around the park, they are going to increase. That's not a question. So eventually there will be people out there, whether you want to incorporate the animals in those kind of systems, I think it's a no, because it's not going to happen. For some of us with the blue capes that have been moving around Kruger and coming to the Kruger, you can see the density of people that is increased around Bushbuck Ridge, and it's ongoing. It's not going to change. So from there, I suggest what Johan says. We should now relook at what functions do we want to maintain in these small islands that we have. If we have a system where we can integrate people out there somewhere, not necessarily Kruger, then we can also try and accommodate those. But thinking of pristine things driving that way, it's not going to get us anywhere. Thank you.
Thank you for that thought. Yes.
Yeah, just thought I'd just give an alternative view about the relative importance of function and species, because I think it all comes down to function for what. And so outside of the park, we have very functional ecosystems like sugarcane, very functional. And the question of function for what? The function is to grow sugar. But in a conservation reserve like Kruger, as far as I can tell, the function is to conserve species. And so I can't see why one would consider that we just ignore species in a conservation reserve and focus on function, because the question is, well, what is the ultimate function of the conservation reserve? And to me, it's to conserve species.
Yeah. Okay. We have one few minutes for one short question. Few seconds actually now. Okay. Oh well, we can always continue this discussion during the breaks. Thanks.
Thanks, Kansani. Our next speaker is going to be Natalia. She is going to be talking about when lions hunt alone. Thank you, Natalia.
Okay, good morning. Thank you for coming today. There we go. No, there we go. Thank you for coming this morning. I'm going to be talking about my research on African lions and one aspect of their behavior, their cooperative hunting behavior, but more importantly, when do they choose to hunt alone versus when do they cooperate? Current prevailing theory on why lions cooperatively hunt is byproduct mutualism. So they benefit because hunting in a group allows them access to a greater range of prey species. So it allows them to overcome prey that are much larger than them, that are dangerous, or that are fleet-footed and that they couldn't otherwise catch if they were hunting by themselves. Studying cooperative hunting in lions, however, is quite difficult, especially if you're trying to collect data on their behavior, not just on what they're taking down. They're often hunting in really low visibility conditions. Hunts are infrequent and unpredictable, so you have to follow them for a very long time to even capture one hunting event. And it's really difficult to observe all of the participants that are partaking in a hunt and even see what is happening. For example, here's a picture of a hunt. It's 4 lions taking down a zebra. I know this because I was there, but actually observing what is happening is really difficult, as hopefully you can see from this image. Furthermore, our understanding of lion behavior is heavily biased towards lions in the Serengeti population. Not to say this isn't a really important population and provides foundational knowledge on their behavior, but as we all know, lions occur in a variety of habitats, from beaches to tropical forests, woodlands, mountains. They don't just exist in a vacuum of the Serengeti. So one question that I'm really interested in is, what does lion behavior and sociality look like under different ecological contexts? And one aspect of that is, what does their hunting behavior look like? So today I'm going to talk about a study site that we have in Botswana in partnership with Leopard Ecology and Conservation. 2 of my collaborators, Jen and Trevor, are actually here. So if you're interested, you can also find them to ask them questions about this study. We operate in a semi-arid system in Kutsi Game Reserve. It's right at the base of the Central Kalahari, and lions can move between the border of Kutsi and the Central Kalahari. One aspect of this NGO of LEC that really first kind of was exciting for me to study there is that they employ teams of indigenous trackers. So there's SAN tracker tracking teams that are, their skills are incredible. Their ability to interpret spoor and collect behavioral data on where the lions are going and even what they're doing during a hunt is phenomenal. So we work alongside these teams to collect data in a way that isn't possible from modern technology alone. Not to say that we don't use modern technology, so a subset of the lions do have GPS collars, at least one female from each pride, sometimes female from each subgroup. We use the collars to identify clusters, so when a lion has stayed in one spot for over 2 hours, it indicates that there might have been a kill. When the team sees a GPS cluster, they will go out and investigate it, and then they will search the area for a kill, and if they find that it wasn't a resting spot, it was in fact a kill site, they will then do what we've termed backtracking. And what this does is it gives us really fine-scale data on what happened during a hunt. So we get the paths that the lions took, we get the paths that the prey species took, we even get behavioral data, so the portions of the path where the lions were stalking, walking, or chasing, and we get information on what happened after the kill, so which lions ate from the kill, which lions participated in the hunt. And so in this way, we're able to collect data that you couldn't collect just from remote observation alone, and we're able to collect a lot more data because we're not having to follow the lions and wait for a hunting event to occur. So what did we find? What was really interesting about this population is that they show a significant preference for hunting by themselves. These lions don't seem to be cooperating, and one potential explanation for this could be that they're just taking down smaller prey or prey that are easy to capture. But when we delved into the data further, we found, in fact, that's not the case. They're hunting by themselves even when they're taking down large prey species like giraffe, eland, and hemsbuck. When we look at what prey species we captured in hunting events, the vast majority of prey that they were taking down were eland and hemsbuck, which are really large, really risky. Hemsbuck are really fast and fleet-footed. So classic theory would predict that they should be cooperating to take down these prey, but that's not what we're seeing. They're also taking down adult giraffe by themselves. So a single female lion will take down adult giraffe, which is not a small feat. So why is this happening in this study system? One thing we thought might be occurring is this is a really low density environment. When your next meal is coming is really uncertain. It's very low prey availability. And the lions might also, they operate over really large ranges, so perhaps they're spending less of time together and their partners aren't in a recruitable distance. And if you see a prey item, regardless of what size it is, because you don't know when your next meal is going to be, you have to take the risk of taking it down rather than wait for a partner to cooperate to take it down. Theory would predict that if this is what is happening, if you're taking all of the risk and all of the energetic effort to take something down, you wouldn't be sharing your kill. Surprisingly, that's also not what we're seeing here. Lions that are taking all of the risk and incurring all of the energetic investment, they take the animal down. There are other lions coming in and feeding at the carcass, which was surprising. And it is it's significantly more likely that there will be non-participants at a kill when it was a solitary hunt. So if they're hunting cooperatively, it's less likely to have animals coming in and feeding off the carcass than if you've hunted by yourself. So what is going on here? One potential explanation is that instead of byproduct mutualism driving cooperative hunting in this system, it is some form of reciprocal benefit. So your subgroup of 2 females, you don't know when your next meal is going to be, you're going to maximize your search efficiency and split up. Whichever one of you encounters a prey item first, you take down that prey item, and then you share with your group mate, you recruit them in, knowing that the next time when your partner finds a kill, you're going to receive the benefit. So over the long term, you're both getting the benefits. Additionally, what we found was that 95% of the time, the non-participants were offspring and a single adult female. So this also indicates that there might be some sort of kin selection happening here, where one female is staying back with inexperienced offspring or offspring that are too young to hunt. The other one is going out and making the kill, and then they're sharing with their closely related kin. We are investigating this further to figure out exactly where the females are. Are they in a recruitable distance? And which of these explanations is the most feasible for this population. But the big takeaway here is that in prey-rich systems like the Serengeti, cooperative hunting might be expressed more often, not because lions lack the ability to hunt alone, because partners are more frequently nearby, waiting costs are low because your next meal is more predictable, and risk to take down a large prey species by yourself is actually still high. Whereas in low density systems, it's not that the risk is eliminated, but it's being reallocated. The individuals are accepting a higher short-term capture risk to avoid longer-term starvation, and they're gaining indirect fitness benefits via kin selection and reciprocal mechanisms. And so I think this is just one example of how cooperation and behavior are context-dependent strategies, and ecological constraints in low-density systems might be preventing cooperative hunting from being expressed even when it's predicted to be selectively favored. And I imagine there are other behaviors where we're going to find the same types of things. So thank you very much. I think I have a little bit of time for questions.
Thank you. That was a great presentation. There's maybe time for like one quick question. Let me.
Good morning. Hi. Very nice talk. Enjoyed it much. I recall we have had some situations where lions were hunting buffalo on their own, females in particular. We know males can literally take down anything on their own. It's not an issue. And also understanding that, a pride of lions has got different functions within the pride. Some members are responsible for scaring prey, others are responsible for just attacking. Would you really consider this a new behavior, or is it just a new observation from your side? Because I think, if you look at it historically in the past, I'm sure a lot of this has happened within the different spaces where lions occur. So I'm really interested to just understand if this can be classified as a new behavior in the Botswana region, or it's just a new observation. Thanks.
No, I don't think it's new behavior. I think it's a behavior that just hasn't been, because studying cooperative hunting is so logistically difficult. I don't think there's a lot of scientific publications on this. I think people on the ground know that this is happening. It's just a matter of getting it out there in the literature. I have struggled to find, so buffalo are a really interesting example because our study site doesn't have buffalo. And in the current scientific literature, it indicates that buffalo are the one species that lions won't take down by themselves. There's not any recorded instances of that. I don't believe that to be true. Or I'm not sure if it is true, let me say that. So if you have examples of lions taking down buffalo by themselves, or female lions in particular, that would be really interesting. But no, I don't think it's new behavior. I think it's behavior that just needs to be recorded.
Thank you so much. Okay, our next presenter is Holly, and she will be talking about habitat selection and space use within the large carnivore guild.
Okay, hi everyone. My name's Holly. I'm from the University of Bristol in the UK. I'm collaborating with Botswana Predator Conservation, and this is my current study, Patterns of Multi-Scale Habitat Selection and Space Use within the Large African Carnivore Guild. I'm currently only on the habitat section of this study, so that's what I'll present on today. So just a bit of background. Habitat selection and space use are ecological processes that are known to occur at different spatial and temporal scales. And failing to address these scale dependencies in ecological studies could result in incomplete conclusions, and this could result in downstream effects on conservation of the species being studied. So the study system in which I'm working in northern Botswana, it presents a great opportunity to study these processes for an intact large carnivore guild. However, detailed habitat data and ground truth assessments are currently lacking for the study area. And there are various ways to collect ground truth data. I'm going to present on one way that we're using, which I think is interesting. So the aims of my study are to analyze multi-scale habitat selection and space use for the large carnivore guild, and that includes these species: lion, leopard, cheetah, wild dog, and spotted hyena. And for this, I'm going to be creating high-resolution, ground truth, seasonal habitat maps for northern Botswana, and that's today's focus. So my study area is in northern Botswana on the eastern edge of the Okavango Delta, and this is where Botswana Predator Conservation's research camp is based. So for this area, there's no really reliable habitat data to date. Ground truth assessments aren't generally done. The land cover is dynamic and seasonal, and it's complex, and remote sensing of seasonally inundated habitats is difficult. So we thought of this current method to collect ground truthing data. I've split my methods into two sections. So this is ground truthing data. And for this, I have collected ground truth imagery for the core study area using a GoPro 360-degree camera mounted to a 4x4 research vehicle and put on a 10-second time lapse and driven opportunistically around the core study area on these sandy roads. These images are all GPS tagged. And then second step, we have been defining habitat categories and annotating the GoPro imagery. So for this, we subsetted the data with 1,000 GoPro images, and then we ran an inter-observer reliability check. Go back. So myself and one of my co-authors, David Hoffman, we ran this check in the Shiny app and independently classified all of these GoPro images and then compared our results. However, this is an iterative process, so we're still sort of developing this method and trying to improve it. And the way that we're doing that is trying to consider these two important questions. What's important for large carnivores in the landscape? That might be prey catchability, accessibility, lines of sight, but also what will the classification algorithm in GIS detect? So third step, we finalize the ground truthing data set. And for this, we thinned the data set to 1 image per 200 meters squared. And this meant that we removed the duplicates and also made the data set more manageable. I might adjust this grid depending on the results, but currently that's what we've done. So the second section of this methodology is the habitat classification. For this step, I'm not there yet, but we will train the classification algorithm using the ground truthing data, which would be based on the GoPro imagery. And for this, we'll run supervised classification and use a random forest algorithm. We'll then validate this algorithm using k-fold cross-validation. We'll generate 50 to 100 points per habitat class to run an accuracy assessment. And then we'll generate a confusion matrix and interpret those results. So we'll apply the classifier to satellite imagery from the time period in which the GPS data for the carnivores spans. And because Landsat dates back to that period, we'll use Landsat imagery at 30-meter resolution, collect for the period of 2007 to current and across the wet and dry seasons. We'll then augment this habitat data with additional habitat data such as roads, rivers, settlements, etc. So just some preliminary results. This is the coverage that I managed to get during my last field season. I managed to collect 23,722 GoPro images, and then I thinned this using that grid system to 4,731 images, and this covered about 10 concessions and about 3,030 kilometers squared. So these are just some example images from the GoPro data set. On the left is the format of the image that I'll be using in my analysis. This is called an equi-rectangular image format, and basically there was the GoPro had 2 lenses, took a picture on either side, each lens, and then it stitched it together. On the right is just, I thought, a really fun way to show the data. So you may have noticed from these images that there's quite a lot of warping that occurs as you get further from the lens and at different parts of the image. So a 0 to 5 meter distance from the lens looks wildly different from a 5 to 10 meter distance from the lens, and then this distance looks smaller and smaller the further away you get. And this might be why during the inter-observer reliability check, there was a low agreement between myself and David, the other independent observer. So less than 50% of the decisions we made on these habitat classifications of the GoPro imagery were the same. So initially I defined 19 habitat classes based on species and whether it's open versus closed. But only 2 of these habitat categories, the grassland and the open pan areas, showed above a 65% agreement between myself and David. 2 of these categories showed about 50% agreement, and then the rest were less than 50%. So obviously we needed to go back to the drawing board and think about how we defined our habitat categories and also what sort of training data we wanted to try and get to apply to the classification algorithm. So initially we were thinking of defining a 30 by 30 meter area with the GoPro camera at the center. And each GoPro image represented training data for 1 30-meter satellite pixel. However, as mentioned, the level of warping from the GoPro camera made it difficult to judge that distance. So now we're thinking of using training polygons and using the GoPro images to verify each side of the road what the habitat is, but use these base maps and GIS to kind of draw these polygons for the habitat that we can see from the image. This also meant that the overrepresentation of roads in these images could be addressed, and the edge habitat, trying to define edge habitat in these images, which was very difficult, could also be addressed. So just in conclusion, this shows the importance of checking classification at various points of the process. And I know that I need to consider in future that the classification algorithm may not detect different habitat species or habitat composition. However, I'd rather start with more habitat classes and sort of slim it down through trial and error if there's a high pixel overlap between classes. I also will be adjusting this protocol as necessary. And I'll be using these resulting maps in step selection functions to assess habitat selection and space use by large carnivores. And I'll be doing this at different temporal scales, which will be represented by GPS color fix rates, different fix rates. And then I'd like to summarize this process to provide to Botswana Predator Conservation, and so we can maybe try to get a more regular understanding and record of the habitat in the area. Thank you. Any questions or tips?
Thank you so much, Holly. That was great. We do have just under a minute for any questions. Well, maybe like one question. Okay, I don't see any questions. Where is that one? Oh, okay, at the back. Sorry about that.
Hi, that was a really great talk, and I especially like your idea of moving the polygons on either side of the road. I think hopefully that'll help with your classification differences between you and your partner. I'm kind of curious, have you thought about looking in your imagery? I don't know how many like animals you're able to catch in your imagery, or thinking about using open source data sets like on MoveBank or something to think about classifying these different wildlife habitats based on like what prey species might be there. Thank you.
I think I missed the beginning of that, but I think I got the overall question. You're asking about whether I'm thinking about prey-related factors and how they're viewing habitats.
Yes, the first part was me complimenting you on your study.
Thank you. Yeah, there's obviously a lot of factors to consider. I mean, it's really hard to try and integrate all of that when you're thinking about the habitat classes. I think I would like to incorporate that if I could, but currently trying to create these different classes and make sure that there's an agreement itself is like really difficult. So I will definitely try and consider, but we will see.
Okay, thank you so much, Holly.
Thank you.
Okay, I'm going to hand over to Dan Parker, who will be discussing how carnivores are contributing.
Thanks, Erin. I don't think I'm going to be doing the discussing. I hope not. It'll be boring for everybody. But I think just to kind of kick off this 10-minute session that we've got now, based on essentially using these two talks to kind of kick it off and then sort of extending what we had from the sort of herbivore session. Let's just consider a couple of things quickly based on what Natalia and Holly have presented, which I think is a nice starting point. Natalia was talking about context and how context is important. We need to consider that. Different landscapes may result in different things happening when it comes to carnivores. How are they interacting with people, etc. And then Holly's work telling us about the specifics. Specifics matter, but so does bias. So we've got to make sure that we ground truth, the data that we rely on, so, especially in contexts like remotely sensed data and so on. But what I'd like to do now is ask firstly if there are any special questions or specific questions that you might have that you didn't get a chance to ask Natalia or Holly. You have an opportunity to do that now. Or if you'd like to make some interesting, somewhat provocative statements, or just statements about how you think carnivores fit into this bigger living landscape, breathing landscape. Over there, Paul, and then Corley.
Yeah, okay, thank you. This is a question for Natalia. I was wondering, predators can adapt their hunting behavior based on cover. So obviously in an open system, there's less cover. They might need a group to take down prey versus a dense area. I'm wondering if you considered that as you were analyzing your data. And I think using view sheds would be awesome. And I know that's a lobby for myself, but.
There comes a mic. It's coming behind you, Natalia. There you go.
Okay, thank you. Yes, we did consider habitat and openness. I didn't have time to present the results of that, but there was no effect of habitat type on whether they were, what type of prey they were taking down, or whether it was cooperative or solitary. It was a very rough estimate, so we just looked at percent openness in the 4 cardinal directions. I do have a master's student doing a project right now using satellite imagery and overlaying the GPX tracks to see if maybe there is some sort of habitat effect on a finer scale. So that is to be determined. But I was surprised that openness didn't seem to have any sort of effect.
Cool. Corley's next. Here you go.
Thank you. I don't have a question. I just have been thinking to all the talks that we've been listening to, including those archaeology ones. I don't know if Tim is still around. And I'm thinking that we really are disregarding the importance of people as a predator in these landscapes. And if we just see how many villages there were, if you look at all those archaeological sites that is found in the north, and I'm sure if you start looking for them all over, you're going to find them. And Tim was making the point that a lot of the meat, and we also know from other work on the Letaba that they were just these massive butcher shops, if I can call it that. And the effect that that would have had just on how animals use the landscape and also how other predators would have used the landscape.
Absolutely, yeah. Thanks, Corley. It's actually something that Tim is thinking about a bit more. He sent me a voice note the other night talking about a possible theme where we can start to connect these, what has been the influence of humans over extended periods of time, going back as far as we can to see what has been happening in these systems. That we're focusing on the contemporary ecology, but what was happening before and how is that influencing the ecology of today, I think is important too.
No, I'm about to run off the ladies, but I just want to say, tourists often say, you have a bit of interaction with baboons in the camp, and then they're like, we're in their space. This is their territory. And I'm actually No, they're in our territory. We've been here for a very long time. So I think that's an important thing to remember. These pristine landscapes are not without people. They're not supposed to be. And yours also showed like how people are still envisioning us wilding by taking people out of the landscape, and that is just completely unrealistic.
Thank you. I got it, Mike. Thanks. Yeah, I actually want to agree with Collie. And I was also going to rise on that point of the ancient civilizations and how they were connected to nature. So if you think from a lion perspective, I mean, we all know the Zulu have got the leopard skins, and the Maasais have got the whole tradition around hunting lions. Apart from that, resource beneficiation, I think there's also a very strong Afro spirituality that is associated with the carnivores. And I suppose, the humans and the wildlife have really evolved in this space together for a very long time. And based on our traditions, based on our cultures, that value still hasn't disappeared. So we must actually be aware of the fact that there is science on the one end, but there's a livelihood way of doing things on the other hand, by the local communities or the local people, and we just need to merge that gap between the two, because I kind of feel like there's a separation. But it is important to actually take recognizance of the ancient ways of living and connection to the land and the animals of the land. Thank you.
Yeah, thanks, Stephen. Are you going to respond to that and add to it, Sam?
So thank you, and Johan started the discussion earlier about being process orientated, and that process, as Ellen has highlighted, it depends a bit on what you want to achieve. So I think the contribution of understanding predator processes like Stephen and Corley has highlighted now are really important, but what we need to think a little bit about it is you're generating this landscape of fear, and there's a landscape of fear for a lion, and as Liana said, there's a landscape of fear for a super predator. Our challenge is what happens when that landscape of fear is not heterogeneous anymore. And this is perhaps where we have shifted to now. So how do we think now, how do we recreate that heterogeneity of fear that is generated by the predation process, whether it's us chewing things or whether it is a lion chewing things, etc. Then the second thing, Stephen has highlighted a very important point. The predatory fear is very different now. At the moment, you're not getting caught and slaughtered, and a whole bunch of other things happen. There's very different ways that the fear. So it's also the type of fear that I think is, or the type of predation that is happening that is not historically. So we don't have the process, even the opportunity to restore a process to something what it was before. Like Johan said, don't restore to a state what it was before. What we need to say is, under these new conditions, how would that process generate things for us that are beneficial to what we as society has decided? I hope that helps.
Thanks, Sam. We've got one last word. So over there at the back, I see there. Is there another one? Okay, we'll take here and then that one. Okay.
Thank you so much. Where I come from in Uganda, we are having big issues with man and biosphere reserves. We have Queen Elizabeth National Park in Western Uganda, a very big man and biosphere reserve. But in the recent times, we have the lion population highly threatened. The question is, do we get rid of man in the reserve? How do we protect the lions? Because policy says each family should have at least a goat or a cow for milk. But with time, the families have grown livestock stocks have grown. Yes, we want to be with man, but what do we do to preserve and protect wildlife as well? This is a point to ponder for all of us in the room. I'm sure other countries have similar situations. I'm bringing this out for us to think about it. After we've done habitat classification, used all the technology, and the populations are growing, how are we going to help man live with wildlife? Thank you.
Thanks very much. Yeah, that's very, very good. A very good point, and it connects up with what Joe was talking about earlier, how this, and Corley's comments about we can't just be thinking in isolation. One final point at the back. We are out of time, but let's have one last
Yeah,
comment.
thank you so much for these talks and this discussion. I just wanted to mention also that I think what's important here too is different conceptions of what nature is. And I think as some people alluded to, that there are many cultures that don't view nature as separate from humanity. And I think that the conservation has often taken a separation approach to as the previous speaker said, like man and animal. And something, sometimes that can be necessary, but something that we lose there is, for example, the knowledge of how to coexist and live alongside wildlife like large carnivores that can be very difficult to live with. So I think that's where also listening to people's ideas from cultures who have not separated nature and humanity ever, knowing that these systems have always, as we've heard, been socio-ecological integrated systems, and thinking about how do we bring back our understanding of how to all coexist, rather than thinking about how do we separate humans and nature completely to avoid any conflicts, because we know that that will just end up with nature, especially for species like large carnivores that require a lot of space, not actually having the space and the resources that they need to survive for the long term.
Thank you very much. That's fantastic, folks. We'll carry on with these discussions as the session goes along, but back to Erin, Sam, are you up now to take over the chairing?
Yeah, no, I evolved. Erin evolved. Or degraded, maybe, perhaps, to me now. Why don't you give Erin a round of applause? That's the first time that she's ever chaired a session in a conference. She did a great job, guys. All right. And thank you for those discussions. We can see there's some sort of general theme emerging out here. Let's go to elephants now. This is not the elephant in the room. It's just elephants for now. Well, first, we're going to have 4 talks, and then Rob will come and facilitate another similar discussion like Dan and Kensani has just done. First, we're going to have Therese, who's going to talk on movement ecology and African elephants. Please. Thank you.
All right, hello everyone. Thank you very much for the introduction, Sam. I'm kicking off the elephant talks, and I'll be discussing the movement ecology of African elephants within a park in the Malawi-Zambia landscape. So as we all know, translocations are often used as a conservation management tool to control the population densities, to supplement populations, and to control population growth. However, translocations do come with their downsides, such as being one of the more expensive conservation measures, especially when it comes to moving large animals such as elephant bulls or whole herds of elephants. Now, despite the common use of translocations and their success in the long term are seldom evaluated, especially for long-lived species where the usual parameters such as reproduction and survival aren't effective parameters to measure the success of the translocation. Now, this is where movement ecology comes in. Movement ecology tells us the when, where, why, and what of animal movement. And the incorporation of movement ecology allows then for spatially temporally flexible conservation measures. And this is especially useful for long-lived species For example, the movement behaviors and patterns, such as the exploratory behavior away from a point of release, can tell you a lot about the likely survival of a translocated population within a novel environment. So just a little bit of background on my study site, Kruger National Park in Malawi. In the period between the 1970s and 2012, poaching resulted in a 90% reduction in the elephant population. And subsequently, assessments indicated that the local population would go extinct by an estimated 40 years. And this led to demographic rescue plans in which 263 elephants were translocated from Liwonde National Park to Kruger National Park. And this was an estimated threefold increase in the elephant density within about a month. This large-scale translocation and the movement telemetry data of the resident population prior to the translocation allows for a unique opportunity to look at both the daily activity patterns of both the resident and translocated populations, and also allows us to look at the exploratory behavior of translocated elephants in a long term. So as I mentioned, my study site is Kruger National Park, which is found within the Malawi-Zambian landscape, and it spans approximately 2,316 square kilometers. The park is primarily dominated by Miombo woodland, interspersed with drainage lines and seasonally wet grasslands. And as you can see, the rivers stretch across the park as indicated in blue, and we have one artificial dam called the Fupa Dam within the park. And the park is also partially fenced on the Malawian side. 30 elephants were collared with African wildlife iridium satellite collars with very high frequency transmitters that record the GPS fixes on an hourly rate. Of the 30 elephants, 5 were resident elephants, of which 4 were females and 1 was a bull. The remaining 25 collars were then fitted on translocated elephants, of which 20 were females from different family units and 5 were adult bulls. Now, just to show you what the telemetry data basically looks like, we'll look at the preliminary results that my colleague Ryan found. And when Ryan looked at the first year and a half of telemetry data, he found that largely the elephants remained within Malawi. However, there were a few excursions into Zambia. And one excursion of note was a 2 to 4 month excursion into the Luanda Valley that 3 translocated cows and 1 translocated bull undertook. It's just important to note that for the remainder of the talk, we'll only look at the movements within the park. So I firstly wanted to look at the total ranges of the resident and translocated population in a period between 2022 and 2024. So I ran 95% minimum convex polygons to look at the space use. Now, of course, as we all know, elephants alter their range use and movement patterns depending on seasonal resource availability. So I ran these 95% MCPs for both the dry and the wet season. And what I found is that the total ranges between my resident population and my translocated population was significantly different. I also found that the total ranges for both the resident and translocated population was not significantly different when we compared the dry season and the wet season with one another. Next, I looked at the space use activity budgets of both populations. So just to break down what we're basically seeing here, firstly, I divided my elephant population into 4 elephant classes based on sex and population. So we have the resident females at the top, for example, and the translocated bulls at the bottom. And again, I then looked at both the dry and wet season. So the heat maps were plotted for the spatial activity for each of the elephant classes next to each other. So we have dry season versus wet season for the resident females. And what I basically found, as we can see here at the top, is that there wasn't a significant difference between the spatial activity of the resident females compared to the resident bulls, and same for the translocated population. I also found no significant difference in the spatial activity between the dry and the wet season for each of my elephant classes. However, once again, I found that there was a significant difference in the spatial activity between the residents as compared to my translocated population. I then wanted to see whether or not the translocation potentially affected the spatial activity of the resident population. So I compared the spatial activity of the resident population after the translocation with the spatial activity of the 2005 resident population. And what I found is that the historic population had a significantly higher spatial activity as compared to the resident population after the translocation. I also looked at the average straight line travel distance from the points of release or the points of collaring in the case of the residents. And what I found is that there was a significant difference in the mean straight line travel distances between all 4 of my elephant classes. My postdoc comparisons also indicated that both the translocated bulls and the translocated females moved significantly further away from their points of release as compared to their resident counterparts. Next, I looked at the average travel speed and found that there was a significant difference in the movement speed between populations and sexes, with my translocated bulls moving substantially faster than my resident study elephants when looking at the bulls. And although less pronounced, I also found that the translocated females moved slightly faster than the resident females. Again, across both populations, we see that the translocated bulls move faster than the females. Finally, I looked at the average step length and found once again that the translocated bulls took the largest steps, which was significantly higher than that of their resident counterparts. Although less pronounced, once again, we also see that the translocated females had slightly larger steps when compared to the resident females. And once again, we see that for both populations, the bulls consistently took larger steps when compared to their female counterparts. So basically, to sum up what we found, the translocated elephants show high exploratory behavior when we compare them to the resident population. I found no seasonal differences in the movements, which is surprising because we expect the elephant movements to change depending on the resources availability. The bulls generally moved faster and much more than the females. And we aren't surprised when we find this because we know that the bulls are the dispersing sex and they aren't limited by the movements of the calves. And finally, what I see is that the current resident population seems to show behavioral changes and potential range reductions potentially due to the translocation when we compare them to the historic 2005 population. So to conclude, I think it's really important for us to truly evaluate conservation measures such as translocations, that we look beyond just singular populations. It is only when we look at both resident and translocated populations that we can fully grasp how these measures alter the behaviors, the movements, and ultimately conservation outcomes. I just want to take a moment to thank my supervisors, my research group, I4, and the National Research Foundation for making this study possible. Thank you very much.
Thank you, Therese, and I'm sure people will have a nice chat with you at tea time. You've used your time perfectly. Thank you. All right, our next one is going to be Jessica, who is going to talk a little bit about the barriers and opportunities for elephants to move, particularly in the GLTFCA. Are you ready, Jessie? Go for it.
Thanks so much, Sam, for that introduction. Hi, my name is Jess Butrelli. I'm a PhD candidate with the Conservation Ecology Research Unit, and I will be presenting very preliminary results from my first data chapter. Since this is so preliminary, I do welcome all feedback. The study site for my project is the Great Limpopo Transfrontier Conservation Area, hereafter GL. It's comprised of 3 countries, 5 national parks, as well as a designated wildlife corridor. The GL occurs across a north, south, and east-west rainfall gradient, with an average annual rainfall range of approximately 300 to 600 millimeters. Kruger and Ghana Reserve are the only national parks with fences, and Kruger has supplemented surface water. Climate change is rapidly altering our natural environment and will impact natural processes such as vegetation greening and browning, as well as surface water drying. Here, we can use preliminary modeled base layer created by the Global Change Institute that interpolates data from 1981 to 2000 and predicts future changes. It is important to note that this is based on a worst-case carbon emission scenario with a representative concentration pathway of an 8.5 RCP. We can see that the temperature increases slightly from 2021 to 2040. However, between 2061 and 2080, the average temperature increases sharply, reaching a maximum increase of 3.4 degrees centigrade. Sheesh, sorry. Again, we have another modeled base layer that examines predictive rainfall changes based on modeled data from 1981 to 2000 for the years 2021 to 2080. This is also modeled in a worst-case scenario with an RCP of 8.5. It is predicted that the average annual rainfall from 2021 to 2040 will increase, but from 2061 to 2080, it will decrease significantly to a deficit of 90 millimeters. We categorize the ability for elephants to disperse into 2 categories: barriers and opportunities. Barriers are anything that it hinders an elephant's ability to disperse, while opportunities facilitate their movement. We identify both environmental and anthropogenic barriers to elephant dispersal. Environmental barriers are defined as steep slopes, wide, deep, fast-flowing rivers, and extreme events such as droughts, fires, and floods. For anthropogenic barriers, we define those as human density, large-scale industrial crops, linear infrastructure such as roads, railways, veterinary fences, and international borders. We organize elephant opportunities into 4 main categories: water, which is distribution and availability, food, which is distribution and availability, as well as seasonality, such as grass availability in the wet season and woody material in the dry, shade in the form of large trees, and safety, which we define as natural habitat and large protected spaces. This slide shows 2 protected areas within the GL, the northern section of Kruger National Park and Benin National Park. I've animated telemetry data from 1 elephant per area to showcase how elephants utilize the landscape and how they move over the backdrop of vegetation changes, which I've used EVI, over a certain time frame. The animation was meant to highlight the difference between extreme environmental conditions and a more typical vegetation precipitation year comparatively. The Kruger analysis shows the northern section of the park north of the Olifants River. This animation was created by analyzing the changes in vegetation during the extreme drought of 2015 to 2016. Citing Malherba 2020, we analyzed the changes from 1 January 2015 to 29 February 2016. We also animated the historical Seru tracking data for the corresponding year. I used MODIS at 250-meter resolution for this presentation, but for my analyses going forward, I will be using Landsat 830-meter. Sorry. Okay. For Benin National Park, I analyzed the change in vegetation for the year 2024. The elephant displayed here, a Peace Park's elephant, traveled 25 kilometers beyond the park boundary, for which I created a 25-kilometer buffer around the protected area. The telemetry data began approximately 20 weeks into the year, but this elephant spent approximately 87% of its time outside of the park boundary. This EVI analysis was created by using Sentinel-2 data at 10-meter resolution, resampled to 250 meters for the purpose of this presentation. One of the contrasts between the 2 animations, besides extreme weather events, is that Kruger has supplemented surface water, shown in blue, that versus Benin that only has rivers and naturally occurring surface water. Okay, okay, okay. Okay, sorry. For the next part of our preliminary results, the next variable I will be adding to the model is surface water. Benin National Park features an east-west rainfall gradient, with the eastern half consisting of a delta-like wetland system, a good case study for starting to look into surface water. For these early results, we used modified normalized difference water index, MNDWI, to detect surface water shown in red in Benin National Park. For the time period of 2015 to 2016, which was the year of the extreme drought, and 2024 on the right for a more normal rainfall year. For our 2016 analysis, I used Landsat 8 at 30 meters, and since Sentinel-2 was only launched since 2017, to show the available surface water, which represent, For 2024, I used Sentinel-2 10-meter resolution to map surface water across the same site. I will conduct this analysis across the entire landscape since Kruger is the only protected area with supplemented surface water, it is critical to understand how surface water is drying and to assess water availability across the landscape, and particularly in areas where we have not established surface water maps. So what I'm busy currently working on is compiling and analyzing dynamic environmental data layers, which includes the surface water data layer with surface water availability, detection, and mapping across the GL. Seasonal multi-year vegetation indices in the form of EVI across all of the sites, temperature data sets, rainfall data sets, as well as anthropogenic data sets such as human populations and linear infrastructure. I'll be expanding spatial and temporal extent of the study variables and integrating additional elephant GPS color data sets across the GL. I will also be analyzing multi-year patterns to capture drought versus rainfall years. I will also be analyzing a lot of movement analyses in telemetry data, so basic analyses such as step length and turning angles, and more complex analyses such as integrated step selection functions, which will hopefully help quantify elephant selection for vegetation, water, and the landscape features. I will also be modeling the effects of anthropogenic barriers, as I said, and then comparing movement strategies across the environmental gradient, which all in all will help predict future linkages and connectivity. I would like to thank my supervisory committee, as well as the Peace Parks Foundation and the Global Change Institute for their contributions and support of this project. Thank you.
Thank you very much, Jessica. You've done such a good job. There's time for at least one question. Corley.
Thank you. I don't know if I've seen a paper, it was Steve Higgins and one of his postdoc students, maybe Simon Scheiter, and they modeled future productivity of woody vegetation in the park in Kruger, and they said that I think what can now be the vegetation that can sustain half an elephant in future will be able to sustain 8 elephants, because the woody vegetation is going to become so productive with increased CO2. So I was just wondering, is there a way to model where we will find these incredible increases in productivity, and then going forward, what is going to be more important, vegetation or water?
There you go.
Thank you.
You want to respond to that, Jessica?
Sorry?
Do you want to respond to that question?
Oh, yes, sorry. So I don't have an answer to that question right now, but I can get back to you later about that. Thank you. We're not that far in.
All right, thank you. We're running out of time now. Thank you, Jessica. We're going to have our next one is by Catherine, who is going to talk also. We're staying in this movement space and talk a bit about elephant corridors. Catherine.
I'm Catherine Bunny from Elephants Alive, and the title of my presentation is Landscape Connectivity of Large Fruited Trees is Sustained by Elephant Corridor Movement. So for most of the Earth's recent history, landscapes were structured by very large animals. Megaherbivores moved seeds across vast distances, linking plant populations across rivers, floodplains, and entire ecosystems. During the late Quaternary, most large-bodied herbivores disappeared from the Americas, from Europe, and from Australia. These animals were not simply charismatic fauna, they were major agents of long-distance seed dispersal. When these megafauna disappeared, long-distance dispersal of large fruited trees likely collapsed across much of the globe. In regions such as the neotropics, many large fruited tree species now persist as scattered adult populations with restricted and fragmented distributions. There's an example of Carriocar genus. But Africa stands apart as one of the few places where this ancient ecological process still persists. Today, African savanna elephants remain among the only animals capable of moving large fruits across landscapes. A decade ago, we did some work looking at how far elephants in Kruger disperse seeds and found that they're capable of moving seeds up to 65 kilometers, further than any other land animal. So the question is not whether elephants are long-distance dispersers, but rather whether, in increasingly fragmented landscapes, elephant movement is still sufficient to sustain connectivity among large fruited tree populations. And so to explore that, we focused on a dispersal syndrome that evolved alongside megaherbivores, and that is the megafaunal fruit syndrome. So megafaunal fruits are typically defined as large, mechanically protected fruits that evolved for dispersal by very large-bodied vertebrates. Their size and structure makes them poorly suited to small dispersers. In southern Africa, one clear example is the lala palm, Hyphene coriacea. Its large, fibrous fruits are swallowed whole by elephants and transported internally. Here are some more of the 31 purported megafaunal fruits that we've identified for South Africa, fitting the definition. So before modeling connectivity, we first needed to quantify, one, how long lala palm seeds remain inside elephants, and two, what does gut passage do to germination? So we conducted a controlled gut passage trial to quantify how long seeds remain inside elephants. We did that at Herd Elephant Sanctuary outside Hoedspruit, where we fed 15 elephants 30 lala palm seeds each in one go. The majority of seeds were excreted around 48-hour mark, with some seeds continuing to emerge up to 4 days, 96 hours. Passage through elephants increased germination probability fourfold. Ingested seeds began germinating after 40 or within the first 40 days, whereas control seeds didn't begin to germinate until the 2.5-month mark. Thus, elephant-mediated dispersal enhances recruitment probability. So having established these realistic retention times and enhanced recruitment, we then asked whether elephant movement, particularly through corridor systems, is sufficient to sustain the connectivity at landscape scales. Elephants Alive has 28 years of elephant movement data across the broader transboundary landscape. But for this analysis, we focused in on the southern Mozambique corridor system, connecting Kruger National Park with Maputo Special Reserve and Tembe Elephant Park. We selected this corridor particularly because it displays incredibly high levels of connectivity. And as a result, corridors represent the principal pathway through which long-distance seed dispersal can occur. If connectivity among large fruited tree populations now depends on corridor movement, then the critical question becomes, are elephant corridors sufficient to maintain this connectivity between existing lala palm, for example, populations? So we combine this high-resolution elephant telemetry data with our gut-derived passage times. And then within our corridor, we modeled habitat suitability for elephants and then converted that surface into a resistance layer to estimate how easily elephants can move across the landscape. We then use this resistance surface to calculate spatially explicit cost distance dispersal within our 96-hour window. So we took our maximum gut passage time to see basically how far they can move in that time, but using this resistance layer to see where they're actually likely to be dropping these seeds. Next, we asked what this means for real palm populations within our corridor system. More specifically for 2 selected lala palm populations, we calculated the 95th percentile dispersal envelope after this 96 hours. And this is the corridor area. The background surface represents landscape permeability. So the yellow shows areas that elephants like to use, while the purple are areas that they don't like to move in. And then the red, brown, and green colored envelopes show the spatial extent of the dispersal. So the red is where it's highly likely, the brown where it's moderately likely, and the green where it's unlikely but still has the potential to move. And where these envelopes overlap, dispersal from different populations can reach the same area of the landscape. Excitingly, we did observe very clear spatial overlap between these palm communities within the corridor system. And this is compelling evidence that corridors are functioning as true ecological conduits and enabling this potential genetic exchange among these large fruited tree populations. And what was really interesting is that if you use, we do intend to use remote sensing and digitize the actual lala palms within our corridor system. But for now, we used iNaturalist, and if you look at the corridor, the majority of lala palm populations do seem to occur within the corridor. So we could be saying, well, the palms actually showing us where the corridors are, because I know in parts, when you fly over parts of Botswana and Zimbabwe, you can see elephant paths and then lala palms just along, at the edges of the paths. So yeah, we've got to do some work on that, and we're also looking at like the historical distribution within the corridor too of these palm populations, because I think lala palms, I know that they can live up to, or are thought to live up to, about 300 years, so we can get a sense for where elephants were moving. Our results show that elephants are enhancing recruitment and sustaining connectivity across landscapes. So in spatially constrained systems, corridors are not just movement pathways, they are ecological conduits for seeds. And if our corridor integrity is lost, so too is this plant connectivity function. Just thank you to Debbie for doing the lion's share of the work on the gut passage experiment and the germination trial. And thanks to Michelle, Cassandra, Sylvia, and Katie, and also Anton Powell and Sandparks, the APNR herd, MWA, and thanks to William Bond, who got me really interested in seeds and mega herbivores.
Thank you, Catherine. A nice interpretation and a nice illustration of some of those roles. We've got time for one question. Lisa?
Thank you very much. That was very interesting. I'm wondering about, I'm assuming that you might want to in future actually test like the genetic connectivity between these isolated populations and see if it matches your predicted corridors in elephants. And I'm wondering to what extent water movement contributes to the movement of seeds, and whether you've at all looked at the the difference in germination likelihood if the seed spends a lot of time in water, and if that also contributes.
Yeah, absolutely we are. That's our next step with another student, is to look at, to go and find these populations and check, take genetic samples and look at that. But yes, very good question about the seed dispersal through river systems, and I do think, have thought about that a little, and I think soaking the seeds in some studies has shown a bit of an increase in germination, so I'm sure it might be playing some role. If you look at where the palm populations are found in Mozambique, they're found within the corridor, and then some at quite a few along the coastline and at the mouths of rivers. So maybe there's some kind of backup dispersal happening through river systems, but I think elephants probably the prime, yeah.
Yeah, just to add to that, I think
Work
backup
through the
maybe
chairman,
also.
please. Work through the chairman,
Sorry.
please. Okay, finish it quickly.
But also, just in addition, not necessarily a backup, but,
Yeah.
linking isolated, and then elephants take it from there, something like that.
That's okay, yes, I'll give that some thought. Thank you.
Thank you very much, Catherine, and thank you, Lisa, for taking us into that discussion. We've been at the front end of elephants. We're going to go to the back end now, because the truth is in the dung, according to Aaron. Thank you, Aaron.
Thanks, everyone. As you probably all know, I'm Erin. I'll be presenting work focused on estimating seasonal elephant density using dung surveys as a part of ongoing elephant management or elephant monitoring within Kruger National Park. Although elephants have access to approximately 100,000 kilometers squared in the broader GLTFCA area, and much more, according to Catherine's one where they're going south even, active management within Kruger National Park remains essential. The Kruger Elephant Management Plan provides guidance aligned with the national norms and standards and the Park Management Plan. The plan is structured around 4 key management objectives: fostering meaningful stakeholder relationships, maintaining a resilient elephant population, and to manage the ecological and cultural role of elephants, allowing for their dynamic function in ecosystems, and then to facilitate opportunities and benefits associated with elephants, as well as promoting their value and ecosystem services. However, elephants' use and impacts are not evenly distributed across the landscape. Some areas experience consistently higher levels of elephant activity than others, which can lead to different ecological outcomes and require different management responses or interventions. To better understand these spatial patterns, elephant impact areas were identified in 2019. I redid the map just so it could fit on the presentation a little bit better, but these are basically the areas, and according to what was why they were classified as an impact area. These areas help managers assess where elephant influence is concentrated and guide management decisions across the park. To better understand these spatial patterns of elephant influence, we use dung density as an indicator of relative elephant space use. By quantifying seasonal dung densities across transect areas, we can estimate how elephant use varies across the landscape and between seasons. This provides a useful proxy for identifying where elephants are spending more time and exerting greater ecological influences. When these patterns are combined with biodiversity indices, they help provide a clearer picture of what levels of elephant use may be considered sustainable in different parts of the landscape, and ultimately support adaptive management decisions. However, for dung density to meaningfully reflect elephant density, it is essential to use locally calibrated defecation and decay rates. So this project consists of 3 components. Firstly, we directly observed individual elephants for 30-minute periods, recording the age and sex and number of boli per defecation event. Then, the second, we monitored 300 dung piles in around the Skukuza area from the fresh stage, so stage A. It's not included here, but it basically looks like B with a bit of bugs and moisture around it, until complete decay, which is stage E. Also not included here because stage E is defined as no longer visible within 2.5 meters. This allowed us to estimate seasonal decay rates using survival analysis. So that is the bugs picking at it. That generally does help it decay. And then it basically reaches E. For the third component, it involves transect-based dung surveys. We established 10 sites within 3 locations to pilot the methodology before expanding to the larger EIAs, as I mentioned in slide 2. Each site consists of a triangular transect with 500-meter sides. All dung within a 2.5 meter, all dung within 2.5 meters of the transect line are included and classified according to their dung stage. These dung form the basis for estimating dung density per unit area. Our total sample size for the defecation rate aspect was 33 observations, 21 in the dry season and 12 in the wet season. The average number of boli per dung pile was consistent between seasons. The dry season defecation rate averaged approximately 16 dung piles per day, which aligns well with Olivier et al., who in their review reported dry season rates between 12 and 16.6 defecations per day. Our wet season estimates was higher than values reported in their study. Given our smaller sample size in the wet season, this likely reflects sampling variability rather than a true biological difference. We then estimated seasonal decay rates using Kaplan-Meier survival analysis. Clear seasonal differences emerged. Decay progressed substantially faster in the wet season, particularly from stage C2 to stage E. Wet season time to full decay was broadly comparable to Olivier et al. 2009, whereas our dry season decay duration was notably longer. This may reflect local climatic and ecological conditions, vegetation structure, or sampling differences. So these elephant density estimates were derived from dung densities using locally calibrated defecation and decay rate corrections. Across all 3 elephant impact areas, densities fluctuated between years, indicating inter-annual variation in elephant use. However, a consistent pattern emerges. Dry season densities are always higher than wet season densities. This suggests that elephants concentrate their use of these areas during the dry season, likely reflecting seasonal resource availability and water dependence. While the magnitude of densities changes between years, the seasonal signal remains strong and consistent across areas. Our results demonstrate that dung surveys can reliably estimate seasonal patterns of elephant use when paired with locally derived defecation and decay rates. While defecation sample sizes should be expanded, the methodology has proven robust and scalable. The next step is to extend this approach to the various EIAs across Kruger, prioritizing areas of management concern. Integrating these data with biodiversity surveys will allow us to assess not only elephant density, but ecological impact. Ultimately, the truth really is in the dung. It provides a measurable, scalable, and management-relevant indicator of how elephants use the landscape. I'd like to thank the Large Mammal Ecology team for their commitment to this project, as well as other members of scientific services who assisted with data collection and field support. Thank you.
Thank you very much, Erin. You've done also a great job. We've got time for a question or two. Tony.
Thanks. I was just wondering how your estimates compare with the sort of general estimate for that area from the game counts. Is it the same sort of range?
Sorry, can you just repeat that?
I was wondering how your estimates of density compared with the estimates from the game counts, the annual census.
I can't recall. I can get back to you though on that. Sorry.
It's an important question Tony is asking. The scale is not comparable, Tony. That's one of our difficulties. We're working here on a very small scale, and it is because the interest is on the scale at which elephants are likely to have ecological influences. Tony and I'm having a discussion, and then Corley, and then we'll finish.
Yeah, no, it was just, I mean, that was the kind of the point. So you can't really take the large scale estimates as an indicator of how much impact they're having in that.
Ready? Corley?
I was just wondering, Erin, that if those 3 sites are spread quite widely again across the south, but that they all look like they're on the granites. So I was just wondering, A, whether there'll be a soil effect, but B, the visitation or the frequency of encountering them increases, across those sites in winter. So like, where do they go in summer?
I think they'd probably use the landscape. They just focus their areas because we're going based on dung count. So they will probably be using the landscape overall, but in the dry seasons, they're probably focusing it more there and using more of that area because they are probably there for maybe a water resource because there's not so much in the greater landscape. So they'll probably be using the whole landscape because they will need to eat and things, but there's water, and because of the water being in the river systems, there's probably better vegetation as well there for the elephants to hang around.
Thank you very much, Erin.
Okay.
We've come to the end of chatting about elephants, but we're going to ask Rob now to have another question on how do we think now elephants help us contribute to a living landscape or a breathing landscape. Thanks, Rob. You have got 10 minutes.
Thank you, Sam. All right. Any early thoughts? Pardon my voice. It's probably some bug from India or something like that. Any other questions for the speakers at this point in time, just before we kick off? Comments? Is anybody? All right. I would like you to sort of like imagine in a virtual, perfect world what a living landscape may look like for elephants, and what the reality is today, and what the differences are, and how you think we can achieve that sort of virtual or aspirational landscape. Any thoughts that you may have at an early stage? 1, 2.
I don't know if anyone has seen the mega living landscape maps for South Africa, but when I look at Catherine's movement data for elephants, I had 2 thoughts. The one was we really need to ask elephants where they want to be moving rather than us drawing those connections. And at the moment, they seem to be all in the wrong places. And then I forgot what my second point was, but I think that's that's good enough.
Thank you.
I was just wondering, what is the sort of time frame that we're looking at? How long is this virtual perfect world going to exist? Because in 5 billion years' time, Earth won't exist because the sun will have run out of fuel. But in the meantime, all the fundamental biogeochemical processes that have been going on for the last 4.5 billion years are going to carry on, are they not? Life will be there, water will be there until the sun burns up. So what is the time scale? 10 years, 100 years, 1,000 years, 1,000,000 years?
No, because it's a fantasy world, you can decide for yourself. Tony, thanks. Yeah, Tony, got him.
Sorry, my question's not nearly as deep as Peter's, but anyway. So I was just wondering, what is the actual objective of the Mega Living Landscape strategy? Is it to protect species or systems that are not currently represented in national parks? Or is it to provide conservation corridors for things like lion and elephants that are becoming difficult to manage? Because it's two very different approaches. And I mean, things like elephants and lions are currently protected in national parks. So if that's not the objective, then why all the concern about how to have them living with people outside parks?
I just want to say that I think that virtual landscape certainly looks to me like an area that has protected areas, but then the connectivity between them is of prime concern, and the benefication of people. So, you know, in the protected areas, it's not human-dominated landscapes, but in the connecting areas, it could be human-dominated landscapes, and that's where we have to think very innovatively about benefication for people and how that should maintain the connectivity, because both those elements are very important in a mega living landscape. Thank you.
Thank you both. Anybody else? Immediate thoughts? Paul?
I've been struck by how the main focus is looking at which state we are going to transition into and the fact that we need to prepare for that. But it seems with all the predictions that the state we're going to transition is transition. It is a constant flux and it is going to be continuing to move. And we've got predictions up to 2021, but it's going to keep moving on there. So just saying, you know, we've got to prepare for the next phase. It's not a stable state that we're preparing for. So maybe it's actually the change we should be embracing rather than what we're actually changing into.
Correct. We seem to be fixated on patterns and not really focusing on the ecological processes to achieve that. Sam, you?
Thanks, Rob. And I'm hearing this fight between what should a system look like to what should a system function like. But I'm also hearing extremely ecological elitism. We are asking questions about, you know, which is ecologically focused. We're not even wondering, well, would other people also like to have elephants in the landscape? And how would they like to have that elephants in the landscape and so forth? And how does this big thing that is walking around here now busy contributing to all these other things, you know? So perhaps, I think Lisa highlighted it right in the beginning, that one of our very first key questions is agreeing what our imaginary future virtual should look like, you know, and that brings in Peter's question about, well, what is that time frame? And I think most people, that time frame is probably about in the 30 or 40 years that you're productively thinking. You know, that's just a comment.
Thank you, Sam. Lisa?
Yeah, thank you very much. I think, so largely in agreement with what was said just now, but earlier, Joe had commented on the huge change that you can see in the community driving up, like the development of the community driving up to Kruger Gate, right? And every time I come back, I just think about when I was a master's student and I started here, and what has, you know, happened in terms of the development of these communities. And not to in any way minimize the complexity of conflict with these animals, but if the question was, what does carnivores contribute? What does elephants contribute? And that is one of the contributions that I think is very visible within this landscape, because tourists are not coming here to see our, you know, I don't know, even though they might appreciate the landscapes and the trees and all the additional things. They come for the elephants, they come for the predators, and that's a huge contribution that I think should be perhaps worked into that vision that we have of the future, with a connectivity that also spreads that benefit further and wider. And again, not to minimize the complexity of that, but I think that should feed into what our vision for the future connectivity of these animals are.
Sally, thank you, Lisa. Sally.
Thanks. I've really found these discussions stimulating. I think when we're talking about tree-grass interactions and, should we have more fire regimes, we're quite comfortable with the fact that we apply a management intervention and there are winners and losers. The grasses benefit and the shortgrass birds benefit, but the trees lose. But when we're talking about having these multifunctional landscapes with elephants wandering past your primary school, we sort of want to just tell everyone that we're all going to benefit, but there are going to be losers. And I think that's what Sam is mentioning. So I think we need to engage as society with the fact when we have this vision of the future that we all aspire to, there are still going to be parts of society that lose out, or who have lost opportunities, or who are more at risk. And instead of trying to persuade them that they're benefiting, we should be thinking really hard about how to compensate them. And I know that comes with very complicated economic perverse outcomes, but we should be listening to the people who are talking about human-wildlife conflict. And how can you compensate people when they're the losers in a way that still results in healthy socio-ecological systems?
Very valid point. I mean, we sit in a very safe space here among 30,000 plus elephants and all the other animals, and we can debate these sort of things because we're safe and secure, but there are people out there not so. So definitely something to think about. Any other? We have a couple of seconds left. If there's any last comment, then I'll hand over back to Sam. Thank you very much.
Thank you very much, Rob. Give a round of applause to all our facilitators. We had a lovely morning session. I'm going to ask Lawrence to come around as quickly. You need to make an announcement. But I really enjoyed the interaction coming from the floor. It is a little bit of an older part of the community here. What are they now? I can't actually say the colored hats because that's not necessarily age, but it's been very useful to think about this, and there's definitely a tendency for us to start thinking far more complex, process-orientated, but recognizing that it's actually not our own objectives that matter. It's a collective of society's objectives that matter, and we may not have defined that very well. Thank you, guys. I appreciate it. Lawrence?
Thanks, Sam. So speaking of mega herbivores, just a quick announcement regarding tonight's memorial dinner for William Bond. We have had an overwhelming number of people apply for the evening. We had expected about 30 or 40. It turns out it's at least double. And so that's going to require some serious nimble logistics. So the first problem is transport, and we only have 15 parking spaces at the SSLI. So we'd like the local drivers to we'll prioritize them, but carpooling is crucial. What we'll do from 5 o'clock onwards, we'll have a sequence of game drive vehicles that will be passing through the main parking lot here, and all the remainder of you lot who've signed up for this evening, hop in those vehicles, and we'll transport you to the SSLI. We'll start the evening at about 5:30 with some presentations and work our ways to the dedication event. We're joined by his family members this evening, which is fantastic, and the dinner will be served at 7 o'clock this evening, and then the transport back to the main camp will start at about 8:30. So please do bring your own, for those who have signed up for the event, please do bring your own drinks. We'll supply the meal, but please bring your own drinks. So just to reiterate, I've sent out an email to everybody who signed up, but for local transporting, for local people, you can drive there, carpool. People who are not living locally, please meet in the parking lot here and we'll start transporting you through, and bring your own drinks, please. And I think Sally just wants to mention something. Oh. You may. the parking lot right here in front of the center. Yeah. All clear? There's also an email that went out, and then just another quick announcement.
Thanks. Sorry, this is also linked to, I guess, to the William Bond Memorial. So there's a few of us have been talking about organizing special issues, journal special issues for William. So we're thinking of putting together an issue for Phil Trans of the Royal Society, and I know there's been discussions about the GSSA journals as well. So what we want to do is kind of gauge interest in being involved. And so what we're going to do is send an email around to the people who are going to come to the memorial, and there'll be a link to a Google form, and you can express your interest. So we're not guaranteeing that you're going to be in a particular issue, or we just want to see who's interested and what kinds of things are people interested in publishing on. We'll probably also ask you how quickly you could get your paper together, because obviously lots of people are interested, but perhaps you can't get it together in the time available, if you're like me, for instance, probably. So there'll be a very short questionnaire, and if you're interested, fill that out. Okay, thank you.
Great, thanks for that. So just 3 quick things before we break for tea. So on the program this afternoon, there's supposed to be a choir performance. Unfortunately, they won't be able to do that this afternoon. So we will be going on to the wrap-up and thanks immediately after the last presentation. Then please also, we do like to keep our name tags. So please keep your lanyard, and after lunch, if you can just pop the plastic part of it at the bottom into that little box there by Jackie, and we can then use them again next year. And then anybody that has a poster up, we're going to be taking them down after tea. If you'd like to take your poster back, that's great. We also every year put posters up in our offices. So if you don't want your poster, please leave it here. We will definitely put it up. And then we have run over slightly, so we'll start again at 11:10. Thanks, everyone.
it's still morning. All right, morning everyone. If you can please take your seats so we can start the session. All right, we are going to continue with our session on the breathing landscapes. There's going to be, once again, 4 presentations, and then we'll have a discussion session afterwards. All right, Stephen, Mitzi, please, can you take a seat? We need to start. Thank you. Thanks, everyone. So our first presenter for the session is Samukelisiwe, who's going to be talking to us about the effects of water impoundments on the distribution and activity of elephants in southern Kruger. Thanks, Samu.
Okay. Good morning, everyone. My name is Samke Ngobo. Today, I will be sharing our findings from a paper we got published early this year. And I would like to acknowledge all the co-authors highlighted in this publication. In semi-arid environments, the scarcity of perennial water sources compels elephants to move between water and forage areas. These movements can allow the vegetation adequate time to recover following intensive use by elephants. However, the widespread creation of artificial water impoundments, which I hereafter refer to as reservoirs, can disrupt these natural movement patterns in many areas, intensifying elephant activity. Consequently, biospheres can be created, which can routinely lead to vegetation degradation and subsequent reduction in biodiversity and ecosystem heterogeneity. For those who are unfamiliar with the term biospheres, they are essentially regions of increased vegetation pressure due to the concentration of herbivores. So when reservoirs are poorly or haphazardly distributed, they can result in intensive use of certain areas, heighten thermal stress, and impose additional ecological pressures on the surrounding environment. While we do know that water influences elephant distributions at large scales, we know surprisingly little about how individual reservoirs shape elephant space use and daily behavior across seasons. Therefore, we examined how individual reservoirs alter elephant space use compared to adjacent undammed catchments, which is crucial for our understanding of the direct effects of reservoir additions to landscapes. Specifically, we addressed the following questions. Firstly, how does reservoir presence influence elephant spatial distribution across the seasons? And how does seasonality and water availability interact to shape elephant daily activity patterns? We applied a paired catchment camera trap design where we compared 11 paired catchments along 3 random transects covering a 2.25 kilometer gradient in southern Kruger. Each pair included 1 reservoir site that provides permanent water and a matching undammed drainage in a similar catchment. This will be crucial for our understanding of, each pair included a catchment with a reservoir that provides permanent water and a matching undammed catchment in a similar drainage for a total of 22 sites sampled seasonally. This design allows us to isolate the direct effects of reservoirs from broader landscape variation, to show how strongly and how far that impact of artificial water provisioning propagates from the reservoir. We collected our data during the wet season of 2023 from October to November, and in the dry season of 2024 from May to July. A total of 15 camera traps operated from 10 to 14 days were placed along each transect, resulting in 330 camera stations sampled seasonally per site. Total camera trapping effort amounted to 3,120 camera days in the wet season and 3,460 in the dry season. We analyzed our data using generalized linear mixed models in R. To refer back to the first question that we posed, how does reservoir presence alter elephant space use across the landscape? This figure will give us insights into that answer. The lines that are in blue represent reservoir sites, and those that are in orange represent the non-reservoir sites, where reservoirs are absent. So looking at the wet season, we see there is a decline in elephant detections the further away we get from reservoirs, and the opposite effect for elephant detections at non-reservoir sites, where there is an increase in elephant detections extending even up to 2,250 meters. While in the dry season, there's only a drastic decline in elephant detections with increasing distance from reservoirs for both reservoir and non-reservoir sites. This may suggest that elephant spatial distributions are associated with reservoir presence and proximity to water, with seasonal variations influencing these effects to some degree. So the second question that we posed was, how does the interaction of season and water availability shape elephant daily activity patterns? So we see here, during the wet season, elephant activity peaks early in the day at reservoir sites compared to those elephants that are at the non-reservoir sites where their activities peak later in the day. But we see that during the dry season, there's a switching in this temporal pattern where we see that elephants at non-reservoir sites are primarily active later in the day, and those at reservoir sites active earlier in the day. This may indicate that reservoirs can prolong elephant activity, especially during the dry seasons. So, in summary, our results reveal that reservoirs do reshape elephant spatial and temporal footprints seasonally. Firstly, we showed that reservoir presence concentrated elephant space use near water during the dry season. This may suggest that elephants optimize their behavior to minimize thermal stress and reduce energy expenditure, especially under heightened nutritional constraints driven by declines in water availability. Secondly, we showed that elephants extended their activity at reservoir sites in the dry season. This may indicate that reservoirs can provide a reliable water source that enables continued elephant activity even during the hottest parts of the day. So taken together, the concentration of elephant pressure near reservoirs raises concerns to the possible potential localized habitats and vegetation changes. From a conservation and management perspective, our results emphasize the importance of reassessing the role of reservoirs and their distribution within Kruger. To mitigate the effects that we highlighted, a more strategic or rotational water provisioning strategies may be necessary. This would promote or encourage rather a more varied distribution of elephants across the landscape, thereby reducing pressure on localized areas and enhancing ecosystem heterogeneity. This is crucial for maintaining biodiversity and enhancing ecosystem resilience. However, we did not explicitly examine the effects of reservoir-associated elephant distribution on either vegetation structure or ecological processes. And therefore, we encourage that future research address this knowledge gap by testing these effects directly. These insights will be vital for developing sustainable management strategies that balance the ecological requirements of both elephants and other species and the broader ecosystem. I would like to acknowledge the collaborators and funders of this work, and especially thank you for your time and attention.
Thank you, Samu. Unfortunately, you've used up your time perfectly, though. But thank you for your talk. I'm sure if you've got questions for Samu, you can either raise it at the discussion session later or grab her during lunch break. Up next, we've got Rob Hildemond, who will present how dispersal drives the recovery of the elephant population in the Greater Limpopo Transfrontier Conservation Area. Thanks, Rob.
All right, good morning, everybody. Ever since the establishment of the Great Limpopo Transfrontier Conservation Area, or the GL, some decades ago, life has changed for elephants and people alike. And there's great concern among the 3 countries and several institutions what will happen in the future. And for this part of the presentation, I'll just be focusing on elephants per se. I guess the hidden question behind it all is, can we find any signals of density dependence that may act as a regulator of population dynamics? And part of our challenge, unlike other transfrontier conservation areas, is that we don't have repeated systematic aerial surveys that cover the entire region. So for the first decade, say around 1995 and 2005, after culling was stopped in the late 80s in Zimbabwe or parts of Zimbabwe, and in the Kruger National Park in the mid-1990s, we also saw fences dropping and water holes being closed, particularly in Kruger. As the blue color becomes darker, densities increase within the 21 or 22 populations that we define here. With poaching being addressed, and mostly successful, densities kept on increasing for the midterm period between 2005 and 2015. And for the last 10 years, as elegantly showed by Michelle Henley's group and by Bob's work up in Zimbabwe, elephants started dispersing beyond protected areas and coming closer into contact with people, even beyond the GL as we've defined the boundaries. So Bob Ngonirizou hosted us last year in July, got a couple of people together, and we decided that we're going to address this question and see if we can get a better handle on what populations are doing across the GL. Our underlying model is the adjusted Eberhard cascading model. It's adjusted, it's adapted by Sam and the other Bob, and currently in review. And it's all about how vital rates and dispersal may start kicking in as densities increase, particularly when elephants are reaching an ecological benchmark value defined for the region. I'm just going to focus on one particular region, specifically here in Kruger, as is the case in all good quality undergraduate textbooks. I'll be using an example where everything works perfectly. All right. Right, so we extracted all the survey data here in Kruger, a total count as depicted here for Marula South. Our next step, and what we did, and if I say we, I mean the collective noun for we is SAM, then transform it into survey estimates, so turning count data into estimated means, and with a 95% confidence limit around it. We then test for directionality of the trend over time from 95 and up to 2025 or 24 at least. And here we notice that from about 2015, that the elephant population now has a density that falls within the ecological benchmark, a definable benchmark values. Then the next step is to take all those survey estimates and using a GAM type smoother, we just removed a lot of the noise from the trend data using a regression spline approach. And it's those smoothed estimates that are then being fitted with 3 different types of population models. Here, the exponential model. The 3 different models gives different indications of what processes are driving that population at that point. So we fitted the exponential model, or the Ricker model, or the theater logistic, all 3 of them. And then using a model selection procedure, in this case, the theater logistic is being received the most support using the model selection standard procedures, which, of course, as I said, is very good news for this population, hence the example. We then solved the Ricker model analytically by regressing the population size in the following year against the current elephant population size. And if we see indications of density dependence based on numbers alone, we expect this curve to be relatively concave. So the number of new animals being added to the population is becoming less and less relative to the increase in the number of elephants for the year-end T. Final test that we did to evaluate whether some of the populations show early signs of density dependence, is that we fitted a quantile regression at the 90th percentile when we plot growth rate, annual growth rate against the population size for that particular year. And again, model selection, if you find this pattern illustrated over an increase of elephant abundance, we may deduce that density dependence are starting to play a role for this population. So this we repeated 21 times with all the populations giving us reliable estimates. And we find that 12 populations are below the ecological benchmark value, so we don't expect vital rates or dispersal to indicate any driving the population dynamics. 8 populations are within the benchmark envelope, of which 3, Marula North and South and Kruger and Gonorrhoeae, are showing early signs of density dependence. When we add all the population estimates, all the modeled population estimates in 2024 across all populations, our best estimate currently sits at just over 56,000 elephants within the GL landscape with relatively wide confidence limits. And we currently just refining and touching up all of this analytical pipeline, and then hopefully we'll then submit for peer review. Right, thank you very much, and Sam is on standby if you have any questions.
Thanks, Rob. I like the way you're shuffling everything to Sam. Thanks, you've presented well. So there is time for one question from the audience.
Thanks, Rob. That was a really clear presentation and a very encouraging message. What I'd just like to get some clarity on is you talk about ecological benchmark. Is that a euphemism for K? And if so, how did you derive it?
That value has been determined by a publication where Rudy and co-workers, the paper about that Africa, sub-Saharan Africa, only have about 25 or a quarter of its possible elephant populations. Within the appendices of that publication, all the different benchmark values is being projected there for the whole region, with and without poaching. And the one that we chose is the one without poaching, just to reflect on the natural state. And simply things are doing very well in the GL. So it's a published estimate as a referral point. It's not set in stone. It's got very wide confidence limits, and we're only looking at the bottom half of that, below the mean. Yes. Thanks. Thank you.
Thanks for that, Rob. Up next, Brooke Friswold will enlighten us on how range expansion improves elephant well-being and how it facilitates reciprocal ecosystem recovery. Thanks.
Hi there, thank you very much for that introduction. So yeah, I'm Brooke Friswold, presenting on how landscape connectivity enhances elephant ranging flexibility, relieves browsing pressure, and reduces behavioral and physiological stress. So overall, we wanted to see how a resident elephant population in a private reserve responded to internal fence removal, thus landscape connectivity. So we assessed spatial, ecological, behavioral, and physiological responses. So this study took place at the Kruger Reserve in the Eastern Cape, which is classified as an Albany thicket biome with about an elephant population of about 76 individuals. So at the beginning of this study, the elephant portion of the reserve was separated by two large internal fences. So on one side, we had an elephant population of about 55 on about a 3,000 hectare area. And on the other side of the reserve, we had about half as many elephants on about double as much area. So there was two internal fences removed. The first one did not integrate the two herds and was removed at the beginning of this study. But the main fence removal that we studied was this internal fence removal in January 2024 that did allow for the two herds to integrate and for them to move across this contiguous area. So our methods included identifying the entire elephant population, conducting 14-minute focal samples of individuals, including both count and duration-based behaviors, applying GPS collars to elephants on both sides of the fence a year prior to fence removal and then following them 2 years after, as well as using GIS and Landsat imagery to conduct ecological assessments. We also assessed for FGCM's stress hormone present in dung prior to and after fence removal. So kind of a side note, an unintended study that we encountered was collar twisting. So we applied 6 AWT LoRa XL collars to 3 cows and 3 bulls. And unfortunately, within 6 months of application, all of the collars on the bulls twisted substantially. These were applied by a skilled vet team to the correct specifications. So we investigated this thoroughly and did a publication on it, and it was concluded that the twisting was a result of browsing behavior by bulls, so pushing through that dense vegetation, as well as potential flaws in the collar design. So overall, we strongly caution use of these collars on bulls in this dense shrub terrain habitat. Okay, back to the study. So prior to fence removal, so on the left, we can see that there was heavy habitat use and clustering in the southern region of the reserve. So high hotspot activity and localized pressure. After the fence was removed, we saw that the elephant habitat use shifted westward and northward, really reducing that intensity of localized hotspots. When we looked at home ranges, we saw a substantial expansion of elephant home ranges and a median annual utilization distribution increase of about 30%. And matriarchs about doubled their home ranges by about 2,500 hectares. Pre and post overlap was generally low right after fence removal, indicating high exploration of these new areas. So when we looked at 1 year of movement data following fence removal, we can see, so a little movie here. So this is when the fence came down. The elephants are moving from the more densely to less densely populated side of the reserve. They're really exploring it heavily, moving westward and northward. However, you can see that after a bit of time, they start utilizing semi-coordinated movements from the north to south and east to west, as opposed to clumping up in the southern region as they were doing before. So they have this initial spike in exploration, but then they return to familiarity as well as seasonal pattern and coordinated movements. When we looked at seasonality, we also saw quite a difference in seasonal patterns, especially in the winter time, being much more broadly distributed. And for elephants on the less densely populated side of the reserve, we saw a less difference in seasonal distribution. When we looked at association, so via proximity alerts and the collars, we saw that all elephants, after the fences came down, were spending more time together, which was a bit of an opposite of what we thought would happen. And this included elephants that were and were not previously separated by the fence. There was a short transition lag, probably high exploration, followed by extended, not just proximity, but co-traveling, indicating that there is higher spatial overlap between herds and then potential social reconnection. Using hidden Markov models, we separated movement into different behavior states using GPS collar data. So we separated that into sleeping behavior, low energy behavior, movement, foraging, and bounce. So bounce included bouncing off of a fence line. There was a really fascinating ghost fence effect that I won't be able to get into here, but we notice that foraging peaked during daylight hours using these Markov models, sleeping peaked late night and early morning, and movement peaked midday afternoon. However, after the fences came down, bouncing behavior decreased to almost none, indicating unimpeded exploration. Foraging decreased and then stabilized. Movement increased substantially, and sleeping and low energy behaviors also reduced and was sustained. But overall, there was no disruption in circadian structure. Using resource selection function models, we looked at where they are engaging in these behaviors. So looking at low energy and sleeping behaviors. Again, these were clumped heavily in the south prior to fence removal. And then after the fences came down, there was a marked expansion into these newly accessible areas, redistributing their sleeping and low energy behaviors across a broader landscape and overall reduced spatial clustering. When we did the same for movement and foraging, we found a similar result. So the movement and foraging areas also expanded with broader landscape coverage, and actually with movement corridors, we saw increased connectivity, not just across terrain, but across different terrain features. And this again increases their range, which is reducing that hotspot amplification we were seeing before. When we looked at habitat suitability, so the greater suitable habitat is in red and the less suitable habitat is in purple, we saw that elephants were generally migrating from areas of lower to higher habitat suitability. But then after the fences came down, there was a net gain in habitat suitability for the entire reserve and was more broadly spread out. We also saw that habitat selection by elephants changed after fence removal. So prior to fence removal, they were selecting based on about 12 variables, and after we found 18 significant variables they were using for selection, which can obviously, again, reduce localized pressure and encourage more natural processes. When we looked at NDVI, so just the level of greening throughout the reserve, we compared it to areas in the reserve that did and did not have elephants. So there's two portions of the reserve that had restricted elephant access. And when we compared NDVI across the whole study period, we found that elephant use areas and non-elephant controls had the same amount of NDVI losses and gains after the fence removal. There was also a large decline in rainfall during this window, contributing to overall losses throughout the reserve, but again, these were not amplified in the elephant use areas or after the fence came down. So we're seeing that NDVI was largely driven by climatic patterns rather than elephant presence or absence post-removal. When we looked at behavior using those focal samples, it kind of corroborated what we saw in the GPS collar data. We saw a reduction in foraging by about 20%, an increase in locomotion by about 100%, and as well as an increase in vigilance. So right after the fence came down, we saw an immediate response increase in active aggressive behavior. This is mostly towards conspecifics, other elephants, as well as stress-related vigilance and affiliative or social behaviors. However, when we looked at it about a year later, we actually saw that aggression, including active and passive, declined lower than pre-fence removal levels. And stress only remained moderately higher. Vigilance and affiliation remained increased. And when we conducted GLMMs, we saw that fence removal and time since removal were the greatest predictors of behavior, and that bulls were the most strongly impacted. They observed the strongest effects in bulls. When we looked at the physiological stress, so via those FGCM hormones, stress hormones in the dung, we saw that there was a statistically significant reduction in stress hormones in dung for all elephants. And then when we separated them into sex and age classes, we also saw statistically significant reductions across all sex and age classes. And then you could see a pretty clear decline after the fence removal there, followed by an increase in spike, which we think was due to all of the activity happening during the fence removal process. So for management implications, we can see that concentrated browsing pressure appears primarily driven by restricted space and not necessarily elephant overabundance. And that increased landscape connectivity via fence removal reduced chronic and behavioral stress. It increased natural movement, behavior profiles, and social dynamics. And it shifted their activity budgets towards a more natural baseline, so more movement, exploration, and less low energy and idle foraging. And expanded elephant movement did not produce a disproportionate vegetation degradation compared to its non-elephant controls throughout the reserve after fence removal. And overall, it enhanced ecological resilience through increased habitat suitability of the reserve and seasonal rotation patterns, and really reduced localized vegetation pressure and hotspot browsing, and overall can benefit tourism value and safety through more natural elephant behavior and less aggression. So we encourage that connectivity restoration can offer a low disturbance alternative to culling, translocation, and other intensive management interventions. I want to thank everyone who was involved in making this research possible, and if you have any questions at all, please feel free to email me. Thank you.
Thank you, Brooke. I'm sorry you have run out of time, so we don't have time for questions at the moment. Up next, it's the other part of the collective we that Rob was talking about. Sam is going to chat to us about ethics, bullets, and dart guns, and present a wide mix of challenges in elephant management. Thanks.
Good morning. I was still in the morning now, sorry. I suspect if any of you have got a problem in the room, you're probably eventually related to elephants. And I think Nico was busy making elephants the cause of South Africa losing the game yesterday in the Cricket World Cup. That was a very common thing in this entire discussions and so far. I've heard lots of elephants and things coming up in just about every session around the case. But we'll have to ask some things. Right now in South Africa, we've got a bunch of rogue and roaming elephants around some parks. We have got some refugee elephants that decided to go to Swaziland. We want to chase them back, but we need to get a CITES permit to do that. And we've got tree thrashing elephants. We heard a lot about that. Okay, so those are the sort of things. Now we have to ask, well, why the hell have we got these problems when we've had a national elephant management norms and standards since 2008, supposed to sort out these problems? And I suspect that part of it is because the moral complexity and factual uncertainty challenges that we had 40 years ago are actually not gone yet, or we have got certain stakeholders that try to maintain that sitting around here. Moral complexity sit around rights, welfare, and also balancing rights between what an elephant wants and what people want. This justice. The factual things, a lot of debates. We've just had Johan ask about the ecological thresholds. I can guarantee Johan there will be a lot of debates about that. So there is one additional thing that happened, because there are legal risks when you deal with this, the way that we rationalize what we do. So I thought, let me quickly reflect on these sort of things. The very first one is dealing with what are our ethical framings. And there's roughly about 8 or 9 major ethical framings in the world. I'm not going to go into detail out there. The difficulty here is that I can logically argue for or against a controversial intervention like elephant culling using any one of those framings. So what matters now is whose ethical values matters. You know, it's really difficult if you start making decisions based on the ideologies that we pursue rather than some other framings. Now, there is some things that Darwin did for us. The very first one is Darwin figured out what evolution did, and we realized that we cannot exist without biodiversity. None of us has got chlorophyll. So we have to utilize it. That means it imposes values in us, like sustainability, thankfulness, etc. And that imposes perhaps a framing of what kind of duty do I have towards biodiversity, and towards elephants for that matter. So if an elephant is stressed, have I provided the opportunities for that stress to play out the coping strategies that they've evolved? That's Darwin's, by the way, his second favor that he's done us. So yes, values shape our ethics, and the ethics shape how we apply those values, you know, to how we make decisions. And we argue that if I'm thinking about a duty of care, you're far better off doing it that way. The one that is contentious is actually the fact that when we look at what elephants do in the landscape, we had a lot of those discussions out here now, there is not a simple link to how many elephants you've got in the landscape. This comes from an extensive meta-analysis that sort of evolved over the years. Now we have about 72 studies that are suitable for that. And when you look at that graph on the top there, any value above that horizontal broken line is a positive, and any value below it's a negative. It's really difficult to actually see there's a pattern that there's going to be more negatives or more positives as elephant densities increase. And I asked the guys to filter out Kruger, and the same thing with Kruger. We have lots of positives and lots of negatives as elephant densities increase. This forced us to really think differently about the ecological framing. We required a paradigm shift, and one of the things that Bob McGleary and I have argued is that it's actually about the activity. It's about where they are, how long they are, what are they doing, who's doing. A bull elephant is doing very different things than a cow elephant, you know. And it is also dictated by what kind of resources: water, food, comfort, safety. Think about a discussion a bit earlier about landscapes of fear. Now, what you typically see when you look at those sort of things, we typically have two kinds of systems. There's an unconstrained system. This is often what Kruger is like, and you can think about those little green spots as nice elephant habitat. There's no corridors there yet, okay? And my seasonal home ranges don't necessarily overlap. They're very comfortable in being separate places. You just saw what happens from Brooks' presentation when those elephants are spending a lot of time on the same place. We often inherit things on the right-hand side, which is in our smaller parks. I've got a fence around the place, I put water in, the elephant is spending a lot of time. So if I understand these mechanisms, surely they're not going to be the same everywhere. So I've got to start thinking about how do I do things differently in these places. Rob has just shown you what happens, what we expect should happen as an elephant population increases. That is in unconstrained systems. As soon as I get into constrained systems, that cascade, moving, weaned calves dying more often, cows having calves at older ages, eventually old ones die. That doesn't easily happen in these small reserves because the constraints doesn't happen. So we have to think very differently. All right, that's ecological framing. How do we think rationally about things? Surely if I'm managing elephants and my purpose is that I want to deal with elephant impact, then I must see a consequence in elephant impact. And we've now seen if I simply just maintain a number, that doesn't mean the impact has changed. Okay. So here's an example of work that we did. We said, imagine you have a population and you need to reduce it by half, and once you've reduced it to 1 year by half, I'm going to maintain it by contraception. So this is what you see what you see here. It popped down and now successfully maintaining that population. But did I actually change anything to how much foliage the elephants need? I did a lot when I dropped them, big time. That's the biomass, that's the foliage use. But over time, because small elephants become big elephants, my total biomass that's needed in there is still increasing. And on top of that, over time, an age structure looks like this now. And over time, I ended up with just a bunch of old elephants, mostly old elephants. So now, how do you make these trade-offs that's sitting out here? And we actually fiddle it around with this, playing around and say that same scenario, I want to reduce them and then I'm going to maintain them at half the thing. I can do it in many, many different ways. So this is the second bit. The first bit was about your rationale. My rationale was reduce elephant foliage or elephant impact, but when I applied the techniques that are sitting out there, my rationality failed because I still had an increase. Okay, so that's a rational test. The proportionate test is asking, well, is there things that I can do in a similar way that is going to require less intervention to be able to achieve the same outcome? So here's an example. Anything in this picture that are diamonds is when you are fiddling with survival or movement, age-specific, anything. Note that many of these things are not allowed by law at the moment. Okay, and anything that's got a square is where you're fiddling with the fecundity element of the population. And then these little round blobs is when you actually start thinking, well, let me do integrated things where I do combined things. And what typically happens is I can get really big consequences. So my impact is big. I reduce foliage use quite big. But look at this difference is what happens when I am intervening at the fecundity scale. I got to intervene with quite a lot of elephants over a 25-year period. And when I'm intervening with the mortality or the survival scales, I can potentially do that far less. You know, I'm actually getting the best sort of outcome here because this is now combined things that I'm working on. And if I look at what happens to the disruption of the age structure over time, anything that I'm dealing with on the fecundity schedule eventually gives me a very big disruption over time. So how do I make those trade-offs is really, really important. Now, what is really concerning for me, and I spoke to Michelle yesterday, under our current legislation, we tend to think about whether the actions is legal under the norms and standards. I'm not allowed to remove just one elephant. I have to remove a whole herd of elephants. But we forget about that there's an administrative action. There's guidance that tells you if you do something, you have got the responsibility that what you do is actually going to deliver the thing that you said you want to do. That's the rationality test. And in fact, I checked the various elephant management interventions, and I've given you examples of contraception and culling here. How are we currently doing it? We can do it very differently. We will struggle to actually survive the legal test of a rational and a proportionality in our current administrative justice. So some of our managers may actually be liable for a criminal act if some smart guy goes and says, well, I'm going to check and see whether you are actually complying with these things. So how do we overcome this? Well, one of the things is we realize that things are not the same everywhere. So I have to tie to what I'm trying to achieve. I think Ellen highlighted that somewhere earlier this morning. And specifically ask, how do elephants affect my ability to achieve my objectives? If I know that, then I ask, well, if that is how they do it, what can I do to interfere with it? Okay, and every intervention that I do is going to have a risk. I then ask, what is the mitigation that I can do for that risk? What we forget is to ask, what are the opportunities? And I think, Madam Chair, claim boss is what I call her, it's probably time, in particular all the red hats in the room, please don't go and do more of the same, what all the other colors in the room has been doing. It is really time for some serious rational elephant management. Thank you very much.
Thank you, Sam. You've used up your time, unfortunately. Okay, so I will ask Lisa Leroux to please come and assist. She's going to be facilitating the discussion session, right? On what should the system look like? I know we've alluded to some of this already in the morning, in the previous discussion sessions, on what the system should look like, or should we ask, what processes should the system sustain? Thanks, Lisa.
Thank you very much. Yeah, so we already started discussing this a little bit in the morning. Johan mentioned some of the, shifts in focus that we might want to consider, and I think it's a good time to start having this discussion, specifically in terms of thinking about what are the conceptual tensions that comes with moving from a shift in status of what does a system look like to a shift in process-based thinking, like what are the processes that is part of a functional system, and then also the practical implication or application of this. So if anybody wants to add to this conversation, I'd love to hear your thoughts. Joe in the back.
Thanks, Lisa, and thanks for the exciting talks this morning. I have a few points that I want to raise. Maybe that will help us maybe craft something more practical. The first one is elephants respond slowly, density-dependent issues. One, because they are clever buggers themselves. And two, they are not affected by very simple things. That's the first one. And the second point is we are giving them space to move around. So it means it will weaken even further the density dependence aspect. And elephants are long-lived. So if we want to come up with the real solutions and not be disappointed with what happens later, those are factors that we need to think that will affect the outcome of whatever we put in place. Thank you.
Thanks, Joe. But I also think that we, in discussing like what are those outcomes? Are those outcomes some picture that we have in mind of the system, or is it more process driven? Is that the outcome that we should be chasing? Anybody else? Sam?
So I'm really glad that George has asked that, because this is one of our challenges. I think Sally also highlighted around this. We are working with various ecological processes that are happening at different speeds, at different paces, but our societal expectations change rapidly. And one of the problems for us, I think, in a living landscape, if you just think a little bit about where people are living now and what their expectations will be, we may not even know when do people consider that their expectations have been met. But one of the 8 challenges is, so for example, the Kruger management plan is about, 10 years old now. I think we're reviewing it in a couple of years' time or something like that. Just about when the minister signed off the plan, the societal expectations has already changed. So how do we accommodate that sort of thing when I've got different processes that are running at different rates that are also influencing each other?
Anybody else? Oh, lots of people. We've got a cluster over there. All right, yeah, go for it.
Yeah, thanks. I already had the mic, but I just wanted to confirm what I think is Joe was talking about with the longevity of elephants. I mean, to think of it, there's probably an elephant walking around Kruger today that was still a baby in the 1960s, when a lot of the things were changing, from the movement of the people to the borders of the park to the opening up of water and that kind of stuff. So I think one of the realities of it is that as much as we might have a perceived system that we want to see, there's no way that we control the perceived system that the elephants wants to see. And elephants being long-lived animals, particularly elephants, as much as us as humans have got a memory of our forefathers from 400 years ago, elephants have the same. And you can imagine elephants have been in a space where they were persecuted for a long time. From the Arabs trading in ivory to whatever has been happening, elephants have been in that space where they had this connection with humans, either negatively or positively. It doesn't matter what you look at it. But, you know, it's a very interesting thing now in modern times because we are living with elephants predominantly in protected areas. Back in the day, elephants were not in protected areas. They were roaming around the landscape quite wide and far. So it is a very, it's a big challenge for us today, I think. And I think we need to have this conversation. This is like where we start unlocking also things and thoughts and so forth. Thank you.
Talking about process and landscape, I think one of the things that I haven't really picked up is the fact that we're going into a new reality of climate change. In 2000, we had the major, large and frequent disturbance, and I've lost count of how many disturbance floods we've had since then. And certainly drought's going to have a long-term change in the landscape, so we need to bring in those realities into any functional thinking.
Yes, for sure. I often think about, if I think about living landscapes, then I think about, in part, adaptive capacity and to what extent, by focusing on status rather than process, are we disallowing adaptive capacity in such a changing landscape, in such an influenced landscape, and a landscape that is predicted to change in the future. Anybody wants to add to some of this? Oh, yes, sorry.
So we talked a little bit about function versus species, and Sam was saying about, you know, the different rates, but something else that some tension that I also pick up in the literature is this, you know, where should management happen, at what scales? We very focus on on landscape scales, but as we saw from many of the talks, the effects that elephants have is, you know, happens at the local scale. So like, how do we balance the landscape versus local drivers, effects, whatever you want to call it?
Anybody'd like to respond to that?
I'm not responding specifically to that, but the conversation here brings me to the point of saying that we really do need to stress the need as professionals that the discussion about elephant effects on savanna ecosystems really has to be evidence-based. We really do need to be very careful about the way in which belief-based recommendations and statements gain currency and gain traction and gain prominence. And as scientists, we tend not to talk about our data if we don't have enough data to have the solid evidence. But we do need to feel confident that, I mean, I think Rob's presentation showed that there is now starting to become some evidence about density dependence in elephants. That's fantastic. And we don't have those kind of data from elsewhere. But in the absence of evidence-based information, we do tend to get inundated by belief-based statements, which really sway the agenda. And so I just urge that we be rigorous about demanding evidence-based information. And we also apply some rigor in thinking about what are the counterfactual situations. If we had a Kruger landscape in which the elephant densities were much lower than they are now, how would the vegetation look in the face of variations in rainfall and fire and other herbivores? And be rigorous about thinking, you know, are elephants in fact the major agent of change in the system? And if that rigor is applied, I think it will keep us clean.
Rob and Sam, you can fight it out about who's first.
I won. No, just credit where credit is due. We've been sort of inspired by the work that Simon did in Wangi quite a while ago, probably one of the first pieces of early evidence of density dependence, Wangi being an elephant population nearly double or even more than double the density that we experience in this part of the world. So just to clarify, even though we say it's early signs and one of the first sets of evidence, Simon did it first, just to stay in the clear.
Sam, we've run out of time, so if you can make it super fast.
I think Johan and Corley has captured very important ones. So I think Corley's point is we got to do local work at landscape scales, if I can summarize that way. How do I think about that? But Johan raised a very important point. Evidence is gold for us in this community. Evidence is not gold in society. In fact, they're challenging all the time. No, you've just picked evidence that works for you or anything like that. I mean, we've had that in the climate change debate and stuff like that. So I think there's a bigger thing than just evidence as well. That's important. But how do we do the science translation, what's sitting in this room, that is understandable and actually embeds within the expectations that society has got?
Thank you very much, everybody.
Thanks, Sam. You've alluded to what I mentioned in my opening talk about us as scientists needing to sort of just get the word out there and think of more innovative ways to get the message across. Thanks. Before I hand over to Dumi for the freshwater session, I would like us just to give a round of applause to all the session chairs, the facilitators, and the presenters. Thanks.
Thank you so much. It was a really good session. We'll be moving on to the freshwater session. We have some few talks, so we'll start with Andra. She'll be telling us about the Cerrado wetlands and how's the biodiversity doing there. Thank you.
Hi, my name is Andra. I'm a PhD candidate from Brazil. And so today I would like to talk to you about the situation we are passing through in the Savanna Cerrado and its threats to the biodiversity and to the ecosystem services. Well, so the Brazilian savanna that we call Cerrado covers about 20% of Brazil, and it is the wettest savanna in the world. And there we have many rivers which are perennial. And actually, the Cerrado is known in Brazil as the birthplace of water. And this is because the headwaters of many of the major rivers in Brazil are in the Cerrado. And those rivers are very important because many of them goes, for example, for this region, which is the Caatinga, the dry land in Brazil. So it's very important for the livelihoods, but also for supporting biodiversity, agriculture, and also for the hydroelectricity in Brazil. And these rivers born in a very special ecosystems that is the wetlands. So the wetlands in the Cerrado are very important to regulate these groundwater and surface water relations. And then they are able to store and to filter the rainfall and slowly release throughout the year. That's why the rivers in Brazil are perennial. Even the small stream, we have a regulate discharge along the year. And they are also very important because there we have a very distinct plant species and also support animals, mainly the migrate birds in Brazil that likes to stop in the wetlands and do the nests. But this water of the wetlands come from the groundwater. These wetlands are groundwater fed, and they depend of the patterns of rainfall. So we have here the groundwater that arise and goes deep depending of the patterns of rainfall. So we need the rainfall to recharge the groundwater. But the problem is the Cerrado is becoming hotter and drier. So we are having more temperature, a higher temperature, but also less rainfall than the normal. So thinking about that, we study how the groundwater dynamics structure the plants community in the wetlands. So we have 19 sites in the southeastern of Cerrado, where we measured 31 square meter plots, and we also have 9 wells to measure the water table. So in those plots, we identify and measure the cover of all species. So, well, we found out that in those plots that we have waterlogged conditions, we found less species than in those plots that we found it's also wet, but it's not waterlogged. Because in this wetlands patch, we have a natural gradient of water. So it causes a high turnover of species. So we found that in the sites where we have more oscillation of the groundwater, we have more turnover of species. So then we can understand how important is the oscillation of the groundwater to maintain the biodiversity. And because this oscillation is so important, we then study the water table dynamics. So we choose 9 sites to monitor every month the groundwater. And then we are seeing that some of those we can't ever measure anymore because the water is so deep and our wells can't reach the water. So in some of those, we see that the water oscillation is very regular, but in some of those, the hydrological pulse is falling. And it's better to see when we put all together and see the average. So here is in April 20, in June 23, we see that in June 24 and in June 25, we are not having the normal level of water table. Even when we have a large rainfall, this blue bar, it's not enough to recharge the groundwater anymore. And this falling in the recharge goes beyond, because it causes shift in the ecosystem. So these beautiful pictures here are also actually the bush encroachment that is going on. So we already know from some researchers that we made in the area that gallery forest and generalist species are able to colonize the wetlands when they are in the period of the dry. And it's caused a looping, because when we have an increment of the basal area, we have a deeper groundwater. And because we have these trees colonizing the wetlands, we also have smaller recharge. That's what, because I bring here one study from America and one from Africa to show you how trees intercept the water, and then the water can't reach the ground and can't feed the groundwater. So just to wrap up, I want to say how important the wetlands are for feeding, for water supply in the service of Cerrado. And the groundwater dynamics are key driver of wetlands biodiversity. And this seasonal water table are also very important to maintain the species turnover. But because these trends of drying in the Cerrado, these wetlands are becoming susceptible to the wood encroachment. And this is also a problem because in Brazil, we have many carbon-centered restoration strategies that compromise, because then people still planting trees, and we already know how this carbon storage makes the water yield decrease for us. That's why, thank you.
Thank you so much for the talk. We still have some time for questions. Question over there. Okay, just give you this.
That was a great talk. We've seen a lot of similar things in other ecosystems that are very complementary to what you just shown. I was wondering if by chance with your changes in water levels, if you're also getting changes in pH. Because a lot of times as the woody plants come in, they change soil respiration, which then acidifies the water table over time, which then has cascading consequences on the bedrock. And so I was just curious if with your wells, if you've been getting pH too.
I'm on? You mean pH of water? Oh, we are not studying the water, just soil. We are not analyzing the water. But I didn't know how the pH of water also changes with the wood encroachment. It's very interesting. Thank you.
Thanks, Andrea, for your presentation. Yeah, I am. I was wondering at the start of your monitoring, did you maybe come out of a very wet period, and then those dips that you had, I think it was 2023 or 2024, did that coincide with the drought? What was your rainfall doing over those years? Yeah.
So the rainfall, we have a seasonal rainfall in Brazil. So in the beginning of the dry season, we expect the higher water table level because we have all the rainfall. It wasn't. How long I'm studying?
Do we have time? Otherwise, I can talk to her after.
Okay, yeah, you'll have a discussion after.
Thank you.
Okay, cool. Thank you so much. Round of applause, please. Okay, our next speaker is Annelise. Thank you. And then she'll be telling us about the resource assessment of the Nkomati Basin and how the hills are moving around.
Good. Hi, everyone. I'm Annelies Erasmus, and I'm here to talk to you about our ungulate eels in the Nkomati catchment. So the ungulate eels we find in the Nkomati catchment all comes from the Western Indian Ocean side. So they are basically born right here in the plateau. They drift down as larvae and glass eels, go up into the freshwater sections, eat, mature, grow up, and then come back to their breeding grounds again. Within the Western Indian Ocean regions, we do find 4 species of ungulate eels. So this is just again showing you the life cycle. So they're born here, go down, grow big as yellow eels, and grow big in the riverine environment, and then go back as silver eels to breed. Now, by occupying both freshwater, estuarine, and marine environments, they're exposed to multiple threats throughout their life cycle, which is not only bad for them, but it's an advantage for us as it makes them great ecological indicators of these marine, estuarine, and oceanic environments throughout. So if we come to our focus area today, it's the Nkomati catchments. You guys all know it, basically majority of the Mpumalanga province and within Mozambique. So the Nkomati is quite special. It's home to various ecologically important fish species, and it has multiple fish protection and conservation areas throughout. Although it is being hammered by an overload of environmental and anthropogenic stresses throughout. For example, do you guys know what the eel said when it traveled through the Inkomati Basin? Damn! The Inkomati Basin is home to 12 major dams, more than 200 informal dams, and more than 83 gauging weirs. These are all hindering the migration upstream and downstream of these precious ungulate eels. And this is only one of the major threats that are affecting them within this basin. So, what are we doing about it? Nee, oom, ons is nie besig om die damme op te blaas nie. We are not building or breaking down any dams or weirs, but I like the way you guys were thinking. So what we did is we did a regional risk assessment. So basically what it does, it incorporates all of your environmental stresses or effects into a risk model for each of the different life stages. So we divided the life stages into 4 stages, and to determine which environmental driver is affecting the eels at which life cycle stage. This will then allow us to contribute or to determine a sustainable balance between water usage and the conservation of ungulate eels throughout the catchment. So what we did is we went along the Inkomati catchment. We had a total of 54 sites, which we evaluated the presence of eels or the absence of eels and other fish communities there as well, different depth and velocity measurements, various environmental impacts and stresses, as well as anthropogenic impact and stresses in these sites throughout. Here you can see where we've mostly collected our eels. So throughout the study area and study time of about 4 years, we've collected a total of 58 eels, mostly within the crocodile and Inkomati subcatchments. This came from 3 species as well, so Angula by color, we didn't document at all throughout the study time. And sadly, this is a 60% reduction from previously documented occurrences of ungulate eels within the same study region. So this would be the outcome from a risk assessment. I know it's big and bulky and you have lots of colors and you don't even know where to start. Neither did I, and sometimes even still don't. But basically what you have is you have your risk score. So this is basically an average of your risk percentages, where a low risk, indicated in blue, means a low probability of death, and a high risk, you think of, it's a high probability of death for these ungulate eel populations. And you can see the different life cycle stages as well as the different scenarios. I'm not going to present it to you in this way, so don't stress. But what are the different scenarios we've tested? We've tested 4 scenarios, or I'm presenting for you 4 scenarios today. Under the natural conditions, so this was basically our control for each of the different scenarios. So pre-anthropogenic conditions or much worse anthropogenic impacts. So under natural conditions, how did eels get affected by various things before humans had such a big impact? And the present scenario we have is what we are experiencing today, the impacts of today, the laws and regulations of today that they have to cope with. In our future 1 scenario, we have reduced connectivity, so essentially adding another brick to the wall. We're building more dams and building more weirs in order to supply for the water demand from people. And then future predicted scenario 2 was the lack of implementation of current laws and regulations if we do continue on a non-positive trend as if we can today. And this is a much easier way to represent this. So remember, green is your low risk score, so not as high probability of death. Red is your high probability of death or high risk. Now for firstly, illustrating it as if we don't so we don't take into the account of the different life stages, so just the area that is affected the most under which different scenario. Presently, we can see that it's only the Johannessi catchment, which is in a 0 to low risk. It's interesting because this is also one of the seasonal subcatchments within the Inkomati Basin. If we go further, our future one prediction, so less connectivity, it gets much worse within the Komati system, and the Komati system is already so impacted with barriers and weirs as well. So just adding more to the system will worsen the conditions for eels. And then if we do have lack of implementation, continuing on the trend we are continuing on, we are going to find ourselves in high risk positions within a majority of these catchments as we go on. And then now considering or focusing on not where it is occurring, but which life cycle stage is mostly affected throughout the different scenarios. So under natural condition, our yellow eel maintenance and our escapement stage had the highest risk, which is a low to moderate risk. And this is just because we have so limited knowledge of data actually about what occurred in our natural conditions or our pre-anthropogenic scenario. This is only in the riverine section that did have the highest risk, because they do still experience natural barriers like waterfalls and such in a natural condition. For our present scenario, our spawning and recruitment, so our marine section of the life cycle stage, had a low to moderate risk. This is also due to limited data of what happens to eels within the marine environment. So there's lots of data on what happens to eels such as angula, for example, in the marine environment, but we wanted to focus on just the available data we have from eels within the Western Indian Ocean. Our high risk is within our yellow eel maintenance and escapement. So when the eels are in the riverine system, they need to grow up and then they need to go back to the ocean environment. Now, these high risks are due to high number of barriers within our systems. In Mozambique, we found high amounts of illegal exportation and overusages of these eels. So they're exporting eels to different countries for food. And then limited habitat availability or accessibility due to habitat removal or destruction, as well as barriers preventing them from going up. So for our idea of identifying an area which we could focus conservation and which environmental driver to focus on, that plummet through the roof. Anything worsening, any condition worse than what we currently are expecting are just throwing the populations off the chart and they're putting them in a high risk. So even by introducing less connectivity, making more weirs or dams, is going to worsen the condition or the risk for ungulate eels, as well as a continuous lack of implementation and conservation strategies for our eels are worsening their condition. So for the Inkomati catchment, it's one of the nature's, our ungulate eels is one of the nature's most mysterious and mysterious migration stories that we have. Their journey is silenced by the environmental drivers and anthropogenic drivers that we are applying upon them or forcing them to live by. If we don't do anything, we're going to lose stories that was at least 1 million years in the making already. Thanks.
Thank you, Annelies. Unfortunately, we don't have time for questions. We'll move on to Angelica, who's going to talk about catch, mark, recapture, and manage of tigerfish. Thank you.
Thank you, Jimmy, for the introduction, and good afternoon, everyone. I want you all to imagine a river or dam that's so alive. Sorry, I must probably click. Okay, let me retry that. That's so alive that the top predator can tell us whether this river is thriving or declining. And in South Africa, that is our African tigerfish. Our African tigerfish requires more than science. It requires people, as local communities and anglers can actively contribute to the conservation of our biodiversity through citizen science. To tell you just a little bit more about this amazing species, so this is Hydrocynus vittatus, and it's a little bit smaller in southern Africa compared to its brothers and sisters up in Africa, but it's still one of our top predators in our freshwater systems. It is an indicator species of habitat change because it responds to environmental stresses such as altered flows, pollution, and migratory barriers. They are important from a socio-economic perspective as they are important in the recreational fishing industry, with anglers going around the globe to try and catch this special individual. And they're also important from a subsistence fishing perspective, as the local communities eat this fish and it provides food for the local communities. So despite their importance, we actually still have so limited information about their movement and population dynamics. And we need to understand where they're moving, are they moving, what habitats they are using, to be able to manage the system that they are in and the species itself. And in the last 50 years in Southern Africa, there has been a drastic decline in this species. The reason, altered flows and migratory barriers. This species is very sensitive to temperature. So during the high flows, they're able to cross, for example, a weir. But as the season settles down, the river flows reduces, they cannot get back and they get stuck behind this weir, the temperature declines and then increasing their mortality. If you think from an altered flow perspective, they use deep habitats to be able to thermoregulate by going to deeper areas or coming to the surface based on the environmental temperatures. But now with altered flows, this habitat is not provided for them anymore. A catch mark recapture is the ideal method to look at movement, growth, and population dynamics. But in Southern Africa, it has not had much success because the tigerfish do not retain the tags very well. But even a little bit of tag retention can provide us information that we did not have for the species. Which brings us to my study, a little bit away from Kruger, but still in a conservation area. So I looked at the movement and population dynamics of the tigerfish using catch mark recapture, catch per unit effort, and confiscated gill netting data from the SAPS South Africa Water Police, water wing unit in Lake Jazini. For the catch mark recapture study, we used both dart tags and pit tags to mark this fish. If I zoom into the lake, so the white dots indicate the different tigerfish, and on the tag, it's not very clear, but there was a website where local anglers was able to report if they actually got a recaptured individual. The reason we focused on the part of the dam is because we tagged with collaboration with Mafungana fly fishing, and that's where they operated. For the catch per unit effort data, we used again Mafungana's fly fishing's reported data. Unfortunately, they couldn't tag on all 4 boats, but they did record every tigerfish that they caught during the seasons. So the seasons, we did 6 seasons from 2022 to 2024 during autumn and spring, autumn moving into winter, spring moving into summer. And we marked a total of 188 individuals. Looking at the percentage catch over the standard length, the smallest individual was just over 500 grams, largest was 5.9, which is pretty big for our tigerfish in Southern Africa, and an average of 1.8 kilograms was the size of the tagged fish. So this is the part of my talk where I want you all to pick your favorite color, because luckily for us, we did get some recaptures, and then we can see where the fish moved to. You guys ready? Okay, so the dots indicate the marked location, and the picture of the tigerfish is where it moved to. And you can see that there's differences in home ranges, and I hope you all picked up on the increase of the pink fish, which we'll get to a bit later. So in total, we had 10 recaptures, which was a 5.3% recapture rate. If you pick the orange fish, you were both tagged for the shortest duration and for the you moved just over 100 meters. If you picked the yellowfish, you decided you want to live on the other side of the lake. Another interesting recapture was the greenfish. So we know that tigerfish breeds in the rivers with some information that they can use lakes to breed, and it moved a total of 10 kilometers upstream. So whether it spawned and whether it was successful is not really the point here. The point is they move outside of the reserve. So they get exposed to more gill netting and stresses that might have been accounted for during the reserve or inside the reserve. If we get to our nice little pink individual, so the first recapture was almost 600 days after it was first marked, and it only traveled about 1 kilometer. The second recapture from the first recapture was 440 days later, traveling a distance of 3.4 kilometers. And the third recapture was only 25 days later, moving about 300 meters. So what we think is happening here is that it might be opportunistic feeding behavior, as the tigerfish are following the boats, and after fishing, people discard their chicken hearts, or their livers, or even the sardines that we fish with. So maybe they can use that, or they eat those food. Similar to the Zambezi shark in the paper of McCord and Lambert in 2009 in the Breda estuary. If we look at the catch per unit effort data, this is the catch per unit effort on the y-axis during the duration of the study. You can see the cyclic cycling of autumn being in red being lower than spring, which could probably be the movement increase during the season, the temperature increase. But the problem that we have with the interpretation of this data is there's no independent data to understand the relationship between the population size and the catch per unit effort. And we need to understand that because this could be indicating a hyperstable relationship, which means that the catch per unit effort seems to be stable, but actually the population is declining. Because you're now catching the bold fish or the breeding population and not the shy fish that don't like hooks, I guess. Another big issue is the gill netting. So you can see, I don't know if it's very clear, but this is still a piece of filament of the gill net that is stuck to the fish. And here you can see the net scarring around the fish. The eye, I'm not certain if that was the gill net angler, but the point is gill netting is quite a problem in Lake Drazini. We looked at the water wings operation data from 2016 to 2025, and they removed 134 kilometers of gill nets. So just to put that in perspective, that's further than Satara Camp from where we're sitting at the moment. So if you're ever going to drive there, just think that's still gill net, and just the impact that it has on not just the tigerfish, but also their prey source. With the operations, they collected most of the nets from the bank. So we didn't really get an understanding of the impact it has on the fish species. But during one operation, there was 63 tigerfish recorded. And one of the operations for the tilapia, it was 500 and 151 during a second operation. That's quite a lot of fish. If you look at the gill net intensity over the years sampled, you can see that the SAPS police seem to have caused a decrease in the gill netting activity, which was great. Then COVID happened. We all know how we feel about that. And you can see that the gill net behavior even changed as they started to sink the gill net and wasn't floating on top anymore. The years went on, 2021, it started to become a full-blown netting activity in the river, with piles of decomposing catfish just being left on the bank. And years continue to move on, where the net has become so brazen that they are starting to process the fish next to the reserves or the camps, not even hiding it in the mountains anymore. So my questions remain is the population of the tigerfish in Lake Drazini declining? Does the low recapture rate indicate anything? Is the opportunistic feeding behavior for survival? Does the catch per unit effort show hyperstability? The increase in gill netting activity? And if we do lose this species, it's not just a conservation risk, it's an economic risk. This is also the southernmost distribution of the tigerfish in Southern Africa, and we will lose a lot of tourism if we do lose the species. But catch mark recapture is a good way to look at the movement, and it can be used. And citizen science is a great way to contribute to the conservation of the species. Thank you very much, and I would just like to congratulate and say thank you for the contributors.
Thank you, Angelica. We have one question. Jesse.
Thanks, Angelica. That was awesome. I have a couple of questions about the gillnet fishery. So, first of all, are the gillnet fishers using like multiple mesh sizes, or are they using either really big mesh sizes to catch your big tigerfish and catfish? And my second sort of question was, in places like Lake Kruger, the abundance of the tigerfish is really strongly related to the like prey diversity and prey catchment effort. Is there any information on that as well?
No, unfortunately not. And that's the biggest problem is we actually don't even know what the number of tigerfish in the lake is, what species are in the lake. Well, we know the species, but not the abundance. There's multiple studies that have tried, but there's always like the weather wasn't right, or this wasn't like, and even with us during the study, we tried to look at the community structure. And if we got 80 individuals during those years, it's a lot. And is that because the dam is deep or the fight nets didn't work? I'm not sure. I don't have any information regarding the type of gill nets they're using. Unfortunately, the reports is just like limited information of they confiscated 3 meters of gill net, they confiscated boats. So unfortunately not.
Okay, quick one.
Quickly. My question, Angelica, thank you so much for your presentation, is around the economic threat. You said when the tiger fishes might be gone for tourism. But I mean, I'm just thinking about the socioeconomic side behind it for people, right? So what would be the alternative so that they're still allowed to fish, but in a sustainable way? How do you get around that?
Yeah, so I don't think we need to stop the fisheries. That's not at all the point. I mean, the dam is there to be able to fish for the communities. I mean, there's 36 lodges around the dam that use it, that get paid. There's chartered boats that go on. It's just we need to find a balance between, because the gill netting is illegal, irrespective. So if it's a food thing, we must maybe try and rehabilitate the fisheries thing that is at the lake. But it doesn't seem to, like, you're leaving fish to rot at the side of the bank. So it's, I don't know. I don't know who's doing it. I don't know. I just had the SAPS reports. But yeah, any suggestions of how we can help to improve it? Because it is for the people at the end of the day.
Okay, thank you so much. You can continue the discussion after or during lunch. So we'll go to Gabby, and then we'll be evaluating the conservation corridor success for rare and common dragonflies using zeta diversity.
Sure, I think I need glasses. Hi, everyone. Thank you for being here. It's so nice to see such a turnout on the last day. Normally, everybody ducks out for a game drive. My name is Gabby, and I'm part of the maths department at Stellenbosch University. Originally, I'm a freshwater ecologist, and when I moved over to do my postdoc in the maths department, I decided to use some of my PhD data just to grasp the concept of zeta diversity. So this paper has been a really difficult one. It was just a small one, ecology and evolutions, and I had to do all of this before Sandra and her team developed the easy way to deal with zeta diversity. And it's taken over 1.5 years to get this, and on Saturday, I got published. So it was kind of like a sign that it had to be done. So when it's actually out, I'm going to send a plane with a thing behind it, and all of you are going to have to screenshot it and read it, please. So I'm going to be speaking about that paper and basically looking at zeta diversity on dragonfly diversity using in plantations in KZN. So plantation forestry in KwaZulu-Natal is mainly grasslands or naturally grasslands. And up to date, about 40% of the land area has been planted with monocultures consisting of pine plantations and gum trees. And it's not just about the change in the plants, it's also about the change in structure. So to go from grassland species to these really tall trees that suck up a lot of water, it's really detrimental to the environment. And these forestry plantations that we worked with have come up with a solution to try and deal with it, which is what my team has been working on for the past like 20 years to see how well these things are working. So the solution, that's called a conservation corridor. And these are basically areas of natural grasslands between these corridor plantations. And fortunately for me, so it's law that they're not allowed to plant trees closer than 30 meters to rivers. So as a freshwater ecologist, it makes a perfect area for me to work on these systems. So that's kind of a typical example of what it would look like. So the river runs through it and there's natural grassland. And the idea is that these natural grasslands connect to natural areas or pristine areas or reserves. So you're creating what they call an ecological network to try and help maintain and sustain biodiversity in all its forms. And to date, so it's been already looked at for dragonflies, butterflies, mostly insects, and also plants. But using Zeta diversity framework is kind of taking it to a next level. So the idea of these corridors is that they can either act as stepping stones for species to move between one natural habitat to another natural habitat and find resources along the way, or actually as actual habitats. And it's difficult to discern between those things, but it's one of the ways that we're looking at the variables that can best design these corridors to help maintain diversity of insects and also other species. So dragonflies have always been my passion, and I can speak about them all day, every day. So the reason why we use dragonflies, number one, is because in these corridors, these river systems, it's kind of like if these forestry areas have to preserve areas, why not make it around the river system, because they're not allowed to plant there anyways. And dragonflies are really taxonomically well studied, and they're also used as bioindicators of ecosystem health, but also as umbrella species. So by protecting the environment and the needs of these species, we can also protect the needs and environment of other species below them. And this has been shown for some really other specialized invertebrate species, as well as plants, wetland plants. And they're low cost and easy to sample. I mean, my PhD was an absolute breeze. Stand by the river with a glass of wine watching dragonflies. Like, what kind of better PhD do you want in life? And we only look at males, and they're highly territorial. And the benefit of that is that if you don't manage to catch them, they come back to the same spot if you can't ID them on spot. So I'm sure most of you here for Sandra's talk yesterday. So in terms of looking at their diversity, to date, even like my PhD work, looked at traditional biodiversity measures such as species richness, abundance, and pairwise measures. And these don't consider the impact of both rare and common species. So common species make up mass of the biomass, and we need to consider the needs of both in order to have an ecosystem that functions well. Oh, gosh, time. So the Zeta diversity framework integrates both rare and common species. So if you look at that, so first order of Zeta is just your basic total species richness, and then second order of Zeta is pairwise comparisons. Anything above that, you're leaning more and more towards common species. So if you look at any number of sites, so you have whatever number of sites that you have, if a number of species are shared between, if you have 100 sites, and the same species shared between 80 of those sites, that's a common species. But if you only find that species between 2 of any combinations of sites, it's considered a rare species. So that's what Zeta is trying to differentiate compared to just the pairwise comparisons. Oh gosh, I'm running out of time. So that's just a representation of it. So the higher the number of Zeta is, the more common the species are between sites. And this is not just, it's between any number of sites between your samples. So the objectives of the study were to look at, well, applying zeta diversity rather than just normal scale metrics, and look at what environmental variables were driving the rare and common species to see how we can design and manage these corridors the best way possible. Oh gosh, okay, fast. Okay, so we had 104 sites, 55 of them were the natural areas, and 49 of them were the corridors connecting those natural areas. And we wanted to see what was driving rare and common species in the natural versus the corridor areas, and whether these corridors were functioning well enough that they could be better or equivalent to natural areas surrounding them for biodiversity. And we only looked at adult males, 2 observers, 2 hours per site, 104 sites. So this is a representation of the sites. So the blue is the plantation, the corridor sites, and the green are the natural sites. And yes, they are quite lumped, but we did include spatial, yes. So the variables that we selected were based on previous studies that are known to be important for dragonflies in terms of river and corridor characteristics, because there have been a lot of studies done on them. And the distance that's on the side there, that is distance and space is included in the zeta diversity framework as part of the framework as is. So there were 37 species discovered. If you look at the bottom, there were more species in the corridors, so Anisoptera are your true dragonflies, Zygoptera are your sensitive damselflies, which in itself, these corridors are great, but shows you that you cannot just use species richness as a metric when considering whether things are being successful conservation measures or not. So in the corridors, so all of the other species were pretty much common species, but in the corridors, Chloralistes apricans was super exciting. It's extremely rare. It's Amatola malachite. Not sure how it got there, but it was a really exciting finding, and this was found in the actual corridor. So, and I went back there a couple of times and it was still there. So it wasn't just passing by, it was actually living there. Oh, 40 seconds. Whew. Okay. Species richness, there were 2 main drivers. So we looked at species richness and drivers of zeta. So in terms of species richness, land use, so there was higher species in the corridors than in the natural sites. And in terms of shade, percentage shade cover over these rivers, shade cover reduced species richness. Then we looked at effects on turnover using the zeta diversity metrics. So zeta 2, between 2 sites, we had distance, so for both, distance for common and rare species, distance was the most important predictor. So the further away sites are, the more different you expect the communities to be. And then water temperature, rock cover, and dissolved oxygen, which are all things that are known to influence, especially rare and more sensitive species. Oh, I'm done. Okay. And then common species, distance between sites was most important, water temperature and rock cover. And the take-home message is that these corridors are working, in fact, better than we think. The most important point is, out of those variables that we listed, land use, so whether it was corridor or natural, did not come out as significant in terms of turnover, and neither did alien invasive species. All of the other variables, which were water-related, came out as important, which to me means these corridors are functioning the way that they should be. And the zeta diversity concept came out as really successful in the way that it showed things, and should maybe be considered to be applied in other contexts. And there was one other point. Oh, corridors are great and can enhance diversity. Sorry.
Okay. One question. One burning question, if you have. Okay, so, James.
Just observationally, did you see anything other than dragonflies benefiting from these corridors? From an observational perspective, did you see anything in addition to dragonflies also benefiting from these corridors?
So the group that we've been working with has done a lot of work, 20 years of work of these corridors. And actually our recent master's student has used these 20 years of data, but it's mostly invertebrates. So butterflies, dragonflies, beetles, plants, and they're all kind of showing the same thing. But the main thing that comes out is actually the width of the corridors. So the wider the corridor, the better, and that's the main thing that's coming out for all of those. Yeah.
Thank you, Gabi. And then we'll be going to Stefan. We'll be talking about thermal vulnerability of Nile crocodile in the Oliphant's Gorge, Kruger National Park.
Right, this study of crocodile thermoregulation was initiated following the mass mortality event that took place in the Oliphants River Gorge. When those crocodiles died, this is kind of like what they looked like. And the Sandparks veterinarians discovered that these crocodiles had died from a disease called pansteatitis. Now, pansteatitis is a dietary disease, and we figured if we could work out what the crocodiles were eating, we would solve the problem. Now, this slide sums up what took us 10 years to work out. The mechanism behind the diet at that stage was the raising of Massingier Dam Wall, which backflooded into the Oliphants River Gorge. So this is the Oliphants River Gorge, and it used to be a river, and now with the raised lake level, it's part of a lake. Our pollution flows in there. It produces these massive algal blooms. The algal blooms concentrate filter-feeding fish, crocodiles eat. That was the mechanism and we're pretty sure that that's what was going on. The problem is that these algal blooms have become an annual event, but the crocodile deaths haven't been an annual event. And so there had to be something else going on. And I must say that the Sandparks vets were erudite, man. They thought that there may be a metabolic issue here as well, and that would be regulated by temperature. And so they deployed a set of sensors to measure crocodile temperature variability, and that's what I'm going to be talking about. So how do you measure the temperature of a crocodile? Well, the first thing is you ask it to step out the river, and you ask it to just lie still while you just anesthetize it a bit and apply a sensor onto the nuchal scoot. And I don't know if you can see it in this picture, but there's a little green sensor that we would attach. Now, that sensor is a GPS tracker. It's got an accelerometer in it, and it's also got a temperature sensor. But we also needed to understand what was happening to the core temperature of the crocodiles. And so again, you ask the crocodile to step out the river, and if you ask it to open its mouth, it will. And you can open up the gular flap and you can drop a temperature sensor into its stomach. But we mitigated risk in case it got spiteful and closed its mouth while you were doing it, so we used this pipe. But it meant that we could get these gut temperature sensors into their stomachs. So I'm going to start off just, here's the data coverage that we got. So we did most of the tagging in this area, but you can see the crocodiles went all the way up the Letaba, not so far up the Oliphants, and all the way down the Oliphants Gorge to Lake Massingier. At the bottom, there's the time series of the data sets, and I've got there in blue modeled air temperature. In black is the temperature sensors from the back of the head. About 4 years of coverage there, but unfortunately the stomach temperature sensors, the coverage wasn't so good. That's plotted in red, and then orange is the accelerometers. I'm going to start off with the analysis of the temperature sensors on the back of the head. And I'm plotting here on the x-axis the air temperature, and on the y-axis the temperature that we measured. And I'm separating out summer and winter. And I've plotted the 1-to-1 line through the center of each of these plots. Now on the left is the full data set, and it looks like a 2-year-old got hold of crayons and decorated the wall. But actually there's patterning in there, and if you resolve it by hour of the day, that's what I've done on the right-hand side, then you start seeing that there's a daily cycle in the temperature of that sensor. And I want to start off over here. So in winter, that sensor starts off coolest early in the morning, but by about 8 o'clock, it's 9 o'clock, it's starting to warm up. By 10 o'clock, that sensor on the back of the head is warmer than the air. And it remains warmer than the air all the way up to midday, all the way down to about 4 o'clock in the afternoon. Now, for that sensor to be warmer than the air, it means that the crocodile is lying in the sun. And so that's an indicator of basking behavior. If we look at what happens in summer, about 8 o'clock in the morning, the temperature drops below air temperature. And it stays below air temperature for most of the day. And that's telling us that the crocodiles are probably mitigating overheating by getting into the water. So that external sensor is actually telling us basking or not basking. Let's look at the core temperature, and I think that this is absolutely remarkable. I don't need to describe what's happening on the left, but on the right here, in winter, the crocodiles have their minimum temperature, core temperature before sunrise. We have the day warming up, so we're moving from left to right, but the crocodile doesn't warm up. So the day is getting warmer, crocodile stays cool. Then the crocodile starts to warm up, but the day isn't getting much warmer, and so the cycle goes on. By the time we get to the evening, the crocodile, all these points are lying above that 1-to-1 line. The crocodile is warmer than the air temperature by about 5 degrees. That's a really good thing to do in winter. In summer, we have a similar kind of cycle. We know that the basking from the previous month was affecting this. They're not basking in summer. We see that in the day they're probably going into the water to mitigate overheating. The question is, how do they do that? How can you have, be 5 degrees warmer than the air temperature through the night? Now, to understand this, I need to show you the data on a daily cycle. So what I've got plotted here is for summer, the air temperature in blue, and this is the core temperature in red. And what you can see is that the minimum air temperature precedes the minimum core temperature by a few hours. The maximum air temperature precedes the maximum core temperature by a few hours. And that's something that's been noted in saltwater crocodiles in Australia. And it's got to do with straightforward thermodynamics. If you took a brick and you heated it from the outside and you measured the core temperature, the core would heat up slower than the outside and vice versa. But that lag between core temperature and external temperature reduces the amplitude so that the crocodiles never reach the temperature of the air during summer. Now look at the lag effect. Let me just point out this. This is the activity, this is the accelerometer. And you see that the peak in activity of these crocodiles coincides with the lowest temperature. So they are avoiding generating heat and being active when they are at their coolest. Looking at winter, day temperature, crocodile temperature. But what's amazing is you have this overshoot where the crocodile's core temperature is elevated above the air temperature. And that's got to do with the basking behavior. So they are able to generate sufficient heat in their core so that as they cool overnight, they don't reach that critical, they don't get that cold. So that's how they manage this 4 degree or 5 degree heating through winter. Now, what's the consequence if you don't bask? And we had one crocodile that did some odd stuff. On the right are the traces for crocodiles that were basking. Blue is air temperature, black is basking, and these big peaks here says, yes, they're basking. Red is the core temperature, and you can see these crocodiles that are basking, they're heating up and cooling down, as we would expect. But crocodile number 4, in the black trace, we don't see those peaks that indicate that it's basking. And what do we see as a consequence? It's core temperature. Can you still hear me? Okay. The core temperature drops, and instead of recovering each day, it drops and it drops and drops. So it's really important for these crocodiles, if they want to be able to defend their temperature through the night is to generate this heat during the day. This is what happens if you don't. Now, what's interesting about this crocodile was its GPS position didn't change. So the crocodile was staying in one place, but here's its activity plot down at the bottom. It was more active than the other crocodiles, even though the other crocodiles, okay, they were basking. So it was doing something, but it was doing it in one place. And when I looked at the GPS location, there it was, right off of a fishing village in Lake Masingia at that time. So it had probably been raiding nets. Maybe it got caught, whatever the story is, it's not really important, but it was stuck there for 5 days and couldn't bask. I'm going to wrap this up just by pointing out two things that come out of the study. The first is that lag time that we picked up. It's got to do with the body conditions. So this is the plot of how the lag time varies through the year. You start with long lags during summer, the lags get shorter and shorter, and that coincides with the crocodiles losing body condition through winter. As soon as they start feeding again and they pick up body condition, the lag time goes. So that tells us that the fat that they depend on through winter is acting as an insulator. It all fits into the thermodynamics. So this side of this plot is the thermodynamics of crocodiles being fat or thin. On the right, I've plotted what we call the thermal efficiency. This is for any degree change in air temperature, how much of a change do you get in the crocodile's core temperature? And you see high values up here during winter when it's coolest, and that's because of the basking. So the crocs are trying to really push up their body heat as much as they can. Over here in summer, when they're trying to avoid overheating, their thermal transfer efficiency is lowest, and that's because they're spending time in the water. And so to wrap up, we picked up these two things. There's the thermodynamics, that's the insulating effect of the fat, and then the behavioral side of thermoregulation, which comes from shuttling in and out of the water and from basking. And how does this all reflect back on the pansteatitis outbreak? Well, there's again a picture of the gorge. And prior to the backflooding, there would have been a river here with lots of basking space. When the backflooding happened, suddenly you had a lot of fish for the crocodiles to eat, nowhere for them to bask. And so through that winter, you would have seen the crocodiles not being able to metabolize the fish that they ate, and that led to the disease. Thank you.
Thank you so much. Thank you so much. It was a very lovely session. Unfortunately, we don't have time for questions. I think that's the end. We can go for lunch. Thank you.
Good afternoon, colleagues. We are about to start. We are almost 5 minutes into our program, so we'll try to catch up. I know it's a difficult one after lunch. So I will be monitoring those 5 minutes power naps. And if we catch you, I'm gonna give you a mic to summarize the session, okay? So we have 3 presentations in this session under the theme Looking Beyond the Park Fence. One looking on the patterns of soil organic carbon, the other one on climate change mitigation, and the last one will be on woody vegetation distribution. So first in this session is Robin. Over to you, Robin.
You put it on. Cool. Hi, hello everyone. Thanks for braving the post-lunch slump. I know it's hard. Hope I don't bore you too much with some carbon. Tell you an interesting story in the Eastern Cape. I'd first like to start by acknowledging my co-authors on this work. It is part of a collaborative and a small part of what we are busy investigating. So climate mitigation strategies are on the top of a lot of land use priorities, with carbon as a hot topic. And as an approach, there's currently a global movement to rewild a lot of grassy rangelands as a nature-based solution, or should I say nature-based opportunity, to carbon sequestration gains. And one of the frameworks around this is suggested as the animation of the carbon cycle. And this considers how herbivores may mediate carbon flows beyond their direct biomass contribution. However, a lot of this ACC literature comes from systems that have previously had grazer collapse or have had grazers removed from the system. But Southern African systems have long pastoral histories, and with that come pastoral practice such as prescribed fire. So these systems are predominantly not grazer-absent baselines. So while herbivory effects on your total carbon are well documented, the effect on differing carbon pools is not. And global standards on measurement, reporting, and verification guidelines often report or record total carbon as an absolute pool to assess baselines and sequestration potential. But soil organic carbon differs in its mechanisms, how it drives their transfer from one pool to the next, and how it moves from one state of lability to a state of persistence. And you heard a lot more about that at Mariska's talk yesterday. So here we are interested in 3 pools: your unprotected free particulate organic carbon, or your FPOC, and this is largely made up of detritus or vegetative litter or plant litter with a very low persistence. We're also interested in your occluded particulate organic carbon and or OPOC, and this is physically protected in the aggregates in the soil with a medium persistence. And we are also interested in your stable state mineral-associated organic carbon pools, or MOAC, which is chemically protected and can persist for decades. So understanding the disconnect, or the possible disconnect and gaps between carbon and the ACC scenarios in the Southern African grasslands, we wanted to know, does the short-term attribute of land use management, rather than the long-term attribute of the landscape's inherent abiotic memory, create a divergence in stable carbon pools? So this took us to Eastern Cape. To look at this, we used data from 2 studies in the Sneeuberg region of the Eastern Cape, and this was located between the Kruger National Park and the Mountain Zebra National Park, with the yellow dots indicating where the paired fence line experimental sites were, and the blue star indicating the location of the exposure sites. And this landscape is relatively high altitude, it's semi-arid conditions with climatic variability and historical periods of drought and rainfall pulses. The underlying geology is also limiting with a very low clay content. So the first study, we were given a unique fence line scenario to look at, changes in grazing regime. And there's a long history of pastoral land use in this area, with switches from some land from livestock land use to wildlife roughly 45 years ago. So we identified 26 fence line contrasts and sampled them with existing livestock land use on the one side and rewilded land use on the other. We also collated a lot of geological data and long-term climatic data over 50 years to see if differences would rather vary along an abiotic gradient. Here, we looked at the differences, the delta differences in carbon pools, where values above 0 indicated wildlife grazing, values below 0 indicated livestock grazing. And we found that management, or rather the type of grazing regime, did not matter in any of the pools in the semi-arid region. But memory does. So when considering the abiotic factors on carbon pools, we found that the long-term memory overrides the grazer type as the primary stabilizing mechanism independent of grazing. So interactions between the grazing factors and abiotic were non-significant, but looking at them independently, we saw at higher altitudes and greater precipitation, there was a greater buildup of your labile free POC and your more stable mark. We also saw at higher temperatures, there was sites at higher temperatures rather, there was a reduction in your stable carbon, and as predicted, we saw greater amounts of free POC in your top 5 centimeters of soil and greater amounts of occluded particulate organic carbon in your soils below 5 centimeters to a maximum of 20. So in the second study, we wanted to see if herbivory mattered. So with simulating rewilding or wilding, follow the ACC framework in this context. So here there were 2 sites that were identified in the Mountain Zebra National Park. The Roy plot, which was set up in 1998 by Hugo Besadenhout, where 8 matched exclosures were used, 4 that were burnt under high intensity and 4 that were burnt under low intensity. The second exclosure site, also in Mountain Zebra, was Odam, which was set up 10 years previously to Roy plot. And these were 4 exclosures that were used and where fire was excluded. So composite soils were collected in and out of these exclosures and physically fractionated and analyzed for your solid state carbon pools using the Dumas method. So again, we looked at the delta differences in high persisting mineral associated pools and other pools as well. So values above 0 indicated wildlife grazing, and values below 0 indicated grazing exclusion, with yellow indicating low intensity, orange is high, and your green was your Odam site that was 10 years older with the exclusion of prescribed burns. So we found, interestingly, we found stable mineral-associated pools were greater in ungrazed sites at Roy plot, but the differences did not change with depth or with fire intensity. We also found that no effect of grazing exclusion or depth on your mineral-associated pool was seen at Odam. However, when considering your less protected particulate organic carbon, in this case your OPOC, there was no differences in your OPOC between grazed and ungrazed sites at either depth or fire interactions. But the exclusion of fire rather at your Odam site, fire treatment and depth effects were associated with greater labile occluded particulate organic carbon, particularly in the top 5 centimeters of soil. And this was similar trends were seen with your free POC, it's also a very unprotected pool, and where in the burn treatments at Roy plot we observed no significant grazing effects and no fire intensity or interaction with those. However, in the unburnt Odam site, depth and grazing effects and the interaction were significant. And this effect was notable in soils below 5 centimeters, where wildlife grazing is associated with greater labile carbon. So putting these together, these 2 studies, I mean, they told us an interesting story. So the fence line study told us that there was no overall effective grazing regime on the stability of carbon in these systems, whether it be wildlife or livestock land use. Rather, climatic variables exert a stronger control over the accumulation of protected and unprotected carbon pools. And the grazing exposure told us that short-term land use attributes affect the stability of your carbon pools in these systems in variable ways. So herbivory reduced your stable state mineral-associated pools, but increased your more vulnerable particulate organic pools. So, in conclusion, in these semi-arid systems with a long pastoral history and practice, these practices may not conform neatly to your ACC predictions, but rather organic carbon pools appear strongly constrained by your climatic and geological and legacy or memory effects of this landscape. And so I think that carbon-focused framework should therefore be context-dependent rather than universally transferable. And I'd just like to thank you. These are some awesome people on our team. Loved working with them. And I hope there's time for questions.
Thank you, Robin, for the great presentation. We have time to take questions, maybe one or two. Any questions? Is it after lunch? Oh.
That's okay. Thanks, Robin. I might have missed it, but with your first study where you were sampling across the rainfall and temperature gradient, how extensive were they? What was the gradient? Well, what were the temperatures? What was that precipitation?
Sorry, can you repeat the question?
So you were sampling across a precipitation and a temperature gradient, but it wasn't clear to me what the temperatures were or what the rainfall was across that gradient.
That areas, I mean, it was quite a wide gradient, so I mean, it varied between 4 and 500 moles on average, and then, I mean, the temperatures also were very variable in terms of season. So it was, I think you got lots of frost days in your winter, but it went up to about mid-20s. So it's more, it's not too hot. It's not like Kruger, I think, in the middle of February, but yeah.
There was another question.
Okay, quickly before it dies. I was just wondering, I'm so sorry if you said this, how do the densities compare between the grazers that are wild versus pastoral? And what about the movement? Or is it very concentrated for one and dispersed for the other? Or how do those compare?
Yeah, I mean, obviously there's differences in movement between your livestock, how your livestock move and how your wildlife move. But I think a lot of the farmers in the area say they practice holistic or rotational grazing, so there is a lot of variability in how the cattle are organized or move through that area. But what a farmer tells you and what he does can sometimes be very different. But yeah, I think, and then there's also sort of a congregation of how cattle move in comparison to wildlife congregating on like fence lines and that sort of thing. But there was a lot of variation in that fence line site on cattle practices and cattle movement. Yeah. I hope that answered your question.
So, Robin,
yeah.
Hi. In the workshop last week, we were talking a little bit about the importance of trees. Now, when I look at your photo, it looks very grassy, and it also looks very similar in terms of cover. So I was just wondering, like, maybe you wouldn't expect to find these differences unless you have something that's becoming really invaded by Seriphium or, one of those invasive shrubs. What are your thoughts?
So your question was, what if it was invaded by more shrub? Yeah.
And you're losing, like, I think you have to lose some of the grass cover before you're going to see these effects.
I think it would obviously alter how your herbivores are feeding on that and how they would deposition on those grasslands. So certainly it would change. But I mean, a lot of the farmers burn in that area, so I don't know if that would also alter your plant cover, your vegetative matter there. So yeah, it would certainly probably affect on how those herbivores are digesting firstly and taking up and then depositing back onto the land. So yeah, I think so.
Thanks, Robin. I think if
Thank
there
you.
are still discussions, you can take it up with Collie. Round of applause for Robin. Next is Jobin.
Yeah, just checking. Hello, everyone. Good afternoon. I'm Jobin Varghese from National Center for Biological Sciences, India. And I'm going to take you from Africa, out of Africa, to all the way east in India. And I'm going to talk about the efficiency of tree planting on grasslands and whether they help in combating climate change or not. Yeah, so as an introduction, tree planting has emerged as one of the most sought after climate solutions across the world. And the sad part is that they usually target open ecosystems, particularly the natural grasslands. Now, several earlier studies have talked about or highlighted the impacts of grassland afforestation. And they talk about the impacts on ecosystem services, particularly the hydrological changes, then changes in biodiversity or impacts on biodiversity, grazing lands, and grazing livelihoods. But there are very few proper evaluations on net carbon benefits from afforestation, and this is particularly lacking in the tropics. So there are temperate studies which talk about effects of grassland afforestation, and there's a Jackson study that talks about how, so there are examples of how there are changes in the above ground biomass, above ground carbon, below ground carbon, but they do not necessarily, the above ground carbon gains do not necessarily offset the total ecosystem carbon gain. And like the Jackson study says that there are, but there are few sites that he found where the offsets actually, the ecosystem carbon gain actually goes down to below zero. And so these losses may not be sufficient to offset above ground gains, leading to a net ecosystem loss, a carbon loss. So the broader objective of my study was to look at the effect of afforestation on carbon pools, above ground, below ground, and ecosystem. How does it affect the labile and persistent carbon, what Robin had just talked about, and how do these effects vary across environmental gradients? We looked across our country, across the grasslands of our country. We sampled over 66 sites, covering a range of ecosystems from trans-Himalayan, Himalayan, hot deserts, then Deccan savannas, then even some flooded grasslands in the west. For sampling, we sought after a paired site, one of the plantation, one of the grassland, and one nearby plantation where the trees have been planted on the grasslands. What we did was measure GBH, and using allometric equations, we calculated the above ground biomass and then carbon. We also, within the subplots, we also measured the grassy and the faux biomass and added that to the AGB. And we took soil cores way down till 50 centimeter, and we divided the soil into labile and persistent based on the size differentiation of the size. Basically, the sieve size was 53 microns. And the results. So what we, okay, so to understand the results, what we have to understand is anything below 0 over here would mean that the plantations have more carbon than the grasslands, and opposite. The grasslands have more carbon than plantations, and then anything above 0 would mean plantations have more carbon than the grasslands. So obviously, when we look at the above ground carbon, we have an increased, the obvious increase in the plantations for the carbon, but the effect or the carbon does not show the same trend when we're looking at below ground carbon, where there are a few sites which have more carbon in the grasslands than the plantations. So when we combine the above ground carbon and the below ground carbon and look at the ecosystem carbon, what we see is there are quite a few sites which have very minimal addition to the total carbon. And there's at least one case that we found that has a negative ecosystem carbon due to afforestation. Similarly, when we look at the persistent and the labile soil carbon pools, what we see are that there are quite a few sites where the grasslands have more carbon than the plantations, and for persistent carbon, similarly for labile carbon. So there is no consistent difference in the responses of labile and persistent soil carbon pool. And we also looked at the total soil, the log response ratio with certain environmental gradients. So what we get over here is, again, here the grasslands have more carbon, here the plantations have more carbon. What we see over here is with the precipitation, the wetter sites have more, the wetter sites have more carbon. The colder sites have, sorry, I forgot, wait. Yeah, colder sites show greater loss of soil carbon pools. And with the plantation age, the older the plantation, more carbon gains they get. So synthesizing all the graphs that we had today, what we get is basically tree planting in grassland can lead to a loss of soil carbon in many instances. In some cases, these losses can offset above ground carbon gains. Both the labile and the persistent pools, carbon pools, are vulnerable to loss because of afforestation. We till now haven't found any clear relationship of responses with environmental variables, and thus we require a more widespread sampling effort to pass out the causes underlying these responses, particularly if we are looking also at the species identity of the overstory overstory trees, and also the interaction between the environmental variables. At the end, I would like to thank all the funders and the team who were on the field as well as the lab. And yeah, thank you.
Thank you. Thank you very much, Robin. Do we have questions? We can take two. Collie?
I just want to point out that I didn't have my hand up, so now I have to quickly think of one. The plantations, are they all exotic trees, or did they also, a forest with some of the, if I can call it endemics, indigenous species, and do you think that would make a difference?
Yes, they are. At least most of them are exotic, non-native trees that we had, from acacia to prosopis. We have a lot of exotic tree plantations in India. Yes, they should have an effect, and that's something we will try to analyze in the future.
Thank you so much. Yeah, I have a question. Have you presented these data to the forestry department in India? And if so, what do they say?
I'm scared of doing that. No, seriously. No, we haven't yet, obviously, and yeah, this work is still in progress, and I'm really scared of doing that. And so we are pitching up. We are trying to figure out a better story to say and publish before before we present. Yeah. Right.
Thank you very much. Another round of applause. Oh, there's a question there at the back. So
Okay.
we'll take the last one.
Okay, thank you. Mine is still a point to ponder. We have a 30-year restoration project back home. These were like purely grasslands, and 7,000 kilometers have been planted with indigenous trees. Yes, we are selling the carbon, but to me, what actually drives this restoration is mainly the financial gain and not the ecological importance of a restoration. And where I come from, for those who have been there in this room, it's a mosaic. It's 759, but it has patches of savanna. Guess what? Because there's business, the carbon cell bit, they want to plant all the savanna patches. We have savanna, we have antelopes, we have buffaloes, elephants themselves, because we have a mixture of savanna and forest elephants are using these open grasslands. I received so many calls of people proposing to plant these open grasslands. I throw it on the table for us. I know when it doesn't work in my area, they'll sell it out to the different countries. It seems to be a lucrative business in the recent times. Let's kindly all together say no to this. Because I mean, we've just had the consequences of letting people plant the savannas. I mean, if we allow it, if we'll set the pace, and I mean, before we know it, we know what happens. Thank you.
Thank you.
Putting us on a very serious and awkward position there. So I think we can take this discussion outside. Next is Nesisa.
Hello, everyone. Once again, my name is Lisa, if you don't know from yesterday. And today I will talk to you about woody vegetation diversity, distribution and assessment that we attempted to do in the Greater Limpopo Transfrontier Conservation Area. And I know yesterday I said we should use drones, so just bear with me. This might be slightly different. So yeah, I'll just jump straight in. Yesterday I didn't have time, so I'll make some time. As we all know, hopefully that woody vegetation plays a critical role in ecological research, and particularly in investigating plant species abundance, richness, their diversity, variance, that is important and very effective for conservation strategies. And understanding the species structure or the trees themselves is essential for elucidating the roles that these trees play in ecosystem functioning for different environmental contexts. And these key morphological parameters, such as the height, the crown size, the diameter, breast height, serves as allometric variables that indicate the health and the vigor of individual trees, aiding to effective, again, management and conservation strategies. So for this work, we aimed to assess woody vegetation distribution in the Greater Limpopo Transfrontier Conservation Area, and to examine the species diversity, its abundance, the characteristics it has in relation to topographical factors, to support a field-based habitat quality assessment. And we also sought to determine whether these protected areas are effectively in a good state or in a transitioning zone in terms of habitat degradation. So, as I said, the focused area of the Greater Limpopo is an estimated 29,000 square kilometers, which is quite big, and this includes the nearby communities that are in the buffer zone. And this area is dominated by acacia, riverine vegetation, low bushveld, mountain bushveld, and mixed woodlands. And the villagers, the people that live within this area, survive mostly on provisioning and cultural ecosystem services. So we used phytosociological classification to collect this data, which includes saplings, juveniles, shrubs, young trees, and mature trees. And we only focused on trees that are 4 meters and above in height, because we had the hypothesis that these are good indicators of a habitat which has been intact over a long period of time in a semi-arid region like the Greater Limpopo. And we had data from 2011 to 2024. And I'd also like to acknowledge Dr. Robert Carnivore, who also aided and gave us some data from the Kruger National Park, and also Mr. Bob Mandingenge, who gave us some of the data. And for the damage assessment for the trees that were damaged, we looked at trees that had some, I know it's a very sensitive topic, so elephants pushing it, scraping it, and lightning and all of that, so you get the picture. So to assess the use of our data, because a plotless data collection method was used, we checked the distribution of the data set using the analysis of variance, and this supported the PCQT sampling method for us to be able to go ahead and analyze the data that we used. And so in total, we sampled 266 sites from the northern part of Gonorrhoea to the southern part of Kruger National Park. And in total, we had about 2,400 individual trees that we sampled, including about 1,400 trees that were damaged, which is more than 50% of the data set that we had. And our results showed that our trees are clustered. The distribution of the trees are clustered. Mostly the trees that are big, coming big as in height, occurring on the southern side of the Kruger National Park. We also observed that most of the species within these sites or transects were occurring in the south because of the abundance of the water sources that are here. And most in between, the areas are dominated by shrubs or stunted woody vegetation. And within the descriptive statistics here, we saw that most trees have a moderate girth, so they are quite thick, with some trees showing a variation in sizes. Some are big and some are tall, and I think we had a bit of some outliers where some trees were growing up until 40 meters. And the results also showed that some trees are sparsely distributed and are sparsely covered with a mean crown cover of 21%. And these data were collected, of course, in the winter season for feasibility. And within our vegetation structure, we saw the relationship between the diameter breast height and the height of the trees, and we got a strong positive correlation, meaning that our observation showed that as the tree goes taller, it also grows bigger. I almost said fatter, but okay. And when it comes to tree height, we also noted that the diameter breast height was the only significant predictor of tree height, while other topographical factors like slope or elevation did not really significantly impact the tree height. So this affirmed for us that larger trees tend to be taller, but it's not because the tree is on an elevated slope that it is healthier or taller or something like that. I'm sorry, this is not so visible. It was on my slides, but this is a polygenetic dendrogram that shows how the trees are clustered. And here we saw that according to the Euclidean distance, the trees are clustered according to their height. So smaller trees and the shrubs tend to be on their own, and larger trees tend to be on their own regardless of the species. But there are some species that tend to be grouped together in different places. And in terms of the diversity and the dominance, we saw that as the dominance index increases for the entire region, the Shannon diversity decreases. And this suggested that where the few species dominate, the overall diversity is lower. So for example, in the Mopani woodlands, we got the diversity of the species there was low. Here we saw that the weak positive correlation of the dominance and the evenness indicated that a few dominant species are evenly distributed rather than one species completely outcompeting other species. And there are no, again, any significant topographical effects for this, whether it's slope terrain or, that influence this diversity or dominance. And with the above ground biomass loss, for the trees that we measured that were damaged, I will just say the top three because of time, trees that were contributing to the overall above ground biomass loss, which was the Mopani, the Combretum imberbe, and the apple leaf tree. Those were the trees that were targeted mostly by elephants. At least that's what we think. So what does this all mean? So in certain areas of the Greater Limpopo Transfrontier Conservation Area, it is characterized by high vegetation density and often marked by sharp peaks of trees within specific locations, and that these results corroborate with literature where a substantial concentration of a certain species is located in the southern part of the Kruger National Park, which is located near major water sources and which is critical for woody vegetation survival and growth. And the results in our study showed as well that the majority of these trees are large, characterized by tall heights and thick girths. And interestingly, while most species tend to populate lower elevations, our study did not reveal any correlations, any strong correlations between altitude and species abundance. So this is what we concluded. I stand corrected. So we thought that to maintain biodiversity in terms of woody vegetation in this protected area or in this region, management interventions are necessary in sites of high dominance and low diversity to promote species richness. And for the woody vegetation that experiences a lot of damage, where there's elephant or large herbivore damage or disturbances, these need to be the areas that must be looked at, which is a current management conundrum. Do we look at the places that are pristine and we conserve that, or do we find ways in which we can revitalize the patches or the areas that are damaged? And as we observed in our study, the variables closely associated to the disruption of these trees on higher altitudes, our water sources, the animal disruptions, and the lack of woody vegetation regeneration over time. And that our findings hopefully provide valuable insights for targeted management for people here in the Kruger or at Gonarejo, for the interventions for regeneration efforts that have to be evidence-based. And with that, I would like to thank everyone who was involved in our study from the data collection. It was 4 years of a lot of hard work in the sun, which is why yesterday we advocated for drones, but that's an ongoing conversation. And yeah, thank you.
Thank you very much. Unfortunately, we don't have time to take questions, but yeah, we can do so during the breaks. A round of applause for all the speakers in this session. And I will hand over to Dr. Lifuno for the next session. Thank you.
Is this thing on?
It is. It is yours.
Afternoon, everyone. We're going to a different theme now. It's the drivers of vegetation heterogeneity. Peter, you can. Oh, nice.
Some of you may remember 20 years ago at the fourth meeting of the Savanna Network, I presented some ideas of a project that we were about to do at the Nkushli exclosures, looking at chemical defenses of woody plants. I'd already been interested in and doing some experiments on woody plant chemical defenses for 10 years before that. What I want to put to you now, and I'm going to stick my neck out a bit, I think, and I want to put to you 3 questions that I feel have been neglected over the years, and which I think if we could pay more attention to addressing these 3 questions, we might be able to get a much more complete understanding of interactions between woody plants and browsers in African savannas. How do the woody plants manage to survive and reproduce under browsing pressure in these savannas? What I'm going to do to start with is I'm going to give you a brief history of the development of 3 important concepts that I'm going to talk about. These concepts are resistance, defense, and tolerance in terms of the plant-herbivore interaction. In 1792, Havens presented his observations of the Hessian fly. It had recently become a major agricultural pest. He presented these observations to a meeting of the Agricultural Society of New York. It was a major pest for grain crops that were being grown in the area. What happens is that the Hessian fly larvae attack the stem of the wheat plant and disrupt the flow of sap, and therefore reduce the yield of grain. He had noted in the years preceding that particular meeting that grain farmers in the area had been switching over from bald varieties of wheat to bearded varieties of wheat. And he attributed this to superior resistance of the bearded varieties to the Hessian fly. He put this down to a particular trait of the bearded varieties, in that they have a much stronger, tougher stem that prevents the larvae from attacking it. And he went as far as to say or suggest that this particular resistance trait was having a negative impact on the fly population in areas where the bearded variety was being grown. If we leap forward over 100 years into the first half of last century, we start finding a lot of studies being published by applied entomologists and phytopathologists, looking at factors that make certain varieties of crop plant more resistant to insect attack than others. By 1960, resistance traits were fairly generally accepted to be heritable traits that minimize impacts of herbivory, repel or negatively affect herbivores, and they are not necessarily adaptations to herbivory. At the same time, other entomologists were looking at the plant-insect interaction from a slightly different point of view. They were interested in the chemicals that determine food choice by insects that are attacking plants. And they were also contemplating how these kind of interactions may have evolved. In the 1960s and into the 70s, we start seeing this word defense coming into the ecological and evolutionary literature. But it wasn't really clearly defined. I think in the Ehrlich and Raven classic paper, they used the word defense twice, but never actually said what it meant. It wasn't until the 90s that a much more comprehensive, clear definition of this term was emerging, and the people that were contributing to the development of the concept and how it should be defined were largely seeing defense as an overall umbrella strategy encompassing resistance and tolerance. What I'm saying is not exactly new. Every now and then you do see people talking about it in the literature. By the turn of last century, defense could be fairly well accepted to be defined as heritable traits, minimizing impacts of herbivory, repelling or negatively affecting herbivores, and more importantly, they needed to be shown, demonstrated without doubt, to be adaptations to herbivory rather than other environmental factors. At the same time, during the 80s and 90s, other ecologists who were interested in how plants in certain systems with high herbivore pressure were managing to survive and reproduce. And they started using the word tolerance. Pellew in 1984 suggested that the resilience of acacia species to browsing was a result of selection for tolerant genotypes. And so we see this word coming in. Phytopathologists and applied entomologists had been using it, but it was now that the ecologists and evolutionary biologists were starting to use it. There were also a bunch of applied ecologists interested in rangeland management. They were seeing the interaction between plants and herbivores through the same lens as the phytopathologists. They saw the overarching umbrella strategy as being a resistance strategy encompassing defense and tolerance. Now you can see when terms are being used without clear definitions and used in different ways, it can cause a lot of confusion, especially for anyone who's not familiar with the literature. By the turn of last century, tolerance traits were being defined as heritable traits that minimize impacts of herbivory, do not negatively affect herbivores, and are not necessarily adaptations to herbivores. So we can see that these three strategies can be defined by the effects of heritable traits on both herbivore fitness and plant fitness, and very importantly, with reference to their evolutionary origins. There's a little table there that I put up showing similarities and differences between these three strategies, and you'll note that I've arranged them in a non-hierarchical way. A non-hierarchical arrangement of these different plant strategies and strategies of plants confronting herbivores is not a new thing. It does crop up now and then in the literature. What I want to put to you here is a simple flow diagram here, or a simple framework for understanding these three concepts or three strategies. But here I'm emphasizing the effects on plant and herbivore fitness and the evolutionary origins of the traits that we're considering. I think everyone in the room would agree that a rapid regrowth is something that contributes to a tolerance strategy. I don't think anyone would really disagree with that. What about the chemicals? There's a lot of chemicals, thousands of them. A lot of them we know hardly anything about. But of the ones that we know something about, you're either going to go down the left-hand side of that flow chart or down the right-hand side. If you go down the right-hand side there and say that they do not reduce preference or fitness of herbivores, then the next question is, does the trait benefit plant fitness under herbivory? Probably yes, and so, sorry, I'm getting confused. If you go down the right-hand side there, you're probably going to end up discarding that trait and saying it's irrelevant to what you're looking at. If you go down the left-hand side there and answer the first question with yes, because you know quite a bit about it, and the answer comes out as yes, then you need to ask the second question, is the trait an adaptation to herbivory? Almost always your answer is not going to be yes. It's going to be no. Why? Because we know so little about them, so the answer has to be no in the meantime, because it can't be yes. And so until we have more information about these chemicals, we conclude then that they're contributing to a resistance strategy, not a defense strategy. What about the structural traits? We all know, we hear repeatedly that large herbivores like to congregate in areas with lots of thorn trees, because thorn trees are supposedly more palatable and more nutritious than other trees. So the answer to the first question there, does the trait reduce herbivore preference or fitness? The answer has to be no. Does the trait benefit plant fitness under herbivory? The answer has to be yes, otherwise thorn trees wouldn't exist. So you end up concluding that those physical structural traits are contributing to a tolerance strategy, not a defense strategy. And so the three questions that I put and that I feel need a lot more attention in coming years and decades is whether the traits that we're interested in reduce browser fitness, whether they benefit plant fitness under browsing pressure, and whether they really are conclusively evolved adaptations to herbivory. Thank you.
Thank you, Peter. We've got a few seconds for question. No, we don't have. So thank you, Peter. Courtney.
Hi, everyone. Thanks so much for being here this afternoon. Yeah. Oh, perfect. All right, so I've actually already been up here one time this week, but I'm filling in for my advisor, Darren Berkey-Pyle, who wasn't able to make it last minute, but I'm always pretty pumped to share more of my research. So today, we're going to be digging a little bit into some of the more distinct impacts of elephant versus rhino carcasses. So far, we've really been focusing on elephants as part of the Megacarcass project, and here we have some new results to share with you. So before we get started, I just want to go ahead and thank all of our collaborators on the Megacarcass project, and especially Lindsay, Christian, and Hillary, who were all out here last February collecting these preliminary kind of first sets of data for the Rhino project. And then we also want to thank all of our funders and institutions that provide support, including Sandparks, Zion, and the National Science Foundation. Okay, like I said, so far, our research has really focused on elephant carcasses, and that's with good reason, right? So elephants are the largest terrestrial land mammals. Bulls can weigh up to about 5,000 to 7,000 kilograms. And so they have the largest carcasses, right? If you're trying to understand the impacts of the largest animals on terrestrial nutrient cycling, then it makes sense to start with the largest herbivores. Right, but one of the things that makes African savannas so exciting is that they actually have a whole suite of megafauna besides elephants, right? So that includes white rhino, black rhino, which aren't pictured, giraffe, hippos. And so there are a few different reasons that it's really important to go ahead and start incorporating these other megafauna into our studies as well. Right, and the first reason is that there's a huge gap in body size in the carcass literature. So I did a search of carcass literature that specifically looks at terrestrial mammals and measures their impacts on soil nutrient concentrations since the year 2000. And what I found is that in the kind of smaller ranges, up to about 1,000 kilograms, there's pretty good coverage, right? There's everything from mice, specifically deer mice, who weigh about 50 grams, all the way up to bison, who are averaging about 800 kilograms. But then there's a 4,000 kilogram gap before you get to elephants. And so part of what we're trying to do with this rhino research is start filling in those gaps a little bit. Right, and then the second reason that we're motivated to do this research is that differences in the density and distribution of carcasses could be modulating the impacts of these carcasses on ecosystems, right? So animals don't all die at the same rates, in the same distributions, and in the same places. So if we think about Kruger, we know that elephants take up a much larger proportion of the park in terms of their ranges, and we also know that their abundances are much higher than rhinos. But elephants are dying all over the park, kind of at pretty consistent rates, where there have been periods where rhinos are dying at much higher rates. So their densities in those years within those sections of the park could be really making up for some of that size difference. And so teasing out how those dynamics also interact with body size, kind of like an important next step in the research that we're doing. And finally, we know that carcasses of all of these different species are happening simultaneously. They're all on the landscape, they're all contributing in different ways. And so kind of understanding the cumulative impacts of these different megafauna and their legacy impacts after death means starting to fill in those gaps in the species that have been studied. Right, so to address this gap, we looked at white rhinoceros and elephant carcasses, all in the southern portion of the park, and all less than about 6 months post-mortem. So one of the things that became obvious very quickly when we started going to rhino sites is that you actually can't find the site visibly as easily once it gets a little older, whereas elephant carcass impact zones you can see for a pretty extended period of time. So we really focused on sites that were less than 6 months post-mortem and had to filter down our data set a little bit for a final sample size of 8 rhino and 7 elephant carcass sites. And so at each of these sites, we collected soil samples and grass samples, kind of at the center of the carcass site for the on-carcass sample, and then at 15 meters for the off-carcass sample. We took nutrient measurements for soil and grass samples, and then we also measured herbaceous biomass using the disc pasture-meter. And then we ran linear models to check for 2 things. The first is differences between rhinos and elephants and their effect sizes. And then the second thing was looking at whether elephants and rhinos differed from kind of those baseline controls. So just to orient you a little bit to the graphs we're using, right? So these are log response ratios, which is a measure of effect size. So the first thing we look at is we compare the 2 boxes for elephants and rhinos to see if those 2 differ. And then to see if they differ from baseline, we look at whether they're above or below the horizontal line, with below indicating that this metric was suppressed at the carcass site relative to the control, and then above indicating that it was enhanced. The first thing we found is that herbaceous plant biomass was strongly suppressed at both elephant and rhino carcass sites, but it didn't differ between the 2. And that actually makes a lot of sense. These are both huge animals, and they crush everything when they die, right? So you really just get these like above ground kind of dead zones, and especially at that less than 6 months interval. So cool result, not totally shocking. So the next thing we found was that soil ammonium was elevated at carcass sites and also didn't differ statistically significantly between elephant and rhino. Both were extremely highly elevated. Soil nitrate was also elevated at carcass sites, but here's where we start to see differences where the elephant carcass site had significantly greater soil nitrate concentrations relative to the rhino carcass site. And I put these mean and max values just to give you a little bit of context, right? Because I think the log response, I think it's the most helpful way to look at it, but sometimes I just want to show you how big these numbers are, right? So we're looking at a mean of almost 1,600 milligrams per kilogram of nitrate for elephants and a max of almost 5,300, right? And then for rhinos, that's a mean of about 1,200 and a max of about 2,000. So I looked up studies in Zimbabwe looking at termite mounds, and those have means about 200 milligrams per kilogram of nitrate and a max of about 1,000. So like these numbers are absurdly high, and I just want to make sure we capture that here. The next thing we looked at was soil phosphorus, and here we found that it was elevated at carcass sites for elephants, but not for rhinos. And there's a few factors that could be driving this, one potentially being differential kind of spread by scavengers, but we also just don't know for sure. But yeah, that's one potential driving mechanism. The next thing and final thing we looked at was grass foliar nitrogen, which was elevated at the elephant carcass sites, but not the rhino carcass sites. And this was one that really surprised me because the nitrate levels in soil are so high at both these carcass sites. But I think one potential avenue to kind of explore this further would be to start looking at them at larger age ranges, especially since we have these carcass sites marked and can look at them in like following years. Because I think some of these could potentially be playing out at different time scales in terms of when that nitrate is actually making it into the plants. But that's just kind of down the road. But at least the 6-month mark, we don't really see movement of nitrate into the plants at the rhino carcass sites. So just tying this all together, we found that rhino and elephant carcasses both release huge pools of nitrogen into savanna soils, but the effect is greater for elephants, probably because elephants are just bigger. Nitrogen for those elephant carcasses is taken up by plants, but we don't actually see that increase at the rhino carcass sites, which again I found quite surprising. And comparisons between the rhinos and the elephant carcasses fill a huge gap in our understanding of how the body size is impacting the effects of carcasses on ecosystems. And finally, this work is an important step in understanding how megacarcasses of different species contribute to nutrient cycling across whole savanna landscapes. Thank you.
Thank you, Courtney. You're far too kind with time. Any questions for Courtney?
Okay. Hello. In here. You can go first.
No, you go first.
Okay. Just a quick question about the Eurocloa species that you recorded there. This species tends to dominate the site when you recorded that in the field? I can't listen. I can't hear you, sorry.
She wants you to repeat the question.
Repeat the question, please.
Oh, yes. The Eurocloa species, they dominate the site?
Yeah, so Eurocloa trichopis, formerly Eurocloa mozambicensis, is really dominant at the carcass sites and just in general in the areas. So we were able to collect those consistently. Actually, a couple times we did Pan Max instead, because those are Panicum maximum, because those are pretty dominant as well.
That's interesting. Thank you.
Yeah.
Did you measure the size of the carcass site? Like how big the nutrient-rich patch is? And like, is that some of the reasons that you're getting more variability at the rhino sites than at the elephant sites? Is that they're maybe just smaller?
Yeah, that's a really good question. So what we did basically is we had 15 meters for our off carcass. Then we had 0.5 and 2.5 for our on carcass, and we averaged them to account for sometimes uncertainty in like what is actually the dead center of a carcass site. And I think that some of that variability might, like you said, be due to the fact that elephant carcass sites are just bigger, and we know that the effects extend probably between 5 and 10 meters based on some of the work that we've done, whereas at the rhinos, that could be smaller, so I think that could actually be like adding into that variability.
Thank you. Uh, Thank you, Courtney. Round of applause. Our next speaker is Helga.
Hello, everybody. Firstly, I would like to thank my fellow Megacarcasses mob members. I like that very much. And as you can see, this is truly a multidisciplinary, multi-institutional project in which we try and tease out the effects of these megacarcasses. And today I'm going to share a little bit on how they affect species richness and diversity. So decomposing large vertebrate carcasses are a natural disturbance. They are a localized source of nutrient enrichment, and they have strong impacts on ecosystem biogeochemistry, and then also the community dynamics of soil, soil microbes, large mammals, smaller mammals, and also vascular plants. But surprisingly, we have a much clearer understanding for marine ecosystems, where the megacarcasses of whales create these oases of biodiversity and productivity that can persist for a very long time. And surprisingly, we have limited knowledge on the individual carcass effects on the spatiotemporal heterogeneity in terrestrial ecosystems. And then this is specifically true for the African elephant, or the largest land animal. We know little about how the nutrient pulses from these megacarcasses affect savannas. And given that elephant mortality is a natural part of the savanna where they still occur, and annual mortality can total several hundred individuals, nutrient pulses of about 800 kilograms of carbon, 300 kilograms of nitrogen, and 125 kilograms of phosphorus are likely to operate at a landscape scale. So through our ongoing research in the Kruger National Park, we have been able to shed some light on their effects. For example, we now know that these carcasses increase local nutrient pools in savanna soil and plants. We know that these carcass-derived nutrients underpin floristically and structurally distinct plant communities, where we found dominance of prostrate-type growing species closer to the carcass sites, and more tufted graminoid-type species further away, which is, of course, an adaptation for intense herbivory and utilization. And then from talks earlier in the week, we also now know that they drive cold spots of biodiversity and hot spots of ecosystem processes such as soil microbial respiration and grazing. And then they also have impressive effect on the fungal community biodiversity. And then from Joao's talk, we saw that the parent material plays a really important role in the soil nutrient responses. So yes, they do play an important ecological role in terrestrial ecosystems. They are considered nutrient hotspots, since many of the species that we found in these sites were very palatable. They influence community composition and structure, and we now have even more emerging evidence that they do have long-lasting legacy effects. So we therefore anticipated that they will also drive local patterns in diversity and richness. So here we explored species richness, diversity, and evenness, and we did this at plant communities across megacarcasses of different ages on granitic and basaltic soil in the Kruger National Park. So we established transects of 15 meters each in the 4 cardinal directions away from the carcass sites, and we sampled vegetation in 1 meter squared subplots within which they were placed at various distances from the carcass, and within these we identified all rooted individuals up to species level, and also recorded percentage cover bare soil, bone, dung, litter, and so on. So very much in accordance to what Joao found is that soil type is a main driver of species composition at these sites. You can clearly see the granitic sites clustering towards the right and the basaltic sites clustering towards the left. Distance away from carcass was strongly associated with the second axis, and then age was negatively associated, but basaltic plots were generally associated with older carcasses with an average of 3 years old, and granitic sites with carcasses with an average age of 1 year old. So at a coarser scale, irrespective of soil type, we saw a general trend of significantly lower richness and diversity closest to the carcass sites, 0 to 1 meters away, and increasing richness and diversity with increasing distance away from the carcasses. At a little bit of a finer scale, so focusing on basaltic soil, it was generally the same trend, significantly lower levels closest to the carcass site with increasing levels with increasing distance away. And then interestingly for the granitic sites, maybe because they were a little bit younger still, or that this is a nutrient-poor soil, the effects of these carcasses were a little bit bigger, so significantly lower levels of diversity and richness up until 3.5 meters away from the carcasses. Yes, so we could clearly see suppressed species richness and diversity closer to the carcass sites, and this can be due to these extreme nutrient pulses from the elephant carcasses, and they create, of course, toxic environments for plants to be able to grow and persist in. And this is specifically true for ammonium, where we also found up to 31 greater levels of ammonium on versus off carcasses. And then also the physical trampling and soil compaction may only allow also for certain species to be able to persist at these sites. In terms of evenness, so conversely to richness and diversity, species evenness decreased with increasing distance from carcasses. And this could be due to the dominance of certain species that are maybe adapted to elevated levels of nutrients and increased trampling. For example, Irichlua trichoepis, which Courtney also mentioned, was one of the dominant species that we found at most of our carcasses. Yeah, so we can see from these results that elephant megacarcasses do drive local patterns of diversity and richness across basaltic and granitic semi-arid ecosystems. These extreme pulses of nutrients from these carcasses cause unfavorable and toxic conditions for plant growth, leading to suppressed species richness and diversities, and these effects seem stronger on the fresher carcasses on the granitic soils. And elephant megacarcasses do have long-lasting legacy effects on plant species richness and diversity. So elephant megacarcasses represent a relatively unexplored and unique disturbance type. Their effects on vegetation diversity patterns demonstrated here for the first time adds to this complexity and to the ecosystems that still support them. And then more importantly, the widespread loss of mega herbivores and their carcasses across the native ranges leads to a loss of an important driver of plant community dynamics and landscape heterogeneity, and especially the ecological legacies that their carcasses might create. Thank you very much.
Thank you, Elga. Any questions?
Thank you, Helga. I was wondering if I'm sure if in your group you considered the traits of those species that become dominant. So are they traits that's related to species that is adapted to using high nitrogen levels, or are they rather disturbance traits that can deal with that high disturbance? And I was also wondering, like in terms of legumes, did they increase or decrease, or didn't it really matter?
Thank you, Courtney. That's a very interesting question. We would have expected more forbs because they are adapted for increased soil nutrients, but surprisingly, we didn't find a lot. So I would say at this stage, it's more for the physical, like the physical adaptations for trampling and intense disturbance. Legumes, I can't think of the top of my head if we found any of them near or further away. That wasn't one of the traits that jumped out. Yeah.
Hi. Sodium is often thought as one of the nutrients that has a really big discrepancy between plants and the herbivores that eat them. Have you guys ever looked at concentrations of sodium near these carcasses?
Oh, Courtney can help me here, but I don't think we looked at. Will you help me, Courtney? Okay, they're super high. Sorry. Thank you, Courtney. So yes, we did look at it, and they are elevated.
Thank you, Helga. Got our next speaker, Olenirina. Sorry, it's Alice.
Hi, I'm Alice Jones. Thank you for coming to listen to this talk today. Today I'm talking about tree demography. So why is this important to study? So adult trees can persist in the environment for decades, and they can mask underlying instabilities. And understanding what the abundances across the different demographic stages are, we can detect early stages, revealing whether these systems are stable, perhaps they're undergoing densification or desertification. And this can then provide early warning signals for future ecosystem trajectories. I'll briefly give an overview of COSOR, which has been mentioned a couple of times in the last previous talks over these days. But this is a network of people and plots all across southern Africa in the Miombo region, which monitors biomass and vegetation structure over time. And I'll be using a few of their methodologies in this PhD chapter, and further information can be provided on the QR code. I'll briefly give an overview of this conceptual demographic model, and here we've got the more familiar life cycle where you have the seed, which then progresses into a seedling, and then a sapling, and then a more established mature tree, which can continue the cycle. And as Sally's already mentioned, in these disturbance-driven ecosystems with high levels of herbivory, fire, and frost, we also have this stage of Gullivers. And these individuals, although they may appear small and like they've only been there for the short term, have persisted in the environment for a long time. They have these large underground reserves, they have evidence of a root collar and signs of previous resprouting. So when you excavate them, you can see that they have been there for a while, although they look smaller in structure above ground. So the objectives for this chapter are to quantify the abundance of these different stages, a plot scale, and also to see how these abundances compare. This study took place on Gava Game Reserve, which is on the southern border of Kruger National Park in northern Namibia. And although the site does look quite small here, it does take about an hour to cross from east to west. There are very, very high megafauna populations here, so a lot of herbivory disturbances, and there's very low annual rainfall. We have 11 of these COSOR plots set here across the different vegetation types. I've tried to include some photos of the different vegetation structures we have. So at the top, I've got the Cataphractes shrubland, and this species is a spiny shrub about 3 meters tall. We also have vast areas of grassland, which have the occasional acacia tree, often heavily browsed. We also have parkland areas, which we've categorized as kind of grasslands, but with a few occasional mopani trees, and they're often a lot larger than in the other sites across the park. And then we have lots of areas of woodland, which are populated with species such as Coleosperma mapane, which is a really dense wood tree with really thick bark. But we also have other species, like a diversity of Commiphora species, and these are succulent species with very thin bark. So we have a whole diversity of different ecological strategies to survive in this environment. Coming on to the methodology of quantifying these different ontogenetic stages, the first thing we did was look at the soil seed bank, and to do this we used 50 by 50 centimetre quadrats, and we sampled within each of the 11 hectare plots across Ongava. We sampled at 3 different time points as well. We wanted to check for seed viability, so we used 3 different methods to do this. We tried using a dye method, and that also proved a bit problematic at times, and also a germination test. And over time, we kind of found that we could just sense if a seed was viable just by its appearance, if it's been predated by an insect, etc. We then looked at the seedlings and gullivers, and because of the destructive nature of the sampling method, we had to do this outside of each plot. I've tried to include some photos here, so hopefully you can see kind of the difference between the seedlings and gullivers. So on the top left here, we have a Terminalia prinidioides seedling, and it has the cotyledons present, indicating that it's a seedling and less than 1 year old. And then on the right here, we have it as a gulliver, and this is a multi-stemmed individual, so it's very clearly persisted in the environment for a lot longer than a year. And then on the bottom, we have an example of a mapane seedling. And then here, if you can just see, there is evidence of a root collar, and where it previously has re-sprouted the following season. We also used 2 of the CO-soil methods to quantify the larger vegetation structure within the plots. We had the small stems and shrubs protocol, which uses 3 transects across the plot, and this measures all individuals of over 30 centimetres and with a DBH of less than 5 centimetres. And then we also have the large tree protocol, and this measures all trees with a DBH greater than 5 centimetres. We then needed a species-specific method to quantify these individuals into saplings versus mature trees. To do this, we used data collected from Namibian herbarium records, and we recorded the heights at which individuals were reproductive, so taking note of whether they had fruits or seeds, and we then used this at 5th percentile height threshold to then quantify those individuals as saplings or mature trees. Coming on to the results. Terminalia prinidioides and Quercia acuminata were the only 2 species that were found across the 3 sample points. And the scale is logarithmic, so it kind of does skew the results a little bit, but I wanted to show the species richness across the sites. And the abundance in November 2024 was significantly higher than the other 2 sample points in 2025. And we found this quite surprising, as up until 2024, there'd been about a 10-year drought or of lower annual rainfall than normal. So then in 2025, when we had the rains, we expected this to be reflective in an increase in seed abundance, and we didn't find that at all. Now looking at the gulliver and the seedling layer, there is much greater species richness and abundance of the gullivers. So most of the individuals within this height threshold are gullivers, and we would expect that, as these individuals can persist for a lot longer as gullivers, versus a seedling is only going to be there for a year. And then when we compile it all together, these are the different ontogenetic stages compared across the different species. And there is a bottleneck between the seed to the seedling stage, and we would expect this. It's important to note that the seeds is just looking at what we found in the seed bank compared to if we had looked at seed rainfall, which just wasn't possible for the study. And then there's a gradual increase across those different ontogenetic stages with a higher level of mature trees at the end. So to conclude, seed production is a lot higher than the other stages, and there's also high seedling mortality, as we'd expect. Also within that seedling Gulliver layer, most of those individuals are Gullivers, and of those individuals, most of them are multi-stemmed individuals. So even if there was some confusion with identifying them as 1 year or old or as seedlings, most of them were clearly Gullivers. And this also matches other savanna research. And then most of the seedling abundances were much lower than those of mature trees. And this is also similar for savanna systems, but different to what you would find in temperate systems. So yeah, thank you for listening to my talk. Just to say thanks to all of the people who've helped me with this work so far. But yeah, cheers.
Thank you, Alice. Any questions?
What totally great data. It's so seldom, we see tree size class distributions all the time, but to add the seedling and the seed so that we get the full sense of the demographics is really interesting. I suppose I want to know from you, I was really surprised. I mean, I know an inverse J size class distribution is not expected in savannas, but I really was surprised by how few seedlings and Gullivers you had. Were you surprised? And if so, why not?
I don't think I was entirely sure exactly what to expect, but yeah, we didn't find that many, but also we did only sample at one time point of the year, so it would have been cool to have gone back at other time points of the season and seen if that differs. And in an ideal scenario, it would have been great to have tagged the seedlings and to see what their mortality rates are, and then add more data into our results.
I had the exact opposite thought to Sally. I was like, wow, there's such nice distributions of different size classes in many of your species that you wouldn't expect, because we so seldom find seedlings in savannas, and the recruitment seems to be very sporadic. But I was just wondering if you saw any pattern for classically more palatable species, although that looks like Muyombo if I look at the species, and things that's really quite not that nice to eat, like some of the combretums.
Yeah, that's a good question and something that I should definitely look into. Yeah, but I'm not sure. Thank you.
Thank you, Alice. Oli Nerina.
Hi everyone, my name is Nance. It is my first time to attend this conference. Thank you for considering my abstract. I am a PhD researcher in second year based in Madagascar. Today, I will present my preliminary results of my PhD chapter on the spatial patterns of forest diversity in Central Highland Savanna of Madagascar. Madagascar is recognized as a major biodiversity hotspot, both plants and animals, due by its diverse vegetation, ranging from humid forest to grassland that support distinct species. Grassland and savanna cover 65% of the total land area. Pioneer researchers classified all grassy biome in Madagascar as a secondary vegetation resulting from degradation of primary forest, so no conservation value, and wrongly managed by tree planting. However, recent research initiated by William Pond in 2008 challenges this perception, and some evidences demonstrate that grassland and savanna of Madagascar are partially derived from the pantropical savanna expansion in Miocene, long times ago before human arrival. Studies on savanna in Madagascar are very recent and limited into rangeland and graminoids flora, and forbs are understudied. Grassy biomes are among the threatened and vulnerable ecosystems in Madagascar, very few protected areas and terrestrial. However, there is a big knowledge gap on Madagascar's savanna and grassland ecosystem, limiting our ability to fully understand their structure, dynamics, and ecological significance. To address the knowledge gap in savanna ecosystem, specifically, this study seeks to describe the general pattern of forb diversity and endemism of savanna in the central highland of Madagascar, to understand how this diversity varies for the spatial scales, and identify the difference explaining this diversity. This research is conducted across 3 representative protected areas and surrounded villages dominated by grassland and savanna. Ankafupe Special Reserve, highly restricted protected area, unique habitat over the world of Schizolaina tampuketsana, a critical endangered species. EPT protected landscape harmonious, characterized by tapia woodland, a fair adapted ecosystem restricted nowhere else in the world, only in central highlands of Madagascar. And each massive protected landscape harmonious, managed by Q, is defined by its gallery forest and rocky outcrop, embedded within extensive savanna landscapes, support a remarkable endemic biodiversity. Ankafupe and Ipit are both managed by Missouri Botanical Gardens. Plant inventory is carried out by using the GDG plot method developed by Professor Caroline Lehmann in 2022, a standardized sampling method to quantify herbaceous richness and structure into 36 plots with 21 subplots each. We have recorded for each subplot the present absence of species, the vegetation structure, fair history, and grazing for each plot. Accurate plant identification is very important for diversity study, which are referenced into national and international existing collection herbariums, such as TAN, Tropicos, and POWO. What did we find? Savanna in Central Highland records 152 unique species represented into 6 different life forms. Of these, 60% are forbs, such as orchids, gyophyte, legume forb, and succulent. And of course, all forb species are seed-free plants and including 17 families and 56 genera. Most of the species are erect, about 80%, reflecting the fire-adapted species. The most representative families are Asteraceae, Fabaceae, Rubiaceae, Iridaceae, Malvaceae, and Orchidaceae. And the more diverse genus is Elricrhizome with 10 species. Forbs in savanna in central highland of Madagascar are dominated by native plants, about 48%, and endemic plant species, including the frequent species across landscape, about 30%. Introduced plants are represented by few species, about 9%, and all unidentified species remains in known status, which about 13%. Here are some endemic species with Aerosema procumbens and Camae crystalla territicola, frequently found on subplot across the landscape. And some apparate very spontaneous in the site, like Capharanthus lanceus and Aristia kitchingi. Ipit has the highest species richness region with 62 species, and Kafupe and Ichem have a quite similar species richness with 37 and 44. The 3 regions shared about only 16 species, and each region had their own species. And NMDHS shows also that forb assemblage showed a strong location structuring and a clear EGM separation. According to the species richness, Shannon and PLU indices, Ipit shows the highest level diversity and strong evenness across taxa, and Kafupe displays moderate but balanced diversity. In contrast, EGM exhibits low richness and low evenness. Simpson display a different pattern with EGM, showing higher values than Ankafupe and Ipit. suggesting that differences among locations are not captured by richness alone and may reflect shifts in dominant structure and the distribution of common and frequent taxa across subplots. Across all sites, there is positive correlation between graminoid richness and forb richness. But when the models were fit separately by landscape, the relationship was strongest in EPIT, positive in ITEM, but not supported in Ankaphupi. There is low dissimilarity species composition within Ankaphupe and high in EPIT and ITEM, with the highest total dissimilarity between locations. The biodiversity is largely driven by species turnover than nestedness. The GLM analysis reveals consistent directional response into Shannon and species richness. We can see here, for example, in Shannon diversity, a positive association with average grass height, plant cover, and bed cover. Negative association with Thamesin's fire, no clear association with grazing index, and standard deviation grass height, which predicting that species richness are primarily respond to vegetation structure and post-fire dynamics rather than location. In conclusion, this knowledge shows a high diversity of forbs, more than half of total richness, and dominated by native and endemic species, supporting the other evidences for natural and ancient origin of grassland, so deserve conservation and appropriate management. This study have captured 1 year of the forb diversity across space. The temporal pattern in progress will complete the patterns of forbs in Central Highland of Savanna of Madagascar, which will capture of 3 years of study. Other parts of Madagascar need a further exploration, particularly the southern and the western, for a better understanding of Madagascar grassy biome. I want to acknowledge all the persons involved in this research, my supervisors from Royal Botanic Gardens Kew, Royal Botanic Garden Edinburgh, and in the University of Madagascar. All of my colleagues in Madagascar, including the local population in studies area, the Royal Botanic Garden Edinburgh and Kew, and University of Barcelona. And also the other partner institute, Missouri Botanical Gardens, Madagascar, and thank you for your interest to my presentation.
Any questions?
That was really excellent. I really enjoyed it. Good job. I was wondering, your colleague that showed us the history of the soil carbon the other day, there were some of those sites that have been grassland for a very long time. And I was wondering if these sites that you worked at, did you work at some of those that he has shown that's been grassland for millennia?
What is your question, please? Sorry.
I don't worry. You did an excellent job.
Sorry.
Thank you, Colin. Thank you. Thank you. And this is our last one. Andrew, take it us out.
Thank you. Thank you, Lufuno. And yes, it is the last talk. So congratulations to everybody. Give yourselves a round of applause. Thank you. Congratulations to myself. I waited all this time to give my talk. I realize I've probably been relegated to the back because I'm actually talking about forests, but that's fine. We're talking about not just the forest, but the open areas in the forests. And I first want to say that this talk is actually the work of an honors student, Julia O'Donnell, which you can see over here. She did this fantastic project last year. and I'm honored to be able to share some of her results thinking about natural edges in Congo Basin rainforests. And when most people think about the Congo rainforest, they probably think about trees and forests. Now, people in this group might be different and think about other things. And what we think about a lot in Congo are a lot of these open areas, the savannas and the bayous. You would have heard from Catherine Angier earlier this week and Evan Hockridge about some of these dynamics. And that's what we're really focusing on, and the dynamics between these open areas and these closed forests. And what also is interesting about the Congo Basin is not only is it this mosaic ecosystem in many places, but also has large animals that are very similar to what we find here. There's some forest elephants down here. If you can see them, there's some forest buffalo over here. Very similar species to what we find in places like Kruger. And so we're trying to understand what they do in shaping these ecosystems. And one of the ones that we focus on, which you would have heard from Evan earlier this week, are these bayous, these large natural forest clearings that just appear throughout the Western Congo Basin. And we're interested in studying these in many different ways, but we're also interested in the edges of them. And as you can see, there are these open grassland ecosystems or grasses and sedges that come against these really stark boundaries and into these tropical forests. Now, forest edges get a bit of a bad rap around the world. I'm sure most of you know this, and this is a paper that came out about 10 years ago, a very well-known, very well-cited paper, looking at forest edge extent around the world, showing over here all of the forests and how they've been fragmented. This is talking about the distance from the edge. Anything in the reds and yellows is within a few hundred meters of an edge. These are some examples showing how the Brazilian Amazon and the Brazilian Atlantic forest are really fragmented and have so many edge effects, and generally how these edges are really negative. When you think about tropical forests, this is some work that came out just last year or this year, thinking about all of these edge effects around tropical forest edges, like that example over there, how you have reduced canopy height, reduced carbon. Many things are reduced and compromised around these areas, and we actually create these ecotones, and we have these edge effects from the edge into the forest. But when we think about forest edges more broadly, they seem to be very negative in the tropics. In temperate forests, there are actually a lot of benefits that come with forest edges. This is work from North America, showing all these different types of communities of trees and forests, and the red ones are trees near edges, and in every kind of community, the trees are actually larger and bigger because they have more access to light. And so the understanding of forest edges is actually quite context dependent. And the other important thing is all of these edge studies, or the majority of them, are focused on anthropogenic edges, things like roads or clearings that give us really negative effects. When it comes to natural forest edges, we know very little. And this is another picture of a bay showing this edge, which is a natural edge, which has probably been there for at least hundreds of years, possibly millennia. It's been there a very long time. It's very stable. It's not associated with these human pressures that anthropogenic edges are. And as Evan was showing you earlier in the week, they're also frequented by these large animals. And that's important because in most anthropogenic edges, large animals are not associated with them because they get hunted or they get disturbed. And so we wanted to understand the dynamics of these edges and how these animals are actually shaping them. So to do that, we collected LiDAR data using a drone across these bias complexes in the Republic of Congo. We mapped 127 of them. Here's an example of what some of the LiDAR data might look like. As you can see, this very stark edge. Here's just a cross section going from the edge of the bay, and you can see how these forests form these really strong boundaries. And so what Julia did is she went and she looked at these 10 different complexes around Kruger National Park in the north of the Republic of the Congo. And they also can be broken into these two types. There's elephant bayous and there's gorilla bayous. I don't really have time to get into this too much, but there's different kinds of bayous, and we looked at how elephants and gorillas and these different mammal communities shape these edges. Zooming in over here to what the study methodology was. If we start over here on the right, this is the LiDAR data. We basically took the edge of the forest and we broke it up into 1 meter increments as far as we could go, a few kilometers, and looked at different structural metrics within this using the LiDAR data. And then we also broke it into 20 meter bands, which is this dark line over here, and looked at how many canopy openings there are before and after this distance. We combined this with field data and some forest plots where we went and characterized all of the diversity of the tree species as you move away from the edge. And so I'm not going to show you all the results because there's quite a lot of them, but this is an example of 10 different patches that we worked on, 10 different complexes of bayous. And what the approach that we took is that this is all mean canopy height on the y-axis. This is distance from the edge of the bay. These are all these points represent those 1 meter rings. And the dotted line over here represents a significant threshold where that value changed. So in other words, this bay over here, canopy cover was canopy height was much lower, up to about 100 meters from the edge of the bay, and then it became uniform. And that's what you're seeing through all of these. You kind of see that generally there's a decrease in tree cover closer to the bay. Similarly, for gap density, this is the same kind of result. You tend to get fewer, in some cases fewer gaps, and in most cases more gaps closer to the edge of the bay. If we put it all together, this is now looking, each line represents an individual complex of bays, which is multiple bays, 127 altogether. We looked at canopy height. This is the coefficient of variation of height, so really structural heterogeneity, canopy cover, and the canopy gap density. And through all of them, you see that there's these very significant edges or edge effects close to these bays. And what you can see here is you can see also the distance, the average distance across them, which can be hundreds of meters in extent. And we'll get into that a little later. So to summarize these, if you look at this top one, this is thinking about the magnitude of edge effects, the percentage change before and after those thresholds. And so canopy cover in general is depleted by about 20 to 30%, whereas gap density is up to 50% or higher closer to the bays. And then this is looking at how far into the forest we go. And you'll notice that canopy height is about 100 meters. Canopy gap density on average can be about 200 meters, in some cases up to, say, 500 meters, which contrasts a lot with anthropogenic edges, where normally we see these effects attenuate a little bit closer, maybe about 100 meters for all of these, whereas these can go several hundred meters into the forest. We then want to know what's driving these patterns. What is causing the difference in the magnitude of effects? And so we had camera traps around each of these bays. And from that, we looked at elephant density and elephant visitation. And then we also looked at how close together they were by mapping them with the LiDAR data. And what you can see clearly, the bay clustering, so how close together they were. So this axis down here, the more smaller the number, the closer they are together. And elephant density explained 70% of variation in the magnitude of edge effects, which is pretty phenomenal, actually, especially in ecology. And you can see that where you have closer bays are closer together, you get bigger effects for all of these metrics. And where you have high elephant density, which is over here, you get also a larger effect. Whether it's negative or positive, these are the things that are driving these effects. And if you think about why that might happen, well, elephants, as Evan showed you earlier, are really attracted to these bays. So this is GPS data from a whole lot of collared forest elephants across the region. All of these areas where they concentrate on are concentrated on a bay, and they can walk 60 kilometers, spend a couple of days in a bay or a few hours and then leave again. So they're coming to these bays and they're frequenting them. And when they come, they're trampling, they're digging for salt, they're eating the soil, and they often cause these kind of diggings underneath the trees, which then cause them to fall down. And so you can see why this effect is so strong. When it comes to clustering, this is a map of bays from a paper that we published a few years ago, and you can see how they form these clusters across the landscape. And the closer, the tighter these clusters are, the bigger the effect. And that's probably because elephants are coming and moving around these bays if they're all close together and sort of extending the effect across them. We also then looked at biodiversity using those field plots. So these are plots of tree diversity, looking at species richness, abundance, and the Shannon Diversity Index. This is before and after the change point. And in every case, the diversity of these trees is higher before we get to that edge point. So diversity of trees is actually higher closer to forest edges. When we look at the composition, this is looking at the edges in the interior forest in the dotted line and the edges or this ecotone in the solid line. Not all that different in composition, but obviously different in abundance and evenness, which is why we see these different diversity results. And then if you look between the elephant and the gorilla bars, which we don't have time to get into, you can actually see quite different communities between them. So what do we sort of think about all of this when we think about this broader question about open and closed ecosystems in Congo and the role of these biotic drivers in shaping them and how they compare to anthropogenic edges? Well, the first thing we see is that there are these significant effects on all of these structural metrics that we measured. Canopy height, structure heterogeneity, cover, gap density, which is not unusual. We get that with anthropogenic edges, but they extend hundreds of meters into the surrounding forest, actually further than most anthropogenic edges. And we think that's because, like I said earlier, elephants will come and they'll actually spend a lot more time in these edges and actually extend these effects far more than they would at a natural edge where there's a road and there's traffic or there's hunting or all these different kinds of things. Like I said, by clustering and elephant visitation explain most of the effect, which I think supports this notion that these natural edges are attractive to them. And what's really interesting is that biodiversity is increased around them, whereas at anthropogenic edges, it's a huge concern because they actually have lower diversity around them. And so when we think about managing these forests or understanding these forests, these edges play significant effects in the biodiversity value and the structural heterogeneity of these forests, which contrasts a lot with these anthropogenic edges. So thank you.
Thank you.
Hi, thank you very much. Super cool talk. I was wondering whether you looked at savanna forest edges rather than by forest edges, and if not, what would be your intuition about, similar patterns there?
Was your question about, what is the first part of the question? Which edges?
So savanna forest
Savanna
edges.
forest edges. So yeah, we've done quite a lot of work with that. Evan's doing quite a lot of that with his PhD project. And they're really quite different. I think these bios are sort of isolated. They're far away from the savannas. They're out in the forest. They have these really harsh boundaries where savannas are more graded. So we have done some work. There are some differences. We can chat about it, or you should really find Evan and talk to him about it.
Thanks, Andrew. So I'm fascinated by the geometry of these buys, and in particular, what the determinant is of the diameter of a buy. So if you've got the elephants coming into the buy and they are having an effect on creating that edge, is that edge going to be extending inexorably into the forest, or is there some balancing factor that retains a set diameter?
So we don't know, but we should know in about 2 weeks' time, when Evan finishes his analysis. So yes, that's one of the questions we're asking, is what happens with all of these elephants? Are they extending them? Are they stable? Are they decreasing? There's anecdotal evidence from Democratic Republic of Congo where forest elephants have been locally extirpated, that these buys are closing in. And there's some satellite imagery that would suspect that. So we think that elephants are key to maintaining them at least. I think there is a biotic and abiotic interaction because they're in these hydrological low points. They're very rich in sodium, which is what attracts the elephants. But what this role between biotic and abiotic is something that we're looking at through the salting experiment that Evan spoke about earlier in the week, and we trying to understand whether elephants are important for the maintenance or the expansion of them.
Thank you, Andrew. Can we please have the last round of applause?
Thank you very much, Lufuno, and thank you to the speakers of this last session. So we have 2 last items on our program, but before we get to Ernest, who's going to be doing the wrap-up, I have one lucky draw going. Andrew, don't run away. So it's supposed to be a lucky draw, but just because I can, I'm changing the rules. So I want to give this to you, Andrew, to say thanks very much for sticking it out and for being the last speaker of this conference. So thank you. Thanks, colleagues. So now, as mentioned this morning, we don't have the choir that's going to sing for us. So before Ernest gives the wrap-up, he agreed to sing. No, I'm joking. Thanks, Ernie. Can you come and do the wrap-up for us? Thank you.
There is no way that we are in Kruger National Park and we can't have a choir. Anyone risky to join me? Sipokazi? We'll figure it out. Lufuno, you want to join? Okay. You are very brave. I've never had a song in my life. So, but come. Okay, let's go. Who's going to join? Where's the mic? Everyone will have to sing. Da tentekele.
Sana marena.
Da tentekele. Da tentekele. Kuwa buhadi sana marena. Here we go. Good afternoon again, colleagues. I know it's very late, so I'm not gonna delay you with the closing remarks, but I just have few reflections starting on Monday until today, and mostly on the historical growth and the shift in leadership. During the opening by our GM, Kathy, there was a reflection that this meeting involved from a very small group focused on the rivers in the early 2000s to a global event with almost 230 delegates attending the Savanna network this week. So this is a very significant growth for this platform. And also in terms of shift in leadership, historically, men used to dominate this kind of network meetings. But however, recently we have seen that shift in representation and a very deliberate increase in female presenters. I saw a lot of red caps, mostly young female researchers. And during the opening, the first session, I think it was on invasion, species invasion. I think out of 10, we had 2 posters, and 7 out of those 8 presentations were young female researchers. So I think this is really great achievement for the Savanna Network meeting. And I must say, in terms of the organizing team, I'm not bragging, Sandparks has done well in terms of leadership. I saw a picture of people who started this. I think I saw 3 men. Can I see where we are, colleagues, if you can stand, the organizing team, our GMs who are here. I can see you are here, Judith. Tez, I saw 2 general managers at the back, Kathy, Nkabeng. Where are you guys? People need to see you. And what Sandparks is doing in terms of leadership. I think that is a good reflection. I'll move from that. I was just, bragging about our transformation in Sandparks. I couldn't help but, reflect on the most recent event related to biodiversity conservation, because the Savanna Network meeting it's all about conservation. And recently, late last year, we had the National Biodiversity Assessment Report, and that confirmed that the biodiversity remains under significant pressure from climate change, unsustainable land and sea use, pollution, and invasive species. And also that wetland and estuaries remain amongst the most threatened ecosystem types. And when you looked at all the themes this week, I think they were all aligned with what is stated in the National Biodiversity Assessment. So I think we are in the right track. Recently, in February, there was a 6 subsidiary body on implementation of the Convention of Biodiversity that took place in Rome, Italy. And this touched on biodiversity conservation, biodiversity finance, resource mobilization, and capacity building. So I think also in that front, we're doing well in terms of biodiversity conservation. And of course, the Savanna Science Network meeting will surely be one of the highlights for this year's events, reflecting on the importance of biodiversity conservation and all the discussion that took place this week. The upcoming Kruger National Park centenary celebration will surely be a remarkable milestone in the history of conservation. So I'll just reflect on few of the themes that we discussed this year, and among those, the take-home message for me is that the future of savanna system relies on data-driven and collaborative approaches. And this is from the role of invasive species management to use of AI and satellite technology for monitoring species. And we also touched on the need to move away from species-focused approach to more holistic ecosystems. And also, in general, the discussion on fire ecology, freshwater ecosystem, and the balance between the conservation and land use beyond park boundaries are also essential for creating holistic management strategies. We also, I think, touched on the living landscape and SandPark's mega living landscape approach. And this, for me, it's really a paradigm shift and also a sensitive one as it touches on ownership of land, which is central to communities and livelihoods. And this is not business as usual for SandParks, as your usual preservation approach, but this is an inclusive conservation focusing on transformation, equitable access and benefit sharing, sustainable use, and enhanced biodiversity conservation. So we are actually emphasizing that we are going outside our boundaries. The impact of habitat and transformation on biodiversity also underscores how woody densification and shrub encroachment impacts biodiversity across the savanna landscape. And there was a lot of discussion also on how mega herbivores also shape this landscape. The ecological processes that sustain the savanna ecosystem, particularly the interaction between these herbivores, plants, and also soil microbes, are also key in this dynamic, complex ecosystem. We cannot also forget about the discussion that took place under freshwater ecosystem in the savannas, as these are crucial, particularly the threats posed by invasive species and environmental changes such as floods and recent drought that we realized recently. In terms of the drivers in the savanna ecosystem, various factors such as herbivory, fire, climate change remain the driving forces shaping this landscape. And we cannot overemphasize the need to integrate cultural heritage, including archaeology, indigenous knowledge system in our science engagement. I also saw one of the engagement just opposite where we are, where the managing executive of Kruger National Parks was engaging with a group of traditional healers. So you can see the direction that SandParks is taking. It's not your normal science and theories, but we're going out there and making sure we engage with all the stakeholders. And looking beyond these park fences, address the challenges and opportunities of managing landscape beyond boundaries of protected areas, including human-wildlife conflicts and land use. And as we wrap up, remember that science may not have all the answers, but it definitely knows how to ask right questions. Sometimes those questions lead to new discoveries. So I'm sure from this week deliberation and engagement, we will see a lot of projects coming in this platform next year. Let us remain committed to collaboration and innovation and conservation of this important ecosystem for generations to come. Thank you for your participation, engagement, and thanks again to the organizing team, the presenters, the session facilitators. Enjoy the rest of your stay in the Kruger National Park and have safe travel to your different destinations. I was about to drop the mic, but I can't. Thank you very much.
So I think I've got the last word. So I would just ask our AV guys if they can just put up the slideshow, our slideshow from last night. So while I'm going through my couple of words on my sheet, you guys can just have a look and see where we actually came from. So really, it's great to see so many of you here still here after these 4 packed days. So 23 meetings, wow, that's a lot of years. A lot of talks, a lot of posters, and a lot of hard work. When this whole thing started, it was just a small group of passionate people coming together to share their love for the savannas and the ecosystem. Fast forward to today, and here we are. This meeting has grown into something really special. It's safe to say that the Savanna Science Meeting has carved out a pretty important spot in the scientific world and our yearly calendars. So here's to many more. But all of this wouldn't have happened without a lot of hard work from some incredible people behind the scenes. So firstly, to my small team, you guys know who you are, who've been working tirelessly for months to make this meeting a reality. The planning, the coordination, the countless details you've taken care of, from advertising the meeting, sorting through the abstracts, which we also always don't agree on, and a lot of back and forth with changes going back and forth, as well as sending out links, packing bags, and all of that. Thank you for your dedication and your commitment. You are really the backbone of this event. And of course, to everyone else that's been busy assisting with this week, which includes helpers at registration, timekeepers, people doing the roaming mic, session chairs, and the bingo planners, thank you. You've been the friendly faces that have guided us through every step of the meeting and assisted with the small hiccups that we ran into. You really made everything run smoothly, and we couldn't have done it without you. To Kathy and Ernest for starting and ending the meeting, thanks very much for your words of wisdom and your inspiration. Then to our 2 guys there on the right-hand side, to Liam and Smash, thanks very much for helping us be able to see and hear the speakers, as well as keeping our online viewers happy. And even though we lost our 2 aircons on the roof being airlifted out on Tuesday, you managed to keep us cool for the majority of the time, so thanks very much. And then, of course, the catering team, to the Skukuza Lodge and Cattle Baron. Wow, you did a great job. You kept us really well fed, and sometimes I think too well. From the snacks in the morning, the teas and coffees, the meals, the evening meals, I think we can all agree that the food this week was top-notch, and that's definitely something we do appreciate. Then a special thank you to all the colleagues who assisted with adding some special K&P 100-year additions to the meeting. Some of our ideas were very last minute, but they were rapidly taken up by many of you and run with. So thank you very much. I think that did give this meeting a little bit of an extra flair, especially to John from the Honorary Rangers for helping us with the budget for the caps. If he didn't supply that, we wouldn't have been able to do that. Big thank you to Jackie and Mpumalanga Promotion. She's also a blue cap, and she's walked this walk with us for many years. As you can all ascertain to, if you ask Jackie anything, it gets done, and it gets done on time. So once again, she did an awesome job, and we hope that she will continue our partnership going forward. Then to all of you speakers for sharing your knowledge with us. You inspired us with your research, you sparked great conversations, and have given us much to think about as we move forward. So to you, Greg, it sounds like I'm going to have to trade in ChatGPT for Claude, but please don't say anything because Chat's definitely going to be very offended. Finally, to all of you, the delegates, thank you. So whether you are a red cap, a blue cap, or any of the colors in between, you're the ones who make this event what it is. Your questions, your curiosity, your engagement, that's what drives the meeting and keeps it so exciting. We hope you're walking away with new ideas, connections, and some fresh inspirations. So before I wrap up, I want to make sure that you all know that we'll be back next year. So same place, same time, we'd love to see you here again. So mark your calendars, first week in March, for the 24th Scientific Savanna meeting. We know we might even have a few surprises up our sleeves. And really, we might even be able to get the Wi-Fi operational before the meeting. So in conclusion, it's been a pleasure having you all here, meeting some of the new red caps and chatting and catching up with many of the older colleagues. Kruger is 100 years, which highlights the work that we are doing here is so important, and it's only possible because of all of your efforts. So with that, I'll wrap up and close the meeting. Have a safe journey home, stay inspired, and we can't wait to see you next year. Thank you.